Well, welcome. I'm so happy to see almost everybody here. We have 4 people who are stuck and will be joining us throughout the day today. I would like to thank Marinka, who helped with the scientific program organization, and then I will come back to thanking all of you for the amazing work you pulled over the last several days to get the presentations going. Before I bore you with some logistical updates, I'd like to give opportunity to Patricia and Pat, I'll introduce them in a second, to give a couple of opening remarks on behalf of Notre Dame. So let's start with Patricia Clark, who is Professor of Chemistry and Biochemistry and Associate Vice President for Research.
So thank you very much to Tiana. My name is Patricia Clark. I'm a professor of chemistry and biochemistry, and I also have a role as the Associate Vice President for Research. And so I'm a research active faculty member. I'm an experimentalist, but one who has benefited greatly over the years from collaborating with computational people of all stripes, including network biologists. And that's really brings me a lot of joy to be able to welcome a workshop that really is trying to find those and deepen those connections between experimental results and computational models through network biology. So that idea of integrating computation and experiment was baked into me since I was a postdoc in the lab of Jonathan King at MIT. John, more than anybody I personally know, had an amazing ability to bring together people of really diverse backgrounds. So when I was in his lab, there was, in addition to a biophysicist like me, there was geneticists and mathematicians and chemical engineers and marine biologists all working towards a common goal. So it was a really profound way to see firsthand the power of bringing together a diverse group to tackle a really challenging problem. And since I've been at Notre Dame, moved here directly from my postdoc 21 years ago. At that time, the university was making a lot of noises about wanting to increase its research footprint, and it's really been remarkable to see the changes that have been wrought over those 21 years. And I know that you guys have a really packed agenda for the next day and a half, but I hope that I know you took a tour yesterday afternoon. I hope you have some other opportunities to both see and hear from Notre Dame, folks here at Notre Dame, about the really amazing investments that have been made in infrastructure, in large part to really facilitate those collaborations between experimentalists and computational folks. So the university is currently in the early stages of its next forward-looking 10-year strategic plan, and I'm delighted to say that those investments in research have only been reinforced and expanded. So I think we're at a really exciting spot. Over the past 10 years, research expenditures at the university have more than doubled, and we're, I think, really trying to have that doubling again happen over the next 10 years. So that includes things like investments in infectious disease, everything again from very sort of basic immunology and pathogenesis to computational modeling of disease progression and community spread and so forth. And underlying all of that is a really, really robust Center for Research Computing that supports both computational investigators and also experimentalists like me who are really, really trying to get into computational research. So again, I know you have a really packed agenda for the next day and a half, but I look forward to talking with you more. I'll be at the dinner tonight, and I hope you have a great workshop. Thanks.
Thanks, Patricia. See you tonight. Now, Pat Flynn. Pat, Patricia, I promise this was just coincidence. Pat Flynn, Professor and Department Chair of Computer Science and Engineering.
Thank you, Tiana, and welcome everyone, finally. I want to compliment the organizing team, Tiana and her colleagues, for perseverance in the face of unusual obstacles, and I'd like to thank this community for taking up the delayed invitation to come to Notre Dame and share your thoughts. When I think about a workshop like this, I'm tempted to call it, as a person most of whose experiments are in silico, I tend to think of this as sort of an alternate wet lab, right? We're together largely in person in order to make things grow. There are lots of ingredients, and getting together is the yeast that makes it happen. And so my fervent wish is that that's how this workshop will be, and it's up to you to make it happen. So that's my charge to you, is to take the greatest advantage you can from the circumstances that we're in. And those of you who are online, there's still many ways to participate, and we hope you can take full advantage of that as well. I apologize. I will say that I have just the tiniest amount of street credibility in this community, because when I was a newly minted assistant professor on the faculty at Washington State University, Zoran Obradovich was one of my colleagues, and Zoran is, I think, reasonably well known to at least a part of this community. And so he and I worked together with Keith Dunker for a few years on protein folding, and it didn't lead to much, at least not for me, but it led to a great deal for him and for them. And so that's where it ends. Most of my work is in computer vision, image understanding. There are some potential overlaps, no doubt, and maybe we'll have an opportunity to explore that in the future. But as it is, this is a well-knit community with a clear theme for the workshop. I hope you make the most of the opportunity. And I also hope you have the opportunity to brave the heat a little bit, and if you haven't gotten out to see our beautiful campus, please do so. There's many opportunities to see beautiful things and meet some interesting people. Summer classes start today, so you could run to the classroom building and see what's being taught. The department is 32 years old. We're celebrating a binary birthday this year. I am the department chair for 2 more weeks, and my colleague Jane Cleland Wong is taking over in 2 weeks. The department has grown and grown, and we're a top 5 department at the university, and one of the reasons for that, probably the reason, is because we've hired excellent people like Tiana. And so as an old-timer here at Notre Dame, I'm very happy to see the progress that we're making. Patricia alluded to a little bit of that. And workshops like this, sponsored by excellent scientists who have a voice in the community and are interested in organizing activities that make the community better, are really welcome. So thank you for attending. I hope this leads to many, many discussions and collaborations. Enjoy your time here at Notre Dame.
Thank you, Pat. Okay, I have so many thanks to so many people, including all of you, for being here and being brave to take on the journey of traveling by planes in the crazy time we live in. Thank you all for complying with COVID protocols. I think that's, and keeping us all safe while we're here and when we go back, or when you go back, I should say. Suggestion I got is that maybe whoever is presenting, the person should be standing in front of the mic. Maybe take the mask off for the purposes of video recordings, but I will leave it at your discretion, whatever you're comfortable with. Can everybody hear me well with the mask on? I would prefer to keep it on simply for the reason of protecting those sitting right in front of me. But again, I think when the teams present shortly, if you want to come out one at a time when you present, or stand here, whoever is presenting should be standing in front of the mic. That's kind of the only rule, and everything else is up to you. Thanks to NSF who funded this, so we can have lots of scientific discussion, but hopefully some fun as well. The purpose of the workshop, which I kind of shared with most of you when we did lots of Zoom meetings over the last 2 weeks, is to help understand where the field of network biology is. And this is funded mostly from algorithmic perspective. So the goal is to figure out what are, not necessarily just algorithmic, we are interested in applied questions as well, but really the interplay between algorithmic and applied challenges in the field of network biology and where the field is headed. And I think the goal is to maybe inform NSF of what they might want to consider for funding in the future. So I think we have a pretty responsible job ahead of us. I hope this initiates new directions for all of us and possibly cross-institutional initiatives, future grant applications and the like. And shorter term goal will be I'm responsible for providing a written report to NSF, like I'm sure we all love to do those. But in the process, summarizing the whole discussion in the workshop, but in the process I've decided to try to make it into review survey paper, and I will give opportunity to all of you to participate in that should you choose to, but no expectations, of course. Just because we're all busy, but you already did all the work, so why not, right? Could we bring out the schedule and share it with the in-person and online audience? So Marinka, and actually this was funded in 2019 to be held in 2020, and we know how that plan went. Marinka and Jimbo helped me figure out the 5, these are not exhaustive topics in the field of biology, and many of them overlap at a certain extent, but we just wanted to define some set of topics, and you've seen them already, but you can see them in red shown on the slides. For each topic, our goal is to get an idea of key challenges and current solutions to those challenges, and what are the new challenges that we need to think about. The only change in the schedule as of now, which happens because not everybody is here yet, is that we will swap the first topic, inference and comparison of biological networks, with a third topic, higher order networks. So higher order networks will start first. And that's the only changes of now until I know of any potential delays by any people who are still not here. So just an FYI. Thanks to Harriet and all the staff involved into this, Harriet made my life a little hard, but a lot easy. And she has made sure that everything hopefully will go logistically smoothly in terms of meals and breaks. And so thank you, Harriet, and thank you to our tech support and absolutely all the staff involved into this. So everything will be happening, everything today and tomorrow is happening in the hotel. We are here until dinner time. Well, there's a break, which I figured you might need a nap. I might need a nap in my office, which is only 2 minutes away. But everything up to the end of scientific program, 5 p.m. is here. Lunch will be we'll announce it when the time comes, but literally behind this wall. And then for dinner, we'll be moving into Roar's Bistro. If you were at dinner yesterday, you were at Roar's restaurant. So you just, instead of taking left, you go right at the desk, checking, well, not checking desk for the hotel, but for the Roar's. So everything is happening here. And we thought about your departures tomorrow. I know that many of you are leaving. Nobody's leaving before 1 p.m., if I understood this correctly, but many of you are leaving between 1 and 2 p.m. The good news is our airport is tiny, and people who travel through it frequently arrive at the airport 40 minutes before their flight. If you really want to be comfortable, 1 hour is fine. So you don't need to panic about being at the airport like 2 hours in advance. But I suggest that you arrange drive to the airport as soon as possible, because I don't know how reliable taxi service is to schedule. So the sooner you arrange it, the better. If need be, I'll be here, and I can obviously take 4 people who are in the biggest rush. We provided lunch tomorrow in kind of quick, you can grab it packaged, take with you, so if you're in a rush, and we can touch base, but we've adjusted the lunch time so that all of you should be able to make it to the airport without absolutely any problems. I think that's all I have to say, other than we have extra 10 minutes of break time, so grab some more food, chat with each other. We haven't seen each other in 3+ years. And then let's start at 9 as scheduled with higher order network analysis. Thank you. Then I think we're ready to start with higher order network analysis. If you want to do one at a time or stand up, it's 90-minute session, so I don't know how smart it is that everybody stands up the whole time. But say again? Yeah. So up to the presenting team, how you want to logistically organize that. Thank you. All right, I'll lead the way.
All right. Yeah, so this presentation is going to be kind of contributed by everyone in the group. So we'll have a bunch of people kind of coming up here. I'm really excited to be here. Thank you for the organizers for planning this. And today we're going to start by talking about higher order network structures. And this was supposed to come later today in the afternoon after we talked about a lot of network algorithms and applications. But maybe it's a good place to start. When we think about higher order network structures, we think about relationships and patterns beyond pairwise graphs, so the pairwise relationship in graphs. And that can kind of take 2 forms. The first one is thinking about identifying patterns in graphs, and these are undirected or directed graphs, weighted graphs. And we can have, it's okay, they're kind of hard to see, so I'm going to try and move this up. All right. We can have, examples of taking a graph and looking at bipartite representation of that graph to identify groups. Motifs, network motifs, so finding instances of subgraphs in that graph. And also looking for cliques. This is just kind of an example. We're also going to talk a little bit about graphlets. So we can look at finding patterns in graphs. Another type of higher order network structure that many people, I'm sure, are familiar with are the idea of clustering, community detection, module detection. This is just a figure of a bunch of different algorithms from the disease gene module detection DREAM challenge that was published a few years ago. And also another idea of kind of higher order network structures that falls under this looking for patterns in graphs is simply taking an existing graph and trying to kind of elucidate or identify reactions or complexes that exist in this example in a signaling pathway. And we'll see this example in a few slides. Beyond looking for patterns in graphs, there are also representations and kind of data that have come about that never really fit into the pairwise graph structure to begin with. So one is metabolic reactions and metabolic signaling, where you have many reactants and many products. So you kind of lose this pairwise relationship. Another are signaling pathways, intracellular signaling pathways. This is the picture from Reactome that shows that these reactions are not pairwise necessarily. They involve many molecules. There are entire, standardized file formats that are these kind of based on these reaction processes. So those also don't even start with a pairwise assumption. And to kind of pull it away from the molecular side, this idea of beyond pairwise relationships has also occurred in ecology, in food webs and competition networks. So this is a picture of a coffee agro-ecosystem web, and the black edges are direct effects. The blue edges are modifications to those effects, and the reds are modifications to those modifications. Right? So it's capturing this higher order relationship. To also motivate why graphs may not be the best representation, particularly for signaling reactions, I want to just go through one more example. I'm going to talk about 3 different reactions that are real. They're from the Wnt signaling pathway, and they're present in existing pathway databases. And for every single one of these, we have to decide how to represent them as a graph. The first one is simply a complex, a protein complex. So if we have this example of these 4 proteins bound in a single complex, we have to decide how to represent that as a graph. Many people choose to represent that as a clique, right, where we connect all pairs of nodes. You can also think about regulated reactions. This is a case where a particular phosphatase dephosphorylates a protein in this complex. But without knowing which protein it is, the best thing we can do, one decision we can make, is to connect this phosphatase to every single member in that complex. And finally, when we think about complex assembly or disassembly, right, this is an example where a complex sequesters 2 proteins and subsequently releases 2 more from this complex assembly and disassembly. The best we can do, really, is represent each complex as a clique, so we end up with multiple cliques. It is really challenging to go from that graph representation and kind of infer what that original reaction was. And this problem becomes even more exaggerated when we take the union of all these edges, right? Where this graph represents these 3 reactions. So we'll come back to this example near the end of the talk, but I just want to kind of motivate that here. Okay, so our group kind of talked about a lot of different aspects of these higher order network structures in both of those camps, both looking at patterns in graphs as well as looking at alternative representations of relationships. We kind of dove into the theory in our group, so this is a wonderful first session. It's not going to be necessarily the lightest material we'll probably see. But we're really interested in kind of the types of questions people are asking and how hard those questions are, those problems are. I'm going to kind of MC this and introduce each section and then hand it off to the people who will show really cool examples, just kind of like a sampling. And we're going to start with graphlets and the reconstruction conjecture. Okay, so graphlets, if you're not familiar, are small, connected, induced subgraphs of a graph. I'm showing here the 30 graphlets that are small, connected subgraphs up to 5 nodes. Graphlets are technically a little different from motifs when you're trying to find instances of a motif because these are induced subgraphs. That means in your graph, for every 4 nodes that are connected, they are going to be one of that G3 through G8. Right? They're going to be exactly one of those. Okay. There's been a lot of work to count these graphlets in networks, and that's been really useful to characterize networks and compare networks. I want to talk about one other kind of perspective of graphlets that has been also really successful in network biology. And that is thinking about, for every node, calculating what's called a graphlet signature. So just like here, in this top row here, we have the first up to 4 node graphlets, right? We can count the number of those graphlets in a network. But for every node, we can also count the number of graphlets that that node participates in. You see we have these numbers in the graphlets for nodes in the graphlets that corresponds to essentially roles that nodes can play in those graphlets up to an automorphism. And so for each one of those nodes, index 0 through 14, we can count for this node V in this lower section, how many graphlets that node participates in. So there are 4 graphlets of 2-node graphlets, which are just edges. There are 2 instances of G1, of node vertex 1, where it's the edge of one of these, just 2 edge paths. And there's 1 triangle, which is node 3, which is G3, right? That touches that node V. And you can come up with this vector called the graphlet degree vector. And this characterization of nodes is really kind of elegant in the sense that when you look at the 0 orbit, you're looking at the degree distribution of a graph. Right? The clustering coefficient of a node is some combination of G1 and G2, the orbits in G1 and G2, right? The number of triangles that touch a node. And graphlets just generalize this type of statistic. So this has been used really successfully by Tiana and others in network alignment problems to be able to align nodes in 2 networks, and also identify proteins with similar biological processes in nodes. So actually I want to show another picture from one of Marinka's earlier papers that looks at this, this role, the specific roles of graphlets and nodes for disease genes and non-disease genes in a network. Right? And this paper showed that there is, for certain roles, so for certain graphlets, a slight but consistent enrichment in disease genes versus non-disease genes. Right? So this is a way that we can use graphlets to kind of understand more about the role or the kind of local neighborhood of nodes. All right, that's what I wanted to say about graphlets, and I'm going to hand it over to Peja to talk about the reconstruction conjecture.
I will come there. It's a little awkward because we have one microphone, but hopefully this can be smooth. Let's see how it goes. Maybe that's a good idea. I think it might not be bad, and that Anna just keeps this spot. So I want to tell you that many of us are counting these graphs, and it's really important. And I first saw those graph counting techniques from Natasha's paper in 2007 that Anna just said, but actually there's something deeper to that. And what's deeper to that is that this whole theory and reconstruction conjecture that says that the claim is that all finite, simple, undirected graphs with at least 3 vertices are reconstructible. What does that mean? I need to give you an example. So if you look at this first graph G in that middle road with 5 nodes, and then you remove 1 node at a time. And there's 5 different graphs that you can do that. So G1, G2, G3, G4, and G5. So you remove one at a time and all the edges that touch that vertex. The claim says that if you count all of these graphlets with one vertex less than original graph and call this a deck, okay, so that you can reconstruct the original graph up to isomorphism. Okay, so you remove one at a time, you get a lot of graphs of size n-1, and now you can suddenly count them. As you can see, these 2 graphlets are, oh, that's great. So if you can see, this 3rd one and this 5th one are the same. So this is a multiset. The deck is a deck for this particular graph. G is a multiset, and so you count those. That's just graphlets of size n-1. We can reconstruct the original graph up to isomorphism. This has been, what this really means is that not only this set of smaller graphs can reconstruct the bigger one, but it cannot reconstruct any other graph. So it's not that just shows you that one, but it has to be only that graph. There's actually stronger claims that if you remove all the duplicates, that's called set reconstruction, it still works. And it's been shown computationally up to 13 nodes. There's something like 10 to the 14 graphs of size 14. Okay, so I think now this one. But there's something even cooler than that. So that actually gives us theoretical grounding why counting graphs actually matters, is that there's a more general claim that says now you can remove up to k nodes, go to smaller graphs, and you can still reconstruct the original graph. And it's just an existence claim that has not been yet, it's much harder to disprove, of course. I'll just let you read, for any positive integer k, there exists another positive integer, f of k, such that any graph on at least f of k vertices is reconstructible from its collection of subgraphs with k vertices removed. Okay, so this tells us that we can actually remove a larger number of vertices, count the smaller graphs on removed vertices, and that will be enough to be a good representation to fully reconstruct the original graph. Why that matters is actually, for at least one thing that's important in machine learning is that if you have 2 distinct graphs, we all want to build similarity functions. Similarity functions are the core of machine learning on graphs, and a similarity function that can distinguish between any 2 graphs is called a complete function here. And so if the reconstruction conjecture holds, and if you can go to very small graphs, then it means we can uniquely reconstruct almost anything there. And we have very powerful similarity functions to work on these graphs. Now, how does that extend to hypergraphs? What Anna was just saying is that it's an open problem. Very little is known. Bipartite graphs are one representation of hypergraphs. And very little is known except that I probably butcher this, that if you have triangle-free bipartite graph, then it works. But if it's not, then it doesn't. So a lot is here unknown. Okay, but the point here is that counting graphs is actually quite a powerful way of representing bigger graphs. And there's something really deep in that, and it's one of the open problems of graph theory still, to prove the reconstruction conjecture. Let me see, and I'll give it back to Anna.
All right. Thank you. All right. So that was really what we wanted to talk about, just in terms of looking for subgraphs and counting and characterizing subgraphs. The next piece is talking about taking graphs and converting them into other representations. And there's a lot of different representations we could talk about. For the purposes of this talk and this discussion, we're going to talk about hypergraphs. And so I'm first going to just define what we mean by hypergraphs. There are kind of 2 versions that we'll talk about today. One is an undirected version, where we have a hypergraph which has a node set. Both of these representations are just going to have nodes. And the difference is going to be in the relationships among them. So in an undirected hypergraph, each hyperedge E is a subset of V that can contain more than two nodes. If all hyperedges contained exactly 2 nodes, you're at a graph. Right? So like a graph is a special case of this undirected hypergraph, an undirected one. We also will really briefly at the very end talk about ordered hypergraphs, and that's actually instead of having a hyperedge being a subset, it's a tuple. So the hyperedge ABC would be different than the hyperedge BCA, right? But for most of this talk, an undirected hypergraph, we're going to think about sets of nodes as hyperedges. There is also a directed version of a hypergraph, and here again we have a node set, but hyperedges in this case are an ordered tuple, an ordered pair of subsets. And so this directed hypergraph allows us to connect many nodes in, say, I'll just name it. If you look at this hyperedge R2 in the middle of that bottom image, it connects 2 nodes in the tail of that hyperedge to 2 nodes in the head. So it's allowing us to capture many directed relationships. Okay. So with those definitions, we're going to start with undirected hypergraphs, and we'll jump into the next example of building an undirected hypergraph, or just we'll call them hypergraphs, from a graph. That's Teresa.
So obviously this is in general very difficult problems, but there are certain classes of graphs where this problem can be nicely solved. So the 2 family of graphs that I will mention today would be cographs and P4 free graphs. So okay, thank you. So assume that you have now, so this is a binary network that is of actually Fenneron pathway, which are presented as binary interaction. However, in the real setting, we know that this is not happening through binary interaction, but through the cascade of complexes. So I would like to recover the cascade of complexes. Here in color are, this is an experimental network, so in colors are actually the nodes that are presented in the network, white ones are actually absent. So what's happening is when Fenneron is activating the pathways, there's a cascade relationship, then including formation of this complex here, then this complex here, then in this cascade of the signaling pathways, we have 2 proteins that can substitute for each other. Either of them then can bind this complex, and they go to the transcription. So how we can think about it? So basically, we think about complexes usually as, be that either sets of nodes or clicks. So in this case, this is a signaling pathways. So we have a cascade of events. We don't have too long of the cycles. And in special cases, when the graph is so-called chordal graph, which is defined as a graph that in every cycle of length 4 or more has a chord. If a graph has this property, then it can be represented as a tree of complexes. So let's try to represent a graph that we started with as a chordal graphs. Often we will need to add a couple of diagonals to those cycles. If the number of diagonals that we add is not too big, then we can represent the graph as a chordal graph without distorting the network too much. So if we do that, then we can then decompose this graph as a tree of complexes. And here what you see is basically the ovals are the complexes that we uncovered. The path here shows the tree, and the colors kind of show what is the overlap between those complexes. So you can appreciate that ST11 is this complex that is this protein that is enters at the beginning of this pathway. It's continuous for the number of complexes. Then you have a number of proteins joining this pathway. Then we have the red one here. I cannot see from now which one it is. Then we have the purple one joining here, and at the end the scaffold complex joining here. Then we have the partition. One complex goes to forming DIC1, DIC2 together with KSS1. Here we have DIC1, DIC2 with FUS. I cannot see from here. So beautifully construct the cascade of complexes. Now we added the edges. So that perhaps what it does is that makes things that did not interact to interact. Now there's another beautiful theorem coming that what we have here, after we added the each edges, what we have in each of those nodes we think is a click. Let's remove those added edges. And again, if that was a click, what we get after removing these edges is something that is close to the click. And there is another beautiful theorem that if you have a graph that does not contain pathway of length 4, then you can represent this graph as a Boolean expression, using hierarchical, starting from the leaves, all the proteins, and represent as a Boolean expression. So we can then look at this graph, and we know that we added an edge between those two nodes. It did not exist. We added it to make it a chordal, but if we remove it, then we can exactly represent that this actual graph contains this or the other one. So obviously, for this kind of decomposition to work, we have to, the graphs have to be have certain properties. So of course, the challenge is how we can extend it to bigger family of graphs, or how to reduce in the most efficient way a graph that is not chordal initially, does have the long cycle into something that can be dealt this way. Okay.
All right, Peja, I think you're up again.
So I will try to return us a little bit back to, again, very simple stuff, and just push us towards machine learning a little bit, and talk about classification problems potentially on graphs, and then how we can convert this into hypergraph-related problems. So let's just think that we can have two different types of classification on graphs, and one is, this is actually my class slide, which I really like. It took me some time to get these molecules and print them from PubChem. See, you can have these molecules here, caffeine, for example, with some carbons and hydrogens, etc., and then maybe theobromine, which is very similar. I challenge you to find the difference, but one is safe, and the other is unsafe. And then, there could be some others that are safe and unsafe, and this is obviously can be represented as a graph where edges could be different. A double bond could be one type of edge, and so on. This is a colored graph, both on edges and vertices, and you can just classify whether one of those is, safe or unsafe. Okay, so that's one type of problem that we can be solving on graphs. And the other type, oh, by the way, did you see the difference? I think there's this, here there is a hydrogen, here there's a carbon with a couple of hydrogens, 3 hydrogens, and that is all. That makes one of them safe and the other unsafe. And this is remdesivir. It just was popular when I was making the slide. Okay, another type of problem is vertex or node classification. So here's a protein structure where a distance between different alpha carbons was used to generate edges, and then alpha carbons or just residues were nodes. And I believe this is a serine, so the idea is, whether this serine is, phosphorylated or something maybe functional. And so in this case, we are interested in building similarity function on vertices, okay, or nodes. So now the idea here is, and feel free to interrupt me if I'm going too fast or to speed me up if I'm going too slow. The idea is, so of course, we can be classifying those. We can also be classifying edges. For example, you can have an edge of proteins and drug targets, and you might want to say whether it's activation versus inhibition. Okay, so you can just classify on edges. You can classify on vertices. And there's another classification type of problem on graphs, which is link prediction, which is, in this case, we must assume incomplete graph. In other two cases, vertex and edge classification, we do not necessarily have to assume an incomplete graph. And so here, the idea of link prediction is to fill in the edges that are missing, as in prediction of protein-protein interaction. So I want to persuade you here that you can solve all of these problems in a unified framework on hypergraphs. So you only need a hypergraph-based scenario where you all do everything that needs to be done on vertices in the hypergraph. And the concept that is important here is the concept of a dual hypergraph. So you can map graph to a hypergraph and any hypergraph back to, well, here, graph or another hypergraph. And so the idea is this. This is a first example. So here's a graph. And let's say we want to work on either vertices or node or edges. And so now the edges in the original graph will become vertices in the dual hypergraph. So I do not see what number this is. That is E5, and that's E5 node here, okay, I believe. And so now this vertex V1 actually connects 3 edges, and that vertex will be this circle, I believe, here, which is now called epsilon1, okay? So it will be a hyperedge. So a graph in the original space will become a hypergraph in the dual space. And so here's a brief definition. Edges become vertices, and hyperedges are in the dual graph are constructed from edges incident to the original vertices. So there's this is another example. I will not go through it, but sometimes this can create self-loops on the nodes, and that's a little bit tricky. So it's now different type of hypergraph here. Reconstruction conjecture works on simple graphs. If you remember from before, that means they do not have self-loops. So you might want to count it differently. It's a small thing. Now, how can you treat link prediction? Here, link prediction, now you're asking a question whether this edge here, let's call it E bar, exists between these two nodes. Now, you can map the original graph to a hypergraph, to a dual hypergraph, but you have to construct an extra vertex here, which is E bar, and you need to change 2 edges that touch that original edge E bar. So one will become a circle here around 3 different vertices, and the other will just be created over here, epsilon, some number, okay? And so now, by this little extension, let's call it dual extended hypergraph, now you can convert all of these 3 problems on vertex classification on hypergraphs. And so all you need to do is develop vertex classification algorithms, and presumably you can handle all of this. And so one way to do that is to count again. And so now we can think of hypergraphlet counting instead of graphlet counting. And so here's a list of uncolored hypergraphlets of up to size 3 in terms of nodes. And these are rooted. Okay, so there's various ways how people count. We like to call them rooted. Natasha's group likes to call them orbits. I never understood orbits, and so found somewhere the definition rooted sounds really good, because you just put a root on any of the nodes. And so this is a hypergraph 3-1, which means 3 vertices, 1 hyperedge, and the root is this square guy here. And then this is compressed annotation, so there could be 2 different roots. 3 means 3 total nodes. 2 is the second one in the order. 3 is the third one. So here's the numbers are also indices of these hypergraphs. Okay? So now we can count those and create representations, and from these representations we can learn. And this is just what Anna was showing. This is counting. So this is the number of N1 graphlets. So it could be 3 1s and then 3 2s, how many of those, how many 3 3s, and so on. Once you have the representation, the regular kernel function is just a dot product of those between 2 different nodes. If you're rooting this graph, you're predicting on a node. But if you accumulate it over all roots, then you're predicting on a whole graph. So that's the only difference here. And what's a kernel? Just for, if you haven't seen it before, it's a function that maps elements from the original space into a real number with a few additional constraints. One is symmetry, and the other is positive definiteness, which we need for various types of optimizations to work. And now the idea again, count those graphs, find the representation, see how it works. And so this is from our own work, just to show you the results. Obviously, counting these hypergraphs, and we only did undirected. Directed hypergraphs or ordered hypergraphs is going to be much, much harder. And we compare these. So these are different problems. One is drug target classification problem, up to link prediction and drug target link prediction, and so on. And so so what I did not say is that you can also count those little hypergraphs with flexibility, which means up to a certain level of disagreement. So what we count is up to disagreement of 1. That's what tau equals 1 means. So everything can agree in our count, but one, either edge or a vertex or a color on a vertex, can disagree. And the prediction accuracy over things such as random walk type of kernels is significantly better in all of these cases. This is perhaps not surprising because our algorithm is significantly slower as well. And so we are trading specifically speed for accuracy. And this is if you are interested in these random walk kernels across different problems. In linked prediction, we also have this L3 framework. There was a Barabashi paper recently where fills in triangles, and we thought it can work. It's very cool, but not always. And so the idea is if you count those, you will really have a slow algorithm on the hypergraphs. And so everything is here done in hypergraphs, but it will allow you better inference if you have time to wait a little bit. And I think this is it for me. I give it back to Anna.
All right, so in this kind of whirlwind of examples of representations, we talked about patterns in graphs. We've talked about converting graphs into other representations, hypergraph or kind of complex representations. And we're going to end with some kind of rethinking some really classic algorithms, really important algorithms in network biology in a hypergraph framework, shortest paths and random walks. So for this part, we're going to actually go back to going to the directed hyperedge definition. So if you remember this slide from the beginning, we saw that the graph representation was kind of messy. It didn't really convey the reactions. We can think of these three reactions as, in the case of a complex, an undirected hyperedge, like Teresa's discussion earlier. A regulated reaction could be a directed hyperedge where the regulator is in the tail of this directed relationship. And complex assembly could also be a directed hyperedge that really kind of clearly explains how these complexes are being merged and broken apart. And so again, just like we kind of looked at the union of these three graphs, we can look at the kind of union of these three undirected or directed hyperedges. And you can see that what we've done here is we've moved from a pairwise interaction-centric representation to a reaction-centric representation, where by and large, every directed hyperedge represents a reaction. And this kind of difference becomes more stark the more reactions you choose to look at. So if we add a few more reactions for the Wnt signaling pathway, here are 7 different reactions in the bottom left that shows the formation of this complex at the membrane that sequesters a couple proteins and essentially breaks apart a complex. You can see kind of that those reactions are kind of more clearly understood than the graph representation in the upper right-hand corner. All right, so with that, we're going to talk about looking for shortest hyperpaths in this directed hypergraph representation. And Murley's up.
So in case you're wondering, where we get these types of networks from, these are not just theoretical constructs. If you go to the Reactome database, Reactome is a directed hypergraph, and it's fairly easy to take the Reactome files and convert them into these types of representations. You may also know of the now defunct pathway interaction database from the National Cancer Institute that also is a directed hypergraph. And also almost any SBML model that you find, we can think of it as a directed hypergraph. So we can actually lay our hands as practitioners on many large collections of directed hypergraphs. And so the next question is, what do we do with them? So a very fundamental question to ask is, what does connectivity mean in the case of a directed hypergraph? It turns out it's a little bit more complex than we might imagine initially. So one thing I should point out historically is that the literature on this subject is very sparse. There are a group of Italian scientists who worked on this in the 90s, and then some of us in the computational biology literature have taken it up. So the key idea to define connectivity here is to use a concept from biology, a very fundamental concept from biology, which is to say that in a directed hyper edge, if I want to reach a product, that is a node or a complex on the head side of the hyper edge, I must have all the reactants present. I must be able to reach all the nodes or hyper edges on the tail side of the directed hyper edge. So this is captured by the notion of B connectivity, B standing for backward. So the rule really is that you can make a product only when all the reactants are present. So in this hypergraph up on top, if you want to make this product T, I like to walk over there.
Okay, cool.
Hopefully people can still hear me because I'm loud. So if you want to make this product T from S, how are you going to do that? So a B hyperpath connecting S to T is simply a sub-hypergraph, a collection of hyperedges from the original hypergraph, such that this node T is B connected to S, meaning that for all the nodes in the head of the hyperedge containing, in the tail of the hyperedge containing T, those are all recursively B connected to the source. And in addition, the sub-hypergraph you obtain is as small as possible. It doesn't contain any superfluous edges. So you can then take such a B hyperpath and measure its complexity simply by the number of hyperedges in it. And then ask for algorithms that compute the shortest hyperpath, B hyperpath between two nodes in the hypergraph. Not surprisingly, the problem is NP-complete, and in fact, it's NP-complete even if the number of nodes in every hyperedge are small. Fortunately, we don't need to be restricted by the fact that the problem is NP-complete. We can actually write fairly strong, good integer linear programs to solve the problem in practice, and that's what we have been doing so. And when you actually run one of these algorithms on something like the Reactome database, and in fact, this is from the NCIPAD database, and you ask, in the Wnt pathway, how can you produce these two critical transcription factors that control downstream expression? You get a shortest hypergraph like this, and if you actually examine it, you can make out the key elements, key reactions in the Wnt pathway that will allow the transcription, these transcription factors to get activated. And although we don't have the image over here, if you were to do a similar construction on an equivalent graph, these critical events simply do not emerge clearly. So this was an example to show that, try to convince you that when you actually compute shortest hyperpaths in directed hypergraphs representing the Wnt pathway, you can actually make biological sense out of the relations. Now, I should point out that the definitions we have been working with only compute acyclic B hyperpaths. And by that I mean that nowhere in the hyperpath can you have a reaction make a product which itself was a reactant in an earlier reaction in the hyperpath. And in a series of papers, John Casasiglio and Spencer Kriegel have relaxed this restriction, and they have come up with heuristics which allow you to compute directed hyperpaths which can contain cycles. And in some of their recent papers, they have also been able to address negative regulation. In some of the earlier work here, we were only able to look at positive regulation by making every positive regulator, as Anna mentioned, a node in the tail of a hyperedge. Now, it also turns out that in practice, B connectivity is rather a strong constraint because many of these directed hypergraph databases are somewhat incomplete. So in another line of work, what we have been doing is to try to relax this constraint a little bit. And in principle, we can define connectivity by allowing a hyperedge to occur in the hyperpath, even if not all of its reactants are reachable. And as we allow more and more such hyperedges to occur in such a hyperpath, you get a BFS-like connectivity structure wherein you can reach more and more nodes as more and more hyperedges are allowed in the hyperpath without all the reactions being present. So with this, I'm going to stop, and then Petya, you're going to go and talk about random walks and hypergraphs.
So a little more on these algorithms is obviously random walk is a very important concept for many machine learning types of algorithms, and one of the, in my opinion, most important papers comes from Gertner from Colt 2003, where they look at the random walks between a pair of full graphs. So these are not sort of node-based random walks, they're graph-based random walks. And then I'll try to bring this up to the hypergraph random walk. And so the idea is, okay, so we have potentially colored graphs, whether these are nodes or edges, and we consider a walk on graph G prime and another walk, double prime, which is a walk on graph G double prime. I should just say that the walk is a sequence of vertices where these can repeat. Okay, so you can just have an infinite walk. A path is a walk where there is no repeats. And then a cycle is a path where the first is the same as the last node. Okay, so now that we have that, walk is any kind of trajectory there. And so you have to have a similarity between two walks. So if you're walking through the labeled graph, so maybe two nodes have some correspondences and they have some colors, and so you match red and blue and count some sort of similarity between two random walks. So we have to have this similarity function, and then the kernel, or the similarity function between two graphs, is a sum over all pairs of walks. So this is infinite, obviously, because there's infinitely many walks. But the point is, if you put some dampening factor here, let's say some lambda less than 1, perhaps, then this converges, and you can compute those types of things in polynomial time using some kind of matrix inversions on product graphs between G1 and G prime and G double prime. And so this is a beautiful paper with a lot of nice theory. And just before I talk about hypergraphs, I want to show you another way to compute similarity that has not yet been extended to hypergraphs, to my knowledge, but this is a Weisfeld-Lehmann isomorphism test, which is a necessary, as far as I know, but not sufficient condition for graph isomorphism. So you have two graphs, and you want to know whether these two are isomorphic, okay? And so you start by just counting labels. And so you have 1 and 1 over here, and I think this is also 1 and 4 and 3, and so on. And so you just count them. So for example, G1 has two labels, 1 that is a 2, 1 that is a 3, 1, 4, and 1, 5. Okay? And G2 has some other numbers that you just count from the original graph. Okay, so it starts by that. And now you start to merge node labels into supernode labels by saying, if this 1 is next to a 4, you create a joint label 1, 4. So this one is next to a 4, you create 1, 4. This one gets a few other numbers, and so you get this. So now 1, 4 becomes a new label 6. It's also here. And 1, 4 here is another label 6. And so now you have another set of labels, and then you count those as well. And once these two graphs start to differ, you know that they're not isomorphic. But if you go all the way to the end, they could still not be isomorphic. Okay, so this is not yet necessarily true. And so then you can actually test for isomorphism. But you can quickly reject a lot of different pairs of graphs without running full-scale algorithms. And so now you can collect these labels and create your similarity function. And so as far as I know, this has not been extended to labeled vertices. It has not been extended to hypergraphs, and it's certainly looks like an open problem. But one problem where random walks have been done on ordered hypergraphs for kernel functions and similarity functions. And I'll have to apologize here on behalf of Ronnie Cardone, whose paper has 0 images, only formulas and proofs and stuff. So therefore, I have 0 images here, but I will try to explain to you what that is. So first, what is an ordered hypergraph? So we have a set of vertices and a set of edges, and each edge is a tuple of nodes where order matters. Okay, so you can have the same, in hypergraphs, undirected hypergraphs, you have a set to be a hyperedge, and in ordered hypergraphs, it's a tuple. So on the same group of nodes, you can have a factorial number of potential hyperedges. And so here's one where a set of vertices is 1 to 5, and then one edge here has n1, 2, and 3, the other one has n1, 5, and 6, and n3 has 4, 3, and 5 order, and the label is P and Q. So these are labeled graphs. So the way they form a walk is they start with, it's a sequence of edges, right, which connects vertices in the end. So it starts with the first edge, P, n1, n2, n3, and then it exits at the position 1 and enters this next edge at the position 1, so which is n1 and n1, and then P is here, and then it exits at position 2, which is n5, and then enters this one at position 3, which is n5. It has to be the same node. And so that's a walk now, a sequence of edges with entrance and exit positions. And from here, they create a walk type, which is just a labeling with these types of things. And so now they want to count similarities of walk types. The count of walk types is what makes these graphs similar. And that's it. Now, this can be done, obviously, brute force, which is very difficult. The beauty of their paper is that they produce a dynamic programming algorithm to do that. And it's really a beautiful work, and I think very important. And it works on ordered hypergraphs here. And I think this is it for me. I'll give it to Anna, and maybe we move to discussion.
All right, so we end here at 10:30, right? I think 9 to 10:30, great. Okay, so we have a couple discussion topics, but since we have actually kind of 10 minutes before the discussion, maybe we could just open it up for general questions about the smorgasbord of what we just showed you. And then we have some kind of additional topics that we as a group are really interested in discussing. Representation and learning, considering label data for subgraph counting, and just establishing baselines for these higher order representations. But first, I'm happy to open it up and the working group can answer questions about different pieces. Yeah, Ben.
I have a question. So on Morali's section, there was this pathway diagram, this hypergraph representation. I
Yeah.
don't know if you can go back a few slides.
Yeah.
And the question I had was that, this one.
Yeah.
So nodes are here represented multiple times, like beta-catenin
Yeah.
is there multiple times. And so, I mean, strictly speaking, that's not even a graph at all, because the vertex is repeated with the same label. So you must actually have to have a slightly different label.
Yeah.
And so, how do you construct such a thing? What does it mean? These are different beta-catenins. Like, what's, is it a node participates in one edge?
Yeah. Yeah, that's a really good point. I'll take a crack at this, and then Morelli, if you want to follow up. When we first kind of proposed this hypergraph, the signaling hypergraph formulation, yeah, we said that these complexes are hyper nodes, and there's a set membership, and then there was a node set on top, kind of underlying that whole thing. For this, we actually ended up kind of throwing that out for exactly Ben's reason, that beta-catenin is present, the same, kind of beta-catenin is present in different complexes. So I'll just point a couple out. So this is those are the phosphorylated forms of beta-catenin. So basically, every single one of these complexes also has a unique ID. So basically, what we do here is we go back to only a directed hypergraph, where the nodes are unique IDs. So every complex has a unique ID here. So the original idea we had was kind of to have hyperedges or undirected hyperedges and directed hyperedges. We actually don't, in practice, use the mashup of those two things. That's still an open problem, yeah, because how would you have a path that includes many members? Yeah? Okay. Good point.
I was going to say the same thing because you actually did all this work.
Yeah. Cool. Any other questions? Yeah.
So, with the respect of the, a couple of speakers mentioned the time complexity, how big graphs, especially for integer linear programming, for the kernel function, how big graphs can be considered and can be,
Yeah. So the question is about the time complexity, and I can answer that for these directed hypergraphs, and then maybe Paige can jump in for the hypergraphlet kernels. So in practice, these are still really fast to compute, even exact integer linear program solutions, because ultimately these representations are very, very sparse. So Reactome, which I think is kind of one of the biggest kind of consistent sources of these, have maybe 10,000 reactions all together. And so that's essentially 10,000 hyperedges, which is pretty small in terms of the potential density of these networks. So we have been lucky in being able to run these quickly. As the data sets grow, though, that's going to be a big challenge.
Yeah, I would say the same is true for counting hypergraphs. This is obviously exponential because if you're going to start counting, then there's just so many of them. And the real problem happens if you want to count with flexibility, let's say with one mismatch. Those are a killer, those are super slow, and I think to work on networks of let's say, 10 million nodes, I don't think so. But our biological networks with thousands, tens of thousands, I think, maybe even hundreds of thousands, I think it's perfectly fine to use those types of approaches.
Yeah.
Curious, are there, so all of this is when you have a graph.
Yeah.
And we know a lot about the graph, but in many situations we don't know what the graph is. And are there ways to actually learn hypergraphs from data?
Yeah, so the question is, can you learn hypergraphs from data? Because everything here is assuming you start with a graph or a hypergraph. That is a really challenging problem. I think that's an open problem. Thinking about, graph inference, like network inference, and kind of the space of links that could be inferred, that just gets compounded with multi-way relationships. Yeah, Murli.
So we did a brief experiment where one of the undergrads in the group, he took all the set of complexes, maybe if you come up in front, if people know that can't hear me. He took all the set of human complexes, for example, the quorum database, and then try to ask, in how many ways can you combine them so that the stoichiometry meets and you can try to formulate complex reformation reactions. And there's a brute force way of addressing the question of reconstructing hypergraphs, and I think he came up with millions of different ways of doing it, most of which probably don't occur in biology. But that was sort of a very simple brute force approach to try to reconstruct hypergraph. But I also think the work that Teresa talked about is one way of, because you're not just clustering and finding complexes, you're finding an organizational structure among the complexes. So I wonder if we can expand upon that and ask, even if the original graph is not caudal, so you start with a pairwise network and then ask if you can find multiple such tree-like structures because there is not a single caudal graph. So maybe decompose it into caudal graphs and then apply the approach.
So to follow on that, so that gives you a reconstruction. Now the big problem is that reconstruction unique. And sometimes you can have, even for this caudal graph reconstruction into the tree of complexes, it's not necessarily unique, so you can count all the reconstruction, look at the similarity, and I think uniqueness here of that reconstruction is also a challenge.
Could you use an ensemble? Could you
Yes, this is what we are trying to do now, look at all this reconstruction and find which is more frequent, which is, what are similar, what are variants.
I was just going to add, this is a question depends on what you mean by from data, because I think maybe implicit in your question is that data is pairwise, which it would be for say mass spec pull down.
Absolutely.
Like there are other sources of data in crystallography structures. You actually measure the complex, right? So sometimes you do measure that. You could measure the hyperredge in principle. I think you mentioned Reactome is itself already a hypergraph because somehow they're sort of manually curated this information. They're curing these complexes. So somehow based on
Yeah.
whatever biological knowledge they're imposing on this, they're already creating. Right?
They're using
Absolutely.
structural data, right?
Yeah,
They're
yeah.
using all the mass.
It's very interesting what Bud mentioned, because I would consider it's the other way around. So, well, I love all of these. I mean, it's clearly very, very useful, but I was just thinking, because, our research always trying to think about the structure basis of these complexes, right? So the first slide, Anna mentioned that, for complex represent as a clique, I mean, it's clearly, an important way of representing a complex, especially when we have the mass spec, for example, you pull down those, you cannot put any more edges. So I was going the other way as Bud. I'm thinking, as a mass spec is not pairwise, it's actually like a hypergraph, like it's already in a hypergraph format. But if you have a co-crew structure, or EM structure of large complexes, now you know all the context. So it's not like a clique anymore. So knowing what are the subunits within the complex that are actually touching. So we are really interested in finding these touching point or interfaces. And I think that's anyway. I'm just put it into this hypergraph.
Is it actually like a structured hypergraph or?
Yes, right.
Yeah, so and I think it's important of how phosphorylation, which subunit is actually getting phosphorylated, and how this, which is like the STE5 is the scaffolding, what protein is touching which one, how is assembled. I mean, there can be also even a temporal order to it, but that's too complex. Right now, we're just considering what are the subunits that are touching each other, because those are very important for the integrity of the complex, and mutations accumulate significantly on those interfaces and things like that.
How would you represent that computationally? Would you do it?
Right,
That's the
so,
question I'm asking.
so one could imagine that you take a complex like this and imbue it with extra information as to where the pairwise contacts are.
Yeah.
Right, so it won't be a clique, it won't be a glob of nodes, but there'll be internal connections among those. So I think, in principle, it is possible to extend the hypergraph concept to incorporate this type of information. And then the question is, what can you do with this additional information? What are the algorithmic challenges that arise?
Yeah, I agree completely. I think obviously data is going to be a big problem for this, but you could imagine if you can assemble the same complex in different orders, that's two different ordered hyperedges on that same complex, plus there could be an internal structure, because they could maybe be assembled in even different ways. I'm not saying this happens, but that clearly hypergraphs are not powerful enough for, at least as defined, to model some of these things without additional structure.
I have two questions that I want to ask now, but we can wait to the answer even after all the sessions have been done. The first one is maybe Pedro, and I know Marika will want to weigh in on this. Subgraph counting versus deep learning. Which is the answer for classification?
I asked this question Jure when he visited us at Northeastern, because my conjecture was it's subgraph counting, because it's complete. If you can show that it's complete, how can you beat that? He agreed, I believe, but he pushed the agenda of large networks in this case. But I think equivalence is between what deep learning learns and, let's say, what the kernel with counting learns. I think it's still an open question. I saw some work on that. Marika probably knows more than that. It's a very interesting question.
Yeah, so I would love to hear what work
Yes.
has actually been done, and we can chat about it later. And then second one, again, touching on classification, you showed us AUC results and stuff like, how far are we from being accurate that biologists would trust us? Especially with problems like link prediction, where you can't like take your top 10 predictions, it's like your top 10,000 predictions, right?
Yeah.
So how far are we from accurate?
Well, I think it's a tough question, and the reason for that is that what you think is a biologist here. If it's
Well,
say, a
Mike's
person
sitting
who
next
will
to you,
do-
so he can.
Sorry? No, if
Yeah,
it's just an experimental person, not just, but an experimental person who will test in some order, some compounds and hypotheses, I think this could be really working well. If you're thinking medical application where a patient would go for a diagnosis or something, I think we are not close yet. That's a
Yeah,
different
I think
set
that's
of
where I was
demands.
headed.
Yeah.
Yeah.
Well, question about that, I guess, is what do you, how do you define the interactions, right? So, I mean, related to sort of whether you're really trying to predict the two proteins are actually touching each other. Or, I mean, it also based on the starting data, or you mean they're in a complex. Both information are important, but you see what I'm saying? But if you're really predicting the two proteins are touching each other, I think there's a lot of implications of that and a lot of utility of that.
There's no reason why I couldn't do that with these graph approaches. You just would have two graphs where pieces come from different proteins, and you could be looking for those types of motifs. In my opinion.
Okay, so back to this question of the biology, I think that what the value of this approach is really identifying involvement in activity, right? You measure, trying to measure a biological phenomenon and see what's included. So, I mean, so my question is sort of how complete are these models, right? Given reaction, I mean, if I want to say, okay, if I you know, ascribe those, you know, four nodes in the middle to be involved in this pathway, is that enough, or am I missing something, right? And so how do we validate, or at least, how do we measure completeness so that biologists can say, okay, with 80% confidence, this is true?
Yeah, so these pathways, these databases are clearly not complete. They are curated, so they depend on the completeness of the literature. They depend on the expertise of the curator, and they also probably depend on the judgment of the curator. I mean, you know a lot of this from the pathways, right? Two different curators might come up with completely different, somewhat different representations of the wind pathway. If you look at the wind pathway in Reactome, you look at the wind pathway in KEGG, you look at the wind pathway in the NetPath database, even the protein members are different across these.
So if I had to do an experiment, if I have collaborators that want to study this pathway and I'm implicated, then what experiment do I need to do, right? That's, I think, you know, we can work together with biologists to complete this. So, I mean, I don't want to throw up my hand and say, oh, it's not complete. But, you know, if we have, so what's the experiment? Is it another pull down? Is it more modeling and burns?
Can I just say something about it? And I moved here so we can see each other. I think this is a fascinating problem, and morally, and we were meeting for months trying to look into a new way to predict protein function.
Yeah.
They now have something that's called GoCam, causal activity model, which turned out to be not feasible yet quite because it's very sparse. But if you are to build an active learning approach to fill in that GoCam type of model, I think there's so much that can be done. Seems like the community is moving in that direction towards causal determination of function, and those are all going to be graphs of this type.
I'll say one thing and then Alex. Another kind of interpretation, which is something that's very funny for me to say, who just presented about complex network analysis, is maybe if you're trying to identify if a protein is associated in a pathway, graph algorithms may be the way to go because they're way more complete when you build them from experimental data. So it's almost kind of the question you want to ask could help have people decide how kind of, I don't know, careful of a representation of the underlying biology you want. If you wanted to say, is this protein involved in this pathway, there may be other really standard, more complete ways of doing that. And so one of the last discussion topics, I don't know if we're going to get there, is about this trade-off. Like, when do you want to actually go for these alternative representations, harder to solve problems on, data's more sparse, kind of what are the trade-offs there? Alex.
I just have to add, putting on my pathway curator hat. So one reason why we started Wiki Pathways is because we weren't satisfied by canonical pathway databases. And what I mean, KEGG and Reactome, most databases try to have one apoptosis pathway. And so when you ask questions like, well, is it right, can we validate it? In reality, there are 50 or more apoptosis pathways, depending on which cell, what time, what stage of development, what disease condition. The reality is much messier than just trying to find one true gold standard apoptosis pathway. So with projects like Wiki Pathways, we're inviting researchers to add all of their unique models of apoptosis. And some have done it, some haven't. Where we found the most progress was actually going through the literature figures. And people publish, they draw these in Illustrator, unfortunately, and even PowerPoint tools, they draw these things without any knowledge about how graphs should be constructed. And we've pulled out, using OCR and NER, we pulled out the identity of the molecules and pathways throughout 25 years of literature. And there's more content there than any of the pathway databases. Almost none of the, no two figures are alike. They're all very diverse, but we don't have the edges at all because people draw in crazy ways using these non-standard tools. Even kids don't know.
This is months of work.
So there's a grant proposal right there.
Yeah. It got rejected recently.
Monica?
Yeah, I just wanted to say that someone asked about like how far are we from doing link prediction, and I think it's important to think that there are many different types of links, and actually means different things. So we often abstract interactions just as edges, but for example, predicting functional interactions or gene expression interactions, like topologies would be like much more helpful there than it would be in predicting direct interactions.
Yeah.
And so it's also important to think about the types of what we actually mean by an edge.
And I would not have an answer for you for that, how close we are. Yeah, I would not know.
Just follow up on that, actually, I was gonna say, even the starting point, I'm gonna put, because, you know, I'm gonna put my, maybe biologists try to put my biology, because we do experiments trying to find, you know, map these interactions. But if you look at the databases, there's also a lot of noise there, right? So the question, you know, there's actually the trust of a lot of these big databases is not that high. I mean, it's the stuff. So I think there are two things is, you know, one is this, you know, one of the big problems I see is this, what I call the supplementary table 1 problem, right? So a lot of papers now published using mass spec, right? When you do a mass spec, you find hundreds, if not thousands of potential interactors, but a lot of those are just background, right? And then they focus on one or two, the top hits, and then that's the paper. But they do publish that supplementary table 1 with all of those, and all of those are going to be stick into the databases. Again, there's no better way. I'm not saying this is the wrong way to do, because that is indeed published as part of the paper, but then that can create a lot of noise, right? In the literature, so-called curated, you know, all of this. Again, I don't know what is a better way to do that. But the question is, can we use, I think it's a twofold question is, what is the starting, do we need to, I think a lot about, you know, how, you know, what's the starting network? There are two competing views. I'm not saying which one's better or which one's worse. One is trying to use some way of filtering that to find the high quality, which is the path that we often chose, but there's another way is, I mean, for sure that's gonna miss a lot of bona fide interactions. Another way is, I'm gonna take everything out there and you know, and to be as complete as possible, right? So that's one. And the other thing is whether these allow graph-based methods, like the reconstructions, can actually help.
Yeah, when an underlying node is incorrect, right?
Yeah.
There could be another one here, right?
Yeah,
that we mass spec just missed,
So
right?
you can maybe future that, like, yeah.
Well, that's another level. Of course, mismatches can be always somehow accommodated, but to predict on this node is very tricky now, because maybe that node in itself is not correct anymore, right? And so there should be another layer of method that incorporates for that uncertainty in the ground truth data.
Yeah, so I'd like to kind of offer this perspective on combinatorial algorithms and what is the role of possible representation learning algorithms and deep learning algorithms that could exist here as well. And Peter very nicely mentioned that, well, you know, one pitch for why deep learning might be useful is to scale this to larger graphs. But perhaps in biology, we don't have the graphs of that scale as in some other domains when that really becomes important. So is there other reason why kind of representation learning algorithms could help us here? And one area that I'm particularly excited about is that of modeling uncertainty in kind of somewhat better manner, or being aware that these networks are context dependent. It was very nicely mentioned before, like they could vary depending on cellular context, tissue identity, and so on. So then even if we make predictions and go to experimental validation, simply predicting that A and B interact, it seems it's not really actionable, I would say. It's like it really would require almost prediction about A and B interact in a specific context, and then the experiment should be done in their context to validate. Otherwise, which context to use, which animal model, which experimental setup. So in addition to this context dependence information, is this incredible problem of
false positive rates, negative rates in the sense of edges are there but should not be there, edges are missing. Pedia very nicely mentioned, I think in one of his slides, there was this parameter tau that allowed them to kind of take into account for mismatches. And when tau is greater than 1, it's an impediment on computational complexity. So where I see the role possible here for representation learning algorithms is to kind of bring closer combinatorial algorithms, which are kind of very nice because we can explain them and understand the mechanism better. And endow them with one layer of being able to model, for example, look for those subgraphs that are maximally consistent with some other type of data, which could be translated to essentially maximizing then the likelihood of the subgraph structure that is found based on some other data type, for example, right? And kind of that's where I see the role of representation learning algorithms here, perhaps as playing in biology, rather than using them as a way to scale things up if our graphs are really not on the order of hundreds of millions of nodes, as might be in some other domains.
Yeah, I think that is a great segue to the discussion topic on representation learning. And personally, I think if we transition to that and that's what we end up talking about for the rest, that's ideal. So I think to kind of also give kind of an introduction to that, Arjun has a slide here.
All right, I think everyone here has already said a bunch of things that I want to say about representation learning. So the basic idea, just in case, just to make sure all of us are on the same page, is that the idea of representation learning on regular graphs is that ultimately, if we have a graph, and then if we are able to learn a numerical representation, essentially a vector that represents every node or potentially every edge, then we can supply these vectors to standard machine learning algorithms so that we can do things like node classification, edge prediction, and so forth. Okay, that's the idea. And as if we apply this to hypergraphs, then the idea would be to create such a representation, a numerical representation for every hypernode and every hyper edge. Right? And what we essentially want to do is, I need to figure out which is which. So? Okay, I'm totally lost, but that's okay. Oh, okay. God, thanks. Yeah. So ultimately, the idea is that once we have a representation for every single hypernode or hyperedge, structurally similar nodes and structurally similar edges should be very close in this vector space that we learn. And the way this is done, standard way of doing this in hypergraphs is to take a hypergraph and then turn it into a regular graph, where you turn there's something like this clique expansion, where every single node, every single hyper edge is turned into a clique. Or there is a star expansion, where you introduce new nodes, where every single new node represents a hyper edge, and then it's connected to every other node that the hyper edge contains. And then we can apply standard graph embedding methods to the stuff. So that's how it's done. And I want to highlight, a method from a recent paper that talks about this. And then I think this helps us also talk about some of the other discussion points that all of us raised in terms of representation learning. So here the idea, it seems, is that you go through 3 different steps. So one is to take this hyperedge and then do something called graph coarsening, where in a sequential layer, one by one, you take this hyperedge and then reduce a bunch of nodes that were at the original location into a single node based on, and then the way this is coarsened is that a bunch of nodes, 2 hyperedges that share a large number of nodes can be fused into a single edge, or 2 nodes that are part of many shared hyperedges can also be reduced into a single node. And then if you do this, this is done sequentially so that the graph becomes coarser and coarser at higher and higher levels. And then at some point, this graph is simple enough to apply a standard embedding algorithm on top of it. So that gives us some kind of a vector representation. So let's say assume that this is the coarsening step, let's say pictorially. And then we have a standard embedding algorithm that's applied to this high coarse network. So that's the initial embedding step. And then what happens is that what they are proposing is to do a refinement, which is to go from the embedding at the coarsest level and then go one layer down every single step. And at each layer, an embedding for an edge at a finer layer is created based on the embeddings of all the nodes it was part of, the previous layer. And then for every node, the embedding is created by aggregating the embeddings of all the edges it was part of in the previous layer. So that way you have some sense of neighborhood for both nodes and edges, and then you keep averaging this. Now, how these embeddings are averaged, essentially how much you borrow the embedding from your neighbors, and then how you combine that with the embedding of your own embedding in the previous layer, that is part of the learning process. Okay. So ultimately, we have this final embedding of the original hypergraph. One thing that Marinka said that I was thinking about was, how do you induce some context specificity, maybe perhaps into this entire process? And one way to potentially do this is to introduce that context in the coarsening stage. So perhaps the way we coarsen a particular hypergraph to some high-level representation, or rather a simple representation, at every layer we could potentially introduce some notion of context. So let's say maybe gene expression or some other thing. And then where we say we're going to use that data on top of this hypergraph itself to coarsen at various levels. Okay. Some of the other couple of things that I want to mention is that Perja mentioned random walks. So another way to think about node embedding or edge embedding here is to say that you're going to use the hypergraph, but then use the random walks that are defined on hypergraphs, and then use them as inputs to shallow learning models like word2vec, and then learn embeddings directly. I don't think anybody has done this yet, but it's potentially another idea. Okay. Thanks.
I'm happy to MC throw any questions for other people, yeah.
So, for this is the embedding, representation learning, how often is the embedding independent of the task that you're trying to solve? Is this all like independent, like you're just taking your hypergraph and just like embedding into space and?
Yeah, so the question is how independent is the embedding to the problem you're trying to solve?
Yeah, I think right now, most embedding, even in regular graphs, is typically done, seen as an unsupervised problem. Right? But then you can also use a supervised method where, let's say we have some kind of features on the nodes, let's say based on, I think, for example, Jian Ma had this very nice example where his group was studying 3D contacts between points across the genome, and then they were treating this data as a hypergraph. And then what people typically do is to say, I'm going to generate some kind of a node feature where every single node says something about the underlying biology, maybe I know something about the contacts. Or they generate node features based on local graph structure, maybe the degree distribution or the distribution of graphlets. And then use a supervised method to do this embedding. So both are.
Curious because if you look at it as a graph neural net package, almost always by default we assume that there are attributes on the node.
Yeah. Yeah. And so whenever they assume attributes on the nodes and then you don't have external attributes, what the typical idea is to use the graph itself to generate some attribute vector. So for example, this graphlet vector that we presented, that is a good attribute vector to generate based on the graph, and then use that as the vector to do the supervised learning.
Oh, there's a question from Zoom. I figured it out, all right.
Got it. So just want to inform everybody that we have several online participants, Anais Bodo, Gary Bader, Lenore Cohen, Mitra Basu, our NSF program officer who generously funded this workshop, and Pietro Guzzi. So Gary had a comment. A good example of machine learning supporting higher order networks is NLP with n-ary relations. Papers covering this topic may be useful inspiration. Just wanted to know that. Does anybody who's online want to ask any questions or add additional comments?
I have maybe one comment. Can you hear me?
Yes, we can.
Hi, this is Gary Bader. Nice to see virtually many people that I know. Sorry I couldn't be there in person. So there's some interesting discussion about the relationship of what we're talking about here with biological use cases and what biologists need. I think this is really important, but there's also something that's interesting that's happened in the field. I think, in general, people are still working with simple networks. And so why is that happening? It would be interesting to discuss why, people, after 20 years, people still really focus most of their efforts on simple networks. One simple reason for that could be that it's more difficult to work with hypergraphs. There's a lot of extra work that needs to be done algorithmically to work with them, for instance, and visualize them. I'm not sure if that's a good reason to study hypergraphs. But I also wonder if there's a way that the human brain works with this information based on causal reasoning or relational reasoning. I don't know if anyone's thought about this, but there's sort of this natural idea or natural way that the human brain can work with simple network data, just reaching out, finding connections in the literature and other things, social networks, that kind of thing. So it's very intuitive for people. I just wonder, and when we presented hypergraphs to users and tried to visualize them, they get confused frequently. So I just wonder if anyone's had any thoughts about that aspect of the kind of how the brain uses this information. Maybe it's an out there question.
We kind of alluded to that, right? Like Alex was saying, that we've been drawing binary graphs in the literature for decades now. So, and I have also stumbled upon biologists who understand pairwise interactions because that's how they've been operating. I think it's really hard, but we do allude to this fact of why we're still studying pairwise single graphs and, well, in our part of the next talk a little bit. But I do think there is something right about the brain and people's inability to parse hypergraphs. I just feel like it's an open question.
Yeah. I would say for the directed hypergraphs example, I think the end result picture is intuitive for biologists, but getting there is really hard to explain. Whereas with network analysis, we can also just we have that wealth of history where you just pick the graph algorithm and apply it to your application.
Hold on, let me just ask Gary a question because he's the one who was speaking. Could you hear, I'm using a microphone now to communicate with you, and Anna is also standing in front of the microphone. Could you hear the discussion that was happening? You could, so it's pretty good audio, excellent. So that's good to know. Pedro, go ahead.
Would you mind going to slide 11?
Slide?
I think Gary is onto something very important.
Yeah.
And
tell me when.
There will be the Kant paper further back. This paper? No. This one.
Yeah.
I was just this morning asking Worley, what is this graph? And only when I saw reaction that A plus B together give C plus D here, I understood what it meant. That's a directed hypergraph where this first combination of these two nodes gives these two nodes. And I think maybe that is part of the problem, that just this doesn't make much sense unless you already know. Once I see this, okay, now I translate it to that, but we're supposed to understand this immediately, and I did not. And so I think visualization has a role in this place to actually make something that is this simple so intuitive to us.
Yeah, I would also say in our working group, we narrowed in on hypergraphs using, focusing on hypergraphs really quick. And it took us a while to realize that we were not talking about the same hypergraph. There's also, even within this community, a difference in language that we have to kind of overcome.
Could I make one minor comment here that sort of bothered me about the slides from the slide?
Oh, yeah.
Is this actually a bipartite graph? Because there's some extra directionality that's not represented in a bipartite graph.
Oh, that's you're right. It's kind of more like a Petri net type thing.
Yeah, it's like actually.
Yeah.
The bipartite tightness would only separate the reactions from the though, so.
Yeah. I think you're right. Good catch.
But I think when people say bipartite graphs are quote unquote equivalent to directed hypergraphs, they are imbuing, they are adding direction to the bipartite graph edges. So in the top right of the figure C, you see the edge going from A to the node R1.
Yeah.
Such that that's the reactant.
Yeah, I get that, right. But it's actually an additional piece of information beyond the standard bipartite.
Yeah, absolutely.
Yeah.
And if you think about ordered hypergraph, then then there would be like a factorial number of potential. Then this would just be unmanageable to.
Yeah.
So I think somebody online, possibly Lenore, had a question, so let's make this the last question of the session before the break.
Yeah.
I was just going to say that I see some commonalities between trying to push to higher order graphs to some of what people have been doing on knowledge graph representations and networks. I think they're trying to capture and encode some of the same information. I like that this is so graph theoretically principled. I think both the knowledge graph people and the higher order hypergraph formulation have the issue that it's extremely powerful to represent very generic representations, as you said, but the combinatorics become a little bit daunting, and then you get very sparse representations. So I guess the sweet spot will be how you restrict this so you get sort of that fuller representation somehow. I mean, the biologists really want to represent pathways in their networks, and this is toward a language that might let you do that if you do it right. But what to keep in, what to keep out, how to make it not too general so that you can't see anything, that just is a wonderful topic.
Yeah. I totally agree with Lenore, and also just thinking about, we already mentioned how in our very principled presentation, we've already had to introduce some heuristics and relaxations because of sparsity or missing data, that kind of stuff. So as we gain more, will these principled, problems be able to be implemented in practice is a big question too. Any last thought?
I'm just going to add one thing here. I think, Gary, there's a lot to unpack into his comment. So it's actually a great discussion point for me. But I was going to add sort of one triviality to it, but I think it's an important one of why not hypergraphs versus graphs? Yep. I think some of this is familiarity. I think these representations are important for the brain. Some of it, though, I think is sort of more practical in terms of implementation. Yeah. And the analogy I would draw is to the people that are trying to do the pan-genome work. And genomes are strings, and there's all these people trying to get genomes into graphs, and they've been doing it for years, and they're still trying. There's a whole stack, a software stack that's around strings of graphs, or strings for genomes, and multiple alignment, all this stuff versus graphs. And I think there's a similar kind of implementation issue or software stack issue with graphs to hypergraphs, because it's just much more complicated to implement any of these algorithms.
Yeah, it's not an extension of anything. Yeah. You gotta kind of start from the ground up.
You don't have the graph going to the adjacency matrices easily. You gotta keep track of all this stuff. So it takes up. Absolutely.
If we do have a hypergraph algorithm library, if people want to start playing with it.
Yeah, high fun.
So I want to emphasize that we're going to have, after the last topic tomorrow, there will be separate discussion session the last part of the day to hopefully gather all the topics together. So let the juices flow for that ultimate discussion. Thank you, Anna and the team, so much for very engaging and reasonably theoretical presentation. Thank you. Hi, everybody. We're about to start our second session. I can say that the hand clapping is favorite among my students, but it does the purpose. So. How's everybody feeling? Got some stretches out? So we are about to start the second session of the day. It's going to be a long day, but I think I'm pretty confident it's going to be very engaging. So the second topic is multimodal networks and integration. And I believe Sushmita is the one leading this effort.
Right. Can people hear me? Okay. Can you hear me now?
Yep.
Okay, good. Let me just make sure I know how to navigate this. Okay. And then most likely I will.
Just the laser pointer underneath where your
B?
index finger is.
Oh, okay, B. Okay. Okay, all right. Hello, everybody. I just wanted to echo what Anna said. This is such a great opportunity to meet all of the people who've been thinking about this problem. So very excited to be here. So our section is multimodal networks and integration. And I wanted to start off with a disclaimer that we were planning this to be after network inference, but hopefully because it's a closed community, it would be okay. So we have organized this material. So this is being presented by Tony Gitter, Alex Pico, Sarah Goslin, Meng Jiang, and Hang Tang. And we'll be basically doing a tag team where we'll be presenting some material. And then towards the end, we'll have a concluding slides, and then we'll have a discussion. So I'll give a little bit of an introduction, and then just for the other participants of this session, like people who are talking, Sarah is going to watch us on the time. And if she's standing up, this means you just have 1 minute left because we have quite a bit of material. All right, so let's get started. So we thought we want to just maybe introduce what we mean by multimodal networks. So as we know, biomedical data is of many different types, and this can be genomic data, this can be imaging data, this can be network data, and these data sets are becoming increasingly available through a lot of systematic efforts from consortia and whatnot. So trying to leverage all of these data sets to get an understanding of molecular systems or living systems is really a driving question right now. So what do we mean by multimodal networks? And there can be different types of multimodal networks. You can have a situation where, now I'm going to use a laser pointer, where you have a node. You can have a node with, where did the pointer go? Oh, I see. Okay. The B doesn't have any functionality. Okay. So you can have a node, we can have one graph with one node type, but each node can have different attributes. And then you can have a situation where you have the same node type, but you can have different types of edges. And you can have also where diverse types of node types and diverse types of edges. And here are some examples of multimodal networks. For example, from multi-omics, you can have all of these nodes can represent different types of molecular entities like genes, proteins, metabolites, lipids. And then for each of them, you can have some different modalities. And then another type of multimodal network, maybe patient networks, where in addition to patient-specific omic profiles, you may measure other things like imaging readouts, electronic health records, and whatnot. So multimodal network data analysis methods, when we were thinking about putting the material, we thought the kinds of problems that they are trying to solve, so here are some of the things that you'll hear about. One question is, how do we take different types of data and try to reason, try to integrate and interpret all of these types of data sets? So integration of multimodal data using networks as a framework will be an important question to think about. The second question is the lack of systematic measurements of complete data sets. So these data sets by definition tend to be sparse and noisy. So how do we deal with the unknowns in these data sets? And then how do we go and integrate data sets that were not potentially, or measurements or information that was not measured as part of a study? This is where knowledge graphs can be really useful. And so we learn a little bit about that. And then finally, all of us are all excited about how do we build mechanistic predictive models. And then we'll have some discussions about that. So this is basically how our material is organized. We'll first talk about the network alignment problem, which you can think about is suitable to try to integrate different types of data sets, different types of networks. And then we'll talk about how multimodal networks, networks can be used to integrate different types of multimodal networks, multimodal data sets, and then inference of multimodal networks, and then knowledge graphs. And then finally, we'll have some discussions. So with that, Hang, if you want to get started.
All right. So first of all, I want to thank Sushmita for her tremendous effort for coordinating this topic. So I will talk about network alignment. A few words about me myself. My name is Hang Tong. I'm a current associate professor at the University of Illinois. So my group does data mining for graphs and the network. We are interested in a variety of applications where very often graph mining can play an important role. And a large portion of my work is on the algorithm aspect of graph mining. So we do things like querying, detection, prediction, inference, and control. In addition, we are also interested in the theoretic aspect as well as some system-related work of graph mining. So today I want to talk about network alignment, where we want to find the node correspondence across different networks. Now I should mention that this work was largely done not in the context of biology, but rather in the recent DARPA project on modeling adversarial activities. But I believe that applications of network alignment goes beyond adversarial activities, including biology, and also in finance, in social networks, and so on. So here's the plan. I will briefly show you some recent work we have done for network alignment. I will try to keep it at high level. And then I want to share some high-level thoughts about some possible things we can do with network alignment in the future. So very roughly speaking, in network alignment, given the multiple networks, we essentially want to construct a tree of networks where each network lives in certain location of this tree, and then different networks, they are interconnected with each other, if you will, by the branches of this tree. So the general question we want to answer is how can we distill the value, such as the knowledge, from the multiple networks? And our solution is to align them together by addressing the 4 V's characteristics of the real-world networks, including the veracity, variety, volume, and the velocity, which we refer to as the big network alignment. So this is an overview of some work we have done for big network alignment. I will not go into the specific algorithm, but essentially we have tried 2 types of methods for network alignment. One is based on optimization, where we essentially view one network as the noisy permutation of the other. So if we can recover this permutation matrix, we solve the network alignment problem. And then the second approach is based on the graph neural networks, where we first want to find the embeddings of the nodes of different graphs, and then the network alignment problem essentially becomes a point registration problem. So this is a more detailed overview of some recent network alignment algorithm. You can find details in some recent tutorials we give at CICAM and also at the KDD conference. So let me just give you one example on neural cross network transformation. So in the 2 type of method, I mentioned the optimization-based method or the graph neural network-based method. Both methods require some underlying assumption. For example, the optimization-based method either assume the linearity or the consistency, right? Like one network is the noisy permutation of the other. Now, as for the graph neural network-based method, they often assume that the embeddings of the nodes of the different graphs are in the same embedding space, or the embedding space of one graph is a linear transformation of the other. So in this work, we try to remove these assumptions, and we ask a more generic question, that is, given two arbitrary networks, now what will be the mathematically optimal way to transform one network to the other? So our solution is based on the encoder and the decoder architecture. Basically, the encoder will coarsen the source network at the different granularities, and then the decoder will reconstruct the target network at the different resolutions. And during this process, we'll also identify the associations or the alignment between the source and the target networks at the different resolutions. Okay? So next, I will briefly share some thoughts about a few possible things we can do with network alignment. And I will focus on the following three questions, including when or where to align, how to align, and whether we want to do alignment at all. So first, when or where to align. So this is a very typical two-step procedures of network alignment, or generally for network integration or network fusion. So in this procedure, in the first step, we align the multi-source graph into what we call the world view graph. And once you have this aligned graph, then we develop some data mining model to find the desired pattern. So here, our observation is that in the vast majority of existing works, these two steps are often conducted separately. So a natural question is if we can combine these two steps together to construct or to align an optimal graph for some specific downstreaming data mining tasks, such as link prediction, classification, and so on. So next, how to align. So here, our main observation is that for the vast majority of the existing network alignment method, they belong to what I call the additive alignment. So if we have 10 graphs, each with 1 million nodes, let's align them together to have a graph with 10 million nodes. Right? Or if we have the embedding vector for each network, let's concatenate them together to have a longer embedding vector. So this type of additive alignment works well in many cases, but beyond that, how about what I call the compositive alignment? So what do I mean by this? Let me give you an example of the network of networks. So this is some early work we did in bioinformatics, where we are given the collection of the blue tissue-specific protein-to-protein interaction network. Now, in addition, we are also providing a green phenotype network. So in this case, instead of simply adding different blue tissue-specific PPI network together to have a bigger PPI network, we can instead compose this blue tissue-specific networks with the help of the phenotype network into a network of network model, where each green disease node itself will be mapped into another entire network. So this way, we might be able to recover the underlying hidden structures among different blue tissue-specific PPI networks. So finally, whether we want to do alignment at all. So here, our observation is that it seems that in the data mining community, there are two lines of the work, each pursuing a quite different idea, right? Including the one is on the merging idea, the other is on the splitting idea. So for the merging, the idea is that let's merge the input graphs, let's align them into the world view graph, and then let's do the mining from there. Now, on the contrary, for the second idea, it pursues a splitting idea. Given the input graph, let's split them into smaller but semantically more meaningful units, such as the clusters, communities, motifs, and also the graphlet that was mentioned in the previous topic. So the natural question is, when should we pursue the merging idea and when should we pursue the splitting idea? And also, currently, these two lines of work almost conducted orthogonally. So another interesting question could be if there is a sweet point where we can combine these two lines of work together. For example, maybe we want to first split them into smaller motifs or graphlets, and then let's align them together, but let's do the alignment on those semantically meaningful units on the motif graphlets level instead of the individual node level. So I think that's all. Next, I'll turn to Sarah and Tony.
I'm gonna both time myself and you can time, well, we're doing 20 minutes between the two of us and we're gonna jump back. Okay, so. So the work that Tony and I are presenting really focuses on integrating multi-omics data from diverse experiments and using the graph as a tool to facilitate interpretability of that data. And it's really premised on this idea that omics data is inherently noisy. I can go into great detail about various aspects of noise. I give two visual examples here using this. There we go. So we know that RNA-seq counts can exhibit a bimodal distribution, and it's very hard to distinguish poorly expressed transcripts from those that are actually just noise. Proteomics data shown here below, which I didn't complete the citation for, apologize, also has a lot of missingness based on proteins that don't fly or those that don't, isoforms that aren't flying in all of the samples. I'll go into a lot more experiments about that. But there's a lot of missingness. And so I should always allude to this, statistical tools that have been built to compensate for this. We have a lot of, and I just list them here for the purpose of completeness, but we can identify which transcripts, proteins, genes are interested in an experiment because of the phenomenal statisticians that have built these tools and made them reproducible. And then on the right here, I list tools that help us interpret the results of this. So we're given a set of proteins or genes or transcripts or metabolites, which ones are biologically relevant given the experiment that we've carried out. However, a lot of times these bag of genes techniques fail to really provide context for various biologists and really identify testable hypotheses. So the protein interaction network in particular, and if I didn't allude to this because it's going to be covered more in network inference section, is using protein-protein interactions, particularly those from the literature, to really map biological measurements, whether they come from genes, transcripts, proteins, to an interaction network, which is fantastic. So here's an example of one such network, including my favorite protein, NF1, and using the physical interactions from the string database, and the edge width indicates sort of confidence, which is how much they're measured in physical experiments only. So this is what it looks like when you map a biological experiment to a protein-protein interaction network. The network is very dense, the measurements are sparse, and it's really, and so if you give this to a biologist, they'll just laugh at you and will never talk to you again. So I don't recommend doing that. So this slide ended up a little too small. I pulled this from a review that was written in 2013, at least the top, but I tried to sort of categorize these, and there's some mixing and matching, but there's this idea of network pruning versus module detection, which are sort of the most common algorithms that we like to use, that we're again taking omics data, mapping them to a network. And so I linked to them here, and they fall into this idea of network diffusion type algorithms that rely on one formulation where you're diffusing scores of, I don't have all the equations here, scores of nodes based on their presence in an experiment across a network. Community detection, which is used for data integration, which is a slightly different formulation of a similar idea where you're detecting, again, modules that behave together. And then there's this network pruning approach, which we'll talk a little bit further about, which are, the prize collecting Steiner tree, which is one major example that Tony will discuss. There's also max cut min flow, which again takes sort of two sets of nodes and tries to identify the paths between them. And then multi-commodity flow, which will map multiple experiments from these sort of pairwise experiments into a single graph. So with that, I will give it to Tony to sort of dive into the Steiner tree problem as our main example.
Thanks, Sarah. So we wanted to give one specific algorithm that can address this network pruning problem. Sometimes you might hear this called pathway reconstruction or subnetwork inference, but the idea is that we want to avoid this blue and yellow hairball that has all of our experimental interactions, all the edges layered with the multi-omic data that's relevant to some specific condition, disease, whatever it is that we're studying. So this is not necessarily the best algorithm. Everyone has pros and cons. It's the one Sarah and I are closest to, so we can tell you all about it in great detail if you'd like, but plenty of other folks in this room work on other algorithms that have benefits. But let's just talk specifically about Steiner trees for a minute. So Steiner trees are similar to what we might want to solve with minimum spanning trees, but the big difference is that there's a subset of nodes that are of interest. So think about these nodes as coming from the multi-omic experiments. Maybe they come from a mass spectrometry experiment in a specific cell line, in a specific patient sample. So those are going to be colored here as the yellow nodes. And our goal is to find a way to use the graph, the protein interaction graph, to find connections among the yellow nodes. And I may say, again, use protein scores coming from experimental data, from these statistical analyses to decide what nodes are yellow or not. I can use edge weights from protein interaction databases, maybe like the string weights that Sarah mentioned, to come up with confidence scores on edges, and then translate those to costs where high confidence, more reliable edges are going to have very low costs. So the incentive is to try to find connections among these yellow proteins, yellow nodes, using high confidence, low cost edges. And essentially that's going to be our goal. So again, if everything's yellow, it's a minimum spanning tree. The Steiner tree variant is different because we have to choose which of these gray irrelevant nodes might be important for forming the connections to the yellow nodes. To make things just slightly more complicated, but also address the reality of experimental data, Sarah mentioned with mass spec, a lot of single cell RNA-seq. Other experimental technologies are noisy. So we might have false positive interactions. We might have borderline statistically significant changes that we might have hints of being relevant to some specific condition, but we're not sure. Those things might have low scores. On the other hand, we have false negatives, so proteins that are actually relevant in the process, but they weren't measured for whatever reason. So our goal is to come up with a formulation for this subnetwork inference problem that accounts for that. And to do that, we're going to not insist on connecting every yellow node, all these proteins of interest. We're going to give them some quantitative score and then weigh that against our edge costs and say, well, let's grow the network using these low cost edges until it's large enough. It captures enough of the high scoring yellow nodes, but not insist on capturing or connecting all of them in whatever subnetwork we're going to return. So that trade-off is captured in the prize collecting version of the Steiner tree problem, where we're now the protein scores on the yellow nodes are the prizes. And we want to try to find the connections that accumulate enough of the high scoring nodes. And as we see on the right, sometimes we'll be willing to prune or exclude some of those low scoring yellow nodes. So we want to think about this in a little bit of detail. We have these two objective functions. We don't have to have a single connected graph. So we may say we want a forest structure that's a collection of tree structured graphs. And our goal is to weigh these two terms. So we have one term that says, if you look at the vertices in the forest you return, you're going to be penalized every time you exclude some vertex that had a positive prize on it. So we're summing over all the vertices that were not picked up. And then we also have to pay a penalty for all the edge costs that we do include. So incentive to not have a network that's too large, to not use those unreliable high cost edges. But we don't really know how to weigh these, so we need to have some tuning or trade-off parameters that will control the size of the forest networks we get back. And I'm not going to talk at all about how to solve these problems. It's an NP-complete problem. I'll just mention there's been work on that both in theoretical computer science, and that's been translated to practical network biology problems. So Sarah and I in Ernest Frankel's lab worked on this one tool, Omics Integrator, that uses a message passing probabilistic model to come up with approximate solutions. But others, like the R package on the bottom, are actually more discrete. They're using clustering and minimum spanning trees. So all sorts of ways to come up with approximate solutions to this hard problem, which again have their own pros and cons.
Okay. So I did want to allude to this idea that there are other algorithms out there, and one of the approaches that one of the roles that I play at my institution, which is the national lab, is trying to find the best network algorithm given the data set that we have. And so, network diffusion, as I mentioned, really has been, I don't include citations here, used to identify mutational complexes in cancer. Community detection algorithms have been remarkable at integrating diverse data types. Max cut min flow is really for mutational drivers of phenotype. And you can read the rest of the slide. I think the point is that there's no one graph algorithm that seems to be ideal, right? It really depends on the biological question at hand and in the data that you have. And so, Tony and I each kind of put up our projects that we're looking at right now because we're in a field of experts that might have solutions. And so for me, I'm really looking at spatial data right now. And the left is the field of spatial transcriptomics, which I think is really quite cutting edge, at least in cancer, where we're able to look at, RNA-seq from various regions and match them to various regions of a tumor, which are known to be heterogeneous with different ostensibly expression of transcripts that can be mapped to networks. And on the right, I show what we're doing in the proteomics space as well, where we're able to take, with a little less granularity because it's proteomics, but various regions of the same tissue and map and measure proteins up to about 5,000 proteins per little square at this point and map them to phenotype from the images themselves. And this is one of these cases where, and I know in the spatial transcriptomics space, I don't know if I've added this citation, where they've been able to say, okay, not only do we have network information from the protein interaction network, but we also have spatial context. So we can assume that the network in one area that's measured is more closely related to something that's directly next to it than something across on the other side of the image, right? That these signaling networks have some kind of spatial relationship. So trying to describe, refine that spatial relationship in the networks, again, knowing that the data are noisy and that we have protein interaction networks are overly abundant, descriptive in many ways. How do we identify what networks are active in different regions of the tissue? Oh, I did include some citations. Good, go me. Okay, so this is what I'm seeing, at least in the spatial transcriptomics realm. And again, no one has done anything in spatial proteomics yet, but this idea that there are packages out there that is particularly this one in the middle here, it's called Bayespace, which tries to borrow information from both the protein measurements that they're getting in a particular, call them voxels, region spot, I guess, is what they're called in spatial transcriptomics, to better infer the diverse heterogeneity of a tissue. So I really think that there's a lot of room to add networks to this field, and I'm kind of excited to see where it's going to go.
So my introduction almost assumed we had a single omic data type that was static, and I think we have at least tools to deal with that setting. So I want to talk about some other areas where maybe we need more algorithmic development. One, I think, opportunity for growth is to look at changes over time. So we could think about measuring protein phosphorylation changes, maybe with mass spectrometry, phosphoproteomics. And this might say, on very rapid timescales, even within seconds, we can have an existing protein population in a cell that's being not created or destroyed, not changing in abundance, but the phosphorylation state of certain phosphorylation sites on those proteins is changing, and that's leading to downstream activity in the cell. So we can measure these phosphorylation changes of the many proteins. We imagine we have a collection of different proteins here, shown by these different line graphs. The takeaway here is that some of these changes in protein phosphorylation are almost instantaneous. They're very, very fast. They happen early in some cellular response, and others are happening later. So intuitively, if we think about timing or measuring node activity times and combining that with graph algorithms, maybe not Steiner trees, but others that are looking at directed paths through a protein interaction network, how do these things help support or contradict each other? So if I propose that one of these early proteins is regulating directly a late protein, maybe that's the case. If I say it's the other way around, that's some sort of logical violation. So there's this information in the timing that should help inform what sort of paths I find in directed graphs that come from a protein interaction network. And I think that information has been underutilized, especially when we think about moving beyond those local changes of direct interactions, but saying, okay, we have maybe this protein upstream in a pathway is only activated at a late time point for some reason. So if I have this multi-edge path that ends up and proposes the late thing regulates the early thing, that's again a logical inconsistency. It must be something else regulating this early protein early. And there's very little work, I think, on really doing this type of temporal reasoning on graphs. If we think about how protein information, temporal information has been combined with protein networks, there's this really well-known study that uses dynamic Bayesian networks and uses protein relationships or pathway edges as a network prior. That's more of a structure learning take on things. And there's other work, including some of our own, that is closer to the Steiner tree formulation where we have an existing graph. We want to do some reasoning on that graph. And folks have adopted max flow based approaches. Some have looked at, again, just customized logical rules that try to basically do temporal reasoning like the sort of things that I mentioned, saying, well, it's unlikely that this is the regulatory path because we have late things happening before early things as we order nodes along a directed path in a graph. So there's some work here, but I feel like there's a lot more opportunity. And then if we finally actually turn to the multimodal presentation I was supposed to talk about, we can do a lot of different biological assays and measure different things about the same cells, the same samples. So sometimes those directly measure something related to proteins. Other times they might measure maybe transcriptional changes. And there are algorithms for integrating transcriptional information and projecting it back to proteins and saying, well, this is the transcription factor that is a protein and is active based on the transcriptional changes I see, or the kinase that is active based on the phosphorylation changes I see on known substrates. So we can get a lot of scores on proteins. And I don't think there's great ways to merge these scores in a sophisticated way, because if I think about what I've done in our applied projects when we have multi-omic data, it's kind of the most possible naive thing. So I did 4 types of assays. Let's add them up, or let's take the max. And if we contrast that about what I hope we're going to hear about later in the machine learning on graphs paper, you think about graph neural networks, they have these very customized learned aggregation functions that are integrating information at a node, at a protein, and at neighboring proteins, and coming up with something that's really customized and sophisticated. And I just haven't seen those two worlds meet. So there could be opportunities for doing multimodal omic analyses like in the Steiner tree world, but maybe using learned ways to combine some of this information. So there you have it.
Yeah. We have a lot more. We wanted to save time for discussion.
I have my own timer. Okay, I have my own. So yeah. Okay, so far what we've heard about is we have networks as a skeleton to try to interpret data, but in most situations we actually don't know what the network is. And we have lots and lots of types of measurements. So what I'm going to talk to you about is some work that's out there in terms of using different types of multi-omic data sets at the molecular level, so transcriptomics, proteomics, and then use that to try to reconstruct networks. So this is what I want to talk to you about. So we have different omic levels, and then we want to feed that in outcomes and network, where the nodes are different types of measurement types and the edges are specific types of connections. So if you think about the different classes of methods that are out there, we can put them on a range of what we know about the networks that are already there in the network that we're trying to construct. So it's a little bit circular. So on the one end of the spectrum, we have methods based on just link prediction or classification, where we have a reasonable number of already known interactions, and we want to leverage that to predict new interactions. So our network is incomplete, so we try to infer some of those unknown interactions. And then on the other end of the spectrum are methods which don't assume at all anything about the structure of the graph, and then they try to reconstruct what the graph might be. So that falls into the realm of structure learning. So, the first is the very simple framework of assuming to try to cast the link prediction or network inference problem as a classification, as a binary classification problem. So here, what we are assuming is we have a set of examples that we know are true interactions. And in most situations, we don't know what the non-interactions are. So we take the complement of the positive set and we draw as many examples as there are in the positive set, and we train a classifier. And we basically say, okay, which is an interaction, which is not an interaction. So that's the simplest approach, but it's a very powerful approach. And by definition, it is integrative because all of the features, all of this multimodal data becomes features in our data set. Okay, and there have been a lot of ways and a lot of powerful resources, for example, string, which Sarah referred to, which is really, a lot of us actually use this as a way to interpret our data, is using a binary classification framework. And there are also these other examples of some databases, but this is clearly in no way comprehensive. But the problem with these methods is that it doesn't assume anything about the fact that what we are predicting is really is on a network. So there is some graph topology that we might want to be able to leverage. So this is really paired data that we want to make predictions on. And so then the next class of methods that we can think about are these graph embedding based methods. And I'll only touch briefly on this because I know the other sessions are going to talk about it, especially the machine learning and network inference. And so what is this problem? So this is saying, let's assume that we have an incomplete graph. So we have some edges and we have some node attributes. And these node attributes are what these multimodal omic data sets can be. And what we want to do is we want to predict the interactions between the nodes based on the node features and the edge features. Okay. And you can do this by using graph embedding. And you can talk about graph embedding can be done using matrix factorization based approaches, as well as graph neural nets, which I'll not get into, but I'm sure the others will talk about. So here is a very interesting paper which tried to do this. So I didn't find a lot of examples doing this for multi-omic networks, but I think we can probably draw these connections. So this was a paper where they were trying to predict gene-disease interactions, and that is the matrix that they are trying to complete. And for each of the genes and the diseases, they had features, and these features can be gene expression data, it can be interaction network, and then they compress that into features, and then they use something called inductive matrix completion, where you can basically take the low dimensional projection and then parameterize it. So basically you take this little matrix and parameterize it, you decompose it into these lower dimensional matrices, and then you can, it turns out that this is like a bilinear problem, and then this Z is what are the parameters, and then you have the X and the Y that you're actually measuring from all of these different auxiliary features, and then you can use that to try to predict the interactions that you have not observed. So this was a case where you have one type of network, but you have lots of attributes for each of the nodes of the network. Going beyond that, we might want to think about if we have multiple types of networks. So here we have, this is from a review, which again was was multimodal data, but again, molecular networks were not as much talked about here. So you have multiple levels. So each of these could be, say, L1 is a protein interaction network, or a metabolic network, or a gene network. And what the idea here is, for each of them, we have some network, but we are trying to actually predict intermodal interaction. So between gene, so you have gene-gene networks, and you have disease-disease networks. We can predict interaction between genes and diseases, or diseases and something else, individuals. And so the way you could do this is, again, you could take all these different matrices, and then you could embed them in some shared space using graph embedding based methods, matrix factorization, or neural nets. And then you would get a feature vector for each of the nodes, and then you could, again, feed them inside a supervised learning framework or inside a matrix completion type of framework, and then try to come up with what are the missing edges. So, finally, the last type of methods is really not assuming at all much about the interactions that are there, and that falls into the realm of structured learning. The methods that are out there could be broadly grouped into two categories. those where the multimodality is being used to inform the topology of a single node type structure. So let's say gene expression networks, and you're using different types of data sets to try to inform what the connections might be. And the other one is where you have different types of nodes and different types of interactions among them. So, so the first one is what I've said as prior-based methods, and the second one is like multi-type. Some of these names are actually what I came up with, but because it was hard to come up with a nice description, but if somebody has a better way to describe it, more than welcome to use that. So what are these prior-based methods? The general idea is we have one type of data that we are trying to describe, let's say gene expression data, and we have these auxiliary data sets which tell us something about what might be the potential interactions between two nodes in the network. And what these methods are trying to do is they're trying to reconstruct a network. And work in this field has moved in two directions. One is using these auxiliary data sets as priors, as graph priors. And then the other one is to try to infer something about that, measurements that we don't observe, such as protein activity levels, because many of these gene network inference methods are saying, let's use the mRNA level of the protein to tell us what the activity level of the transcription factor might be. But these methods are trying to estimate what the activity might be, and that can help in network inference. I'm not doing very well on time. Anyway, so there are two main classes of methods. One is based on whether you put a prior on the structure, whether you put a prior on the parameters. And here is one way to think about it. So you can think about the thing that you want to infer, which is the graph, as a high dimensional random variable. And all of these auxiliary data sets can be as priors to inform the prior. And what you want to do is infer the graph given the data. And when we actually compare methods that use priors versus don't use priors, we see that, there is a performance gain between the methods that use priors versus not. And then the other side of the coin are where you have multiple node types, and you want to maintain that identity. So multi-omic, you have protein measurements and RNA expressions, and you want to infer edges among them. And there are two classes of methods. One is a simple regression-based approach where you're saying, let's integrate these multi-omic data by predicting one level from the other level. And the other one is actually more probabilistic graphical model-based methods that are trying to learn different types of probability distributions. So this was something that we did where we tried to integrate mRNA with protein levels. Very briefly, what we said was, let's use the gene expression data, which is more diverse and more complete, to infer gene expression modules, and then do like a regularized regression approach to try to predict what the protein regulator might be for each gene expression module. And so that's helpful when you don't have exactly measured, exactly matched sample measurements. But going forward, what it is becoming increasingly common is to be able to measure matched multi-omic measurements, and then how can we actually model that? And there are methods based on what are called mixed graphical models, where you are explicitly modeling the distribution of a particular variable conditioned on the other variables. So you can use a mixture of continuous and discrete variables. And here are two examples. One was actually trying to infer something about a causal graph. So they're basically inferring directed graphs, and they're trying to solve these conditional probability distribution estimation problems. And the other one was using just a Bayesian network. I shouldn't say just a Bayesian network. They're using a Bayesian network, and they had all of these different types of measurements. So just to summarize, really, it depends upon how much known interactions you have. On the one hand, the classification link prediction methods are simple, but they require a lot of training data, whereas the other ones make less assumptions about what's known. I think I will just skip this and move on, because we will come back to some of these questions in the discussion. So let's move on to the next presenter.
Great, thanks, Sushmita. All right, now for something a little different. As opposed to, so this will take about 15 minutes, and then we'll have a few discussion slides. Should leave a good half hour for discussion before lunch. I think we're doing good on timing. Great, thanks to my teammates for that. So I'm Alex, this is Meng. And we're going to talk about something a little different. As opposed to data sourced multimodal networks, we're going to be talking about constructing a specific type of multimodal network called the knowledge graph. And we're going to be discussing a few examples of how these are composed from biomedical concepts from traditional databases or from the literature that are tied together by a common semantics and then some of their applications. Okay, great. So we've seen some examples of multimodal networks being used with omics data, and knowledge graphs can be used in a similar way, serving as a reference or as a framework for integrating data sets. These knowledge graphs are also commonly used in machine learning applications for inference or for drug discovery. Here's a title and link to a paper recently published where they pulled together 18 different databases to construct a purpose-built knowledge graph for predicting drug repurposing. And the nice thing about using a knowledge graph in this way is that each of those predictions come with a mechanistic model to help explain why the predictions were made. More generally, if you're interested in a direct lookup of, say, drugs and their protein targets, any sort of single hop query, you can use a traditional database, and knowledge graphs don't really gain you much. Where they really come in handy is for two or more, these multi-hop problems where you're going across multiple types of nodes or multiple types of edges through or more complicated graph structure. And these knowledge graphs often include, in addition to the direct edges that are sourced from databases, might include inferred edges, or you can perform your own inference. And a key aspect of knowledge graphs that you'll hear from Meng and I will be mentioning semantics a lot. And so tracking semantics are really important to be able to combine across multiple databases so that you know one source's gene is the same as another source's gene, for example. We've seen examples of using network analytics. There's also a whole area interested in reasoning or performing federated queries, which is just queries across multiple knowledge graphs. And again, that's enabled because of the conforming to common semantic standards. Okay, so what I'm going to do now is give a quick survey of 3 different active knowledge base projects. Hopefully you don't already know about all 3. And I picked these 3 because they all take a very different approach to making a knowledge graph. So there's not just one way to make a knowledge graph. So this first one is the Spoke project. It's led by Sergio at UCSF, but really maintained by a whole team of collaborators. It began as a multimodal network called HedioNet, that some of you might be familiar with, which had about 17 different types of nodes in it and connections pulled in from a handful of different databases. It's grown considerably over the last few years and now has many dozens of nodes and edge types and millions of instances of each of these. All of this information is in a single graph database, a Neo4j database. And then there's a collection of scripts that ingest content from databases and update on a nightly basis to harmonize all of those different databases, again, into a single model. There's a web GUI for accessing this knowledge base and programmatic access. To give you an example of usage, in collaboration with NASA, this Spoke network was used to help analyze some transcriptomics data from some spacefaring mice. So NASA had collected differential gene expression data on a set of mice that had taken a trip up to the International Space Station and back. And then using Spoke, they performed random walks through the Spoke network, starting from all the genes that were in the network in the knowledge graph, and just going in all the possible directions that you could go in the graph, and created a series of vectors. The dot product of those vectors and the gene expression data allowed them to arrive at interpretations of the data that these are example paths through the spoke network, from genes to anatomy nodes to disease nodes to symptoms. And what they ended up focusing on are the connections from, again, mouse gene expression data, connecting ultimately to just 6 symptom nodes in the spoke graph that are relevant to human astronauts. Okay, second example, Wikidata. So Spoke is a Neo4j database curated by a defined team of collaborators. Wikidata has taken a commons approach, so anyone can contribute to Wikidata. You can log into Wikidata right now and add new information, correct information, write a bot to periodically ingest new information into Wikidata. There's a self-assembled group of interested members who make a very public, transparent process of making decisions of how to model things together and what belongs in there and what doesn't, and you can participate in those groups as well. So everyone's familiar with Wikipedia. There's about 6 million articles there. You might not know that most of those Wikipedia articles have a Wikidata entry supporting them, a semantically defined knowledge graph entry that's much larger and covers a lot more content than is in Wikipedia. This covers geographies, peoples, everything that's in the encyclopedia. So there's just a small biomedical corner to Wikidata that was initially populated by GeneWiki and WikiPathways. Spoke is a Neo4j database. Wikidata is a virtuoso graph database. So it's compiled into a single graph database. It has this web interface that I'll show you in a second, and programmatic access. So just to give you a sense of how this works, if you were to visit an article at Wikipedia, you could identify that there's a corresponding Wikidata entry where most of the content in that article is actually contained in the Wikidata database as semantic triples. So items, properties, and values, and the collection of those triples make this graph. So of course, corresponding articles about the ACE protein, for example, in the biomedical corner of Wikidata, there's a corresponding Wikidata entry. And you can access this directly on the web or programmatically. Again, getting semantically defined statements about these proteins or other biomedical facts. Today, there are dozens of node types and edge types in Wikidata in this biomedical corner, and lots of connections. So just to give an example, symptoms are linked to diseases, diseases linked to drugs, drugs linked to pathways. All that information is coming from multiple sources and are defined in Wikidata in a way that you can make those connections to these concepts and many, many more. All right, the third and final example I wanted to give is a project called BioThings Explorer. This is led by Andrew and Chun Lee at Scripps Research. And what they've done is, instead of making a graph database, they're not using Neo4j or Virtuoso. They've instead just defined a smart API for traditional databases to register to. And so the way that works is Uniprot, for example, is registered in BioThings, and their existing API gets registered in a way that it defines inputs that take in NCBI genes, for example, and outputs that provide NCHI keys or Uniprot IDs or what have you. And each of the databases, there's now over 200, have registered in a similar way, following the same semantic standards for defining their API in a common way at BioThings. And so what this enables is, when someone makes a query to BioThings, that query is digested into a series of queries that then are sent out to each of the source databases, and the responses are returned and compiled by BioThings Explorer and returned as a single result. And so this can scale in a completely different way than a local graph database, which is interesting. And it's also making real-time queries each time you're making a query. And so there's no asynchronous update issues with the data with BioThings. There's a website interface for this and programmatic access. All right, that's my survey. I'm now going to hand it over to Meng, who's going to talk about constructing a knowledge graph from text.
Yeah, so I'm Matt. I'm a professor at Notre Dame. I'm like a researcher in digital processing. So yeah, thanks for talking about the multiple ways that can.
Get in front of the mic, or you want this one? You want to? Just the microphone. I just stand in front of that mic.
Oh, sure. Yeah, I can stand on here. Yeah. So yeah, Alex just talk about the multiple ways that it can integrate a variety of databases into one large knowledge graph. And you think about knowledge can be represented as concepts or entities and relationships. And the relationships can be represented as head, relation, and tail tuples. There are multiple kinds of relations that I can define to create that large knowledge graph. And another way, so these are like taking the databases as sources. Another way is that I can directly use a text as source. So the text can be biomedical, biological, literature, publications. And then you can use natural language processing to solve the problem. For example, information extraction techniques. There are at least two subtasks to create that knowledge graph. First of all, giving you a sentence, giving you a text, identify the boundaries of the name of the concepts, right? It's called name entity recognition or concept extraction. And secondly, that given the extracted concepts and pairs, then try to classify their relationship, identify their relation. If you have specified the relation schema, if you already define the types of relations you care about, then you can make it a classification problem. So that's how we can combine natural language processing, deep learning, especially deep learning has making these language models really, really efficient and effective. So we can use neural information extraction to create a large graph. At the same time, we may ask this question, will we be able to extract all the knowledge from any sentence in the biological literature if we just represent the knowledge as head, relation, tail, tuples? So the questions are facts enough. Taking the middle, this is a knowledge graph we created. So the middle layer, which is blue nodes, are the statement sentences in the papers. On the left, you probably will see some, the head and tail entities or concepts. They are connected by the relations, like increase or reduce. But in those sentences, they do not only have the facts or the claims, but also associated with the conditions. On the right, you can see the conditions, the means, the experiments that they use to observe this kind of facts, or the temperature values, many things, right? Could be conditions in the statement, and we argue that these conditions they play a very essential role in scientific statements, because without the condition that we scientists, precisely specify, some facts may no longer be valid. So we have to extract the conditions from the text. Then we introduce this kind of thing that's called, condition tuples, not just the fact tuples, but those condition tuples. And even for fact tuples, we say there could be some internal structures in the head and tail. They are not just entities or concepts. It could be the attributes of a concept or entity. For example, like here, the inhibition of the quark influx, right? It's an attribute. You can just say it's a concept. You want to separate them. You say the red thing is the attribute of the green thing, right? So then, given one sentence, we develop neural information extraction techniques to extract the fact tuples and the condition tuples at the same time. That can be more complete, kind of lossless, to represent the knowledge. So the novelty of this kind of approach, including we extract condition along with the facts, we identify attributes together with the concepts, we open the concept of vocabulary beyond the databases, because you can identify some concepts that are outside of the vocabulary of the databases. And we try to open the relation schema. We try to take any kind of predicates in the sentences, try to make it knowledge lossless.
So the technologies we use are deep learning enhanced natural language processing techniques, and specifically it's called sequence labeling. I'm not going to the very detail, but yeah. So with that, you can do something for applications like literature search. Beyond, now most of the search engines, they just use keywords matching. You can match the words, you can match the keywords, you can match the phrases. So think about giving you two literature, giving you two articles, you try to know their similarity or relevance. What if we compare them based on the facts, based on the conditions, based on the heads and tails and relations they talk about, right? So if we can do some extraction of this text, we can extract the graph for each article, and you can compare the articles on the graph based on the conditions and facts. So that's more like a knowledge-based comparison, knowledge-based relevance. So that can make this thing more intelligent to search the similar article. This schema we call the CTGA. Say given you the unstructured content, T is to extract the tuples, G is to make it a 3-layer knowledge graph, and lastly use A, which is the algorithm to find the similar articles based on the graphs. So I think Alex and I mostly covered how to integrate the knowledge data, large knowledge graph, where you can see integrating all this knowledge data, knowledge graph with omics data and the clinical data can be really, really useful. Yeah, so references, and let's welcome Sushmita for the discussion. Thank you.
So I just wanted to conclude, and then I'm going to invite my team members to come and facilitate the discussion. So we sought out to look at these 4 main problems, and you heard about different approaches ranging from trying to use networks as a way to interpret data using things like standard trees, to network reconstruction, to knowledge graphs to try to leverage different omic type data sets with patient level data and so on, as well as natural language processing based methods, how they can be used to try to extract information to annotate these different types of knowledge graphs. I think one of the things that we are all excited about is how do we take all of these very rich data sets and get to what we think are mechanistic predictive models. So what I mean by that is really try to come to a place where the model can make predictions about unseen perturbations and try to tell us about what might happen in the future. And so we listed a few things, and I think I'm going to stop and then ask my colleagues to come to the front. But basically, in order for us to really get to these mechanistic predictive models, and all of these models that we've talked about, and also in the first session, all of them can make a lot of predictions. How can we go to a list of predictions to something that is testable, hypotheses that is going to give us something that is biologically meaningful? So what do models need to be able to do? They need to be able to handle noisy, incomplete data, high dimensional data. They need to be able to work with different types of contexts, and context can be space, time, different types of organisms. And then one way to maybe think about what might be a useful experiment to do is coming from active learning, where one could try to see whether we can actually come up with a cost function for experiments that might be, that we could use to try to say this is going to be the most informative experiment to do in the lab. And then another question is, can we really discriminate from correlation from causal models? And that's going to be really challenging. And then finally, we want our models to be able to compute. We want them to be able to take inputs and then produce some outputs. So there is a literature in the field of phonetic executable models that can be looked at to think about some of these problems. So with that, I'm going to invite the rest of the team members, and let's start having our discussion. If you guys just want to come and that's okay. Yeah, let's
questions.
Yeah, questions for
I hope not start with a random question. Generally, if you're going to do machine learning on these huge multimodal networks, we assume a probability distribution on some data here, and data points are drawn from some distribution, and then maybe there's another distribution, some kind of output. Now now when we have everything here, I can't imagine now where is the probability distribution and on what? How do we formalize this into, you know, what are the assumptions behind these integration systems? And, you know, I'm trying to understand, and I think it can be all kinds of things.
I have an initial fairly naive response because I've never done this myself, but the papers I've read that seem to be successful have a very purpose-driven approach where they either construct the knowledge base themselves from scratch with something in mind in order to make sure it conforms to the distribution they're looking for, or you start with a pre-made one and filter it ahead of time to make sure it's going to have the shape and aspects you want. Yeah, I don't know if these are really just being used off the shelf blindly for precise modeling.
This is probably like, and I thought, is this instance of a huge knowledge graph one instance of from some distribution of good nodes from this?
If the media can add two, one, like immediate comes to my mind is the generative knowledge-based method. Because of the generative nature, it naturally underlies probability distribution assumption behind that. The other probably more related to that is my own work on the graph line. At least one line of research on the optimal graph transport. But there, I think the formulation is quite unique. We define two distributions. Then the alignment problem and comparing the two probability distributions over. I'm not sure if there are in general that approach for handling that.
I might have a rambling question or comment. So you heard about network alignment in social network domain. The field of, well, obviously in graph theory, there's graph matching that's existing for decades. But on the applied end, biological network alignment started way earlier than social network alignment. And I feel like the two communities are completely disconnected. And I'm using network alignment as just example. This whole, like we heard, monkeys in my department, and I have no idea to deal with biomedical knowledge graphs. I kind of have an idea, but I'm learning. How do we reconcile different communities working on related problems, even in the same domain, often without knowing or benefiting from each other's knowledge? So this is more, I didn't expect it, but more discussion of how to get there. And this was one of the motivations I used to tell this workshop to NSF. And then we need to merge data mining, machine learning, and traditional computation biology communities. And this kind of links to also for us computational biology scientists to collaborate with biologists when it comes to mechanistic understanding and hypotheses. Biologists often have no idea what they're trying to test or what hypotheses are. Here's the data, tell me what we should test. And we spend so much time trying to, we can rank predictions, but getting to mechanistic understanding is that they can't verbalize what they are looking for. And that causes so much anxiety for computational scientists. So it's kind of two different aspects of merging knowledge across disciplines. Any thoughts?
So yeah, I'm gonna pull from my hat where I worked at Sage Bionetworks, and we did a lot of these sort of like meta questions of how to get people to, you know, cross boundaries, silos, whatever jargon you want to use. But I think it really comes down to like, I like to think of it as a software engineer, like what are the users asking for? Like what is the problem we want to solve, right? And I think sometimes you get different, and I always ask biologists, like, what question do you have? And then we'll figure out, because I think that there are different ways. And then, because there's lots of different ways to answer a specific question, right? You can go to the knowledge graph space, you can go to the graphs, and everyone has their hammer, right? That they want to, and they're just looking for a nail. So I think if we kind of can generate enough problems, they will actually, and questions, they will actually fall into these patterns naturally. And then we can start to see, okay, these questions, like the mechanistic modeling question, like that, I've seen, you know, we've tried to use graphs, we've tried to use Boolean logic, we've tried to integrate those, deep learning. There was a proposal that came out of that. Like everyone sort of, that is, you know, there's a lot of different tools that we can throw at it. But I think it would be nice to sort of say, okay, given this question, these are the suite of tools, and how we can, and then we can start to integrate within that question column, right?
So my experience is the question isn't precise. It's more try to do and I've been sent down the rabbit hole many times.
Yeah,
And it's like exploring, and we can get what are the technical hypotheses?
what can you see in the
What are
data?
the-
What can you see in the data? But yeah, I think you're right. I think you have to choose the right questions as well.
So I think one of the issues is, I mean, biology is hard. And I think that it really requires people from different disciplines to be really interested in the problem. And it also requires, like time, dedication, and trainings. We have to train students to really think about that biology. They need to think about the biological problem. And I mean, those algorithms that Hang talked about, I mean, it would be very interesting to think about how we can, integrate multi-omic data sets and, try to come up with graphs and try to integrate that. And that would be super cool. But it's not like, you just take the data and apply the algorithm, out comes biology. It doesn't happen like that. 90% of the time we are spending looking at our results and trying to figure out what the hell, what does it mean? So I think that, it requires time investment. Workshops like these are actually great. I'm so glad that, you actually did this. I hope this happens again.
Exactly, the events like this will help a lot. So I remember when we had the Zoom meeting with phone, we suddenly realized we are actually working on something very similar. So the Steiner tree problem, although my work is completely outside the biology, I think the connection between the hypergraphs source and the other side also somehow echo what I think the mentality is important. So if we computer science data mining, the folks say, oh, just give us the data, we'll run the algorithm for you, or the other way around, right? So where the tools I can use the computation, just give me the tool and I will run it. I think if we have this kind of mentality, usually it doesn't work like that. So really work together.
So I have an idea for identifying instrumental experiments they want to try and see what people like. So here, my goal is to identify a set of genes on which I want to do more experiments. I don't know what experiments are, and this goes back to what you said. When you're trying to infer these networks purely from data, there could be some genes for which we can identify the pairwise interactions fairly accurately. But could there not be groups of genes for which the data does not tell us what the pairwise interactions are? You really have to poke your hand. So in a sense, they are like undirected hypergraphs. Right? There's a group of nodes. I can't figure out what the pairwise interactions are. So perhaps we could focus our attention on figuring out what these groups of nodes are. So that's why we should be doing the experiment more carefully in order to identify more specifically what it is. I don't know how to solve the problem, but I wonder if that might be a feasible way in which to try to identify groups of nodes for which we need to do more experiments.
So in other words, we could come up with the precise questions that the biologists could then go answer.
Right.
I like that.
There's nothing wrong with that.
I like that.
It's like having a biology core in a computer science department.
Yeah,
It's kind of the opposite.
I'll run them then in a different direction. When we worked with these pathway models and Steiner trees, I quickly found that even if we have willing collaborators, doing the experiment to follow up on a prediction is so open-ended. And then, you have a meeting, it's like, okay, well, are we doing Western blots or targeted mass spec, or like, we got to tag it and follow localization? Because let's say I solved all the network problems. I could say this is the really important node in your graph. That doesn't tell us what the node, what the protein's doing, and how to look at it, how to even validate something at the level of a graph prediction. So that's something I've struggled with is, well, one, I don't know how to get to the right subset of nodes. I kind of feel like knowledge graphs might actually help because it probably generalizes things that we do in a low throughput manual way. But even that, it's like trying to do active learning in some settings, it's more natural because what you're modeling is similar to the type of data you can generate more of if you just do more wet lab work. But sometimes it's so nuanced to figure out how to test the prediction that we just hit a wall there and maybe try a lot of things. Sometimes that takes 4 or 5 years to figure out what that one node was doing. And how do you repeat that? How do you scale it up, even if you have access to a bio core in a CS department?
Yeah, no, I want to well, chime into this and steer it in a slightly towards one of the other questions up there as well. So I love this question, but it's sort of how do we follow up in a useful way? And I think you're right about cross training. But I want to bring us back to this thing about context, right? Because I think the knowledge representations are a good way of getting at some of that. But I think that, all of what we're doing is about, in some sense about context. That you can't possibly think about representing. I mean, we do a lot of graphs that don't represent context, but they're not as, really useful in a lot of pragmatic ways unless we can say, when is this happening? And I would even say, sort of, how confident am I that this is happening? The context that I know about. How do we communicate about that, and how do we do better than, do we want probabilities? Do we want models that are like, I think we could put that on every single representation we talked about today. When does this happen? How often does it happen? Does it happen in everyone?
Can you elaborate on what you mean by context?
Yeah, right. So that's the other question.
When. I think the easiest is to start with the when, right?
I want to know what species, what genotype,
Yeah, I
what
think-
model system. I mean,
You know what
Yeah.
species, right?
Yeah.
So that's something that frankly isn't in a lot of publications, as I'm sure you all know, but could be captured because people know what species they're talking about. But what is being done with that species? What disease are we talking about? Do we know?
Maybe tissue specificity or.
Yeah, do we know, are we looking at specific tissue cell types under certain circumstances? What don't we know, right? So how do we even represent what context? And I think that's a hard question, and it's a hard question that spans discourse, right? So that's something where often when you're talking to a collaborator from another field, you're not necessarily in the same place all the time. And I don't even know how we get that. How does it present it?
I mean, this is one of the things that I have used, at least the networks for it. I think the knowledge graphs really help is that, provides a baseline, right? Like this is everything that you could possibly imagine has been curated into something, right? And then from there we can glean, okay, this is the experiment we ran in our, slightly, off model and micro tissues of this one tumor, right? And so we only saw it, and so then we can say, okay, this is how we can represent this in the context of this larger set. Maybe we can identify some tissue specific phenomena.
I think you could say pretty confidently, even that can have cell types.
But it's a model system, and the models are imperfect, because we had some cytokines. I mean, there's always something that changes things, right? So, I like to say, okay, this is the baseline, but I think trying to figure out what measurement we're looking at and what, because I think that's the other thing is that when you have so much data, it's hard to figure out. It's more deep when but black box versus white box. Like, how do you interpret the results that you get?
But like if you make a multi-week inference, which is the point of these models, are we can we do that, right? Does that make any sense at all?
There's no
And
distribution.
It's not clear that we're even capturing that in the way we do that.
It goes back to the thing about the distribution,
Yeah.
what the probabilities are on your pulse.
Yeah.
And it's this weird balance between, the source of the data and capturing those conditions so that you can precisely say what it is. But sometimes you want that blurriness because you're trying to find something new that hasn't been measured yet, that you want to make a prediction. And so that blurriness can sometimes be advantage.
Right.
That
The
balance
question is whether you're taking things down by, from 50% to 20% or 50%. How much work you want to do to try to follow up?
Yeah.
There's two of you who wants to.
I'm just maybe a bit excited about these topics. So actually, no one asked my question. Maybe what I'm kind of question is, I was thinking about this network alignment. How this, this seems like this graph representation could be in fact the right way to do it. The reason is, when we think about added distance on strings, like string alignment, it seems like for DNA sequences and proteins, that is the alphabet in which changes happen. It seems like string algorithms are maybe the right way to think about it. In network alignment, nodes are not anymore from that constant alphabet. It seems like there's something really different here. And I don't know whether we can do it properly with, regular combinatorial methods. Maybe just I'm wondering whether this network or graph representation type of alignment actually brings something new to this problem, whether we are learning something about these cost functions that is different, just your thoughts.
Yeah, I think that's a great question. I think there's several ways we can do based on the optimization part of the method. So somehow, some early methods, including the highest event that was actually from the higher origin, that actually has a lot of connection with like the natural alignment algorithm with the outside the context of the natural value. So I do think there's a lot of opportunities that I think we're looking at the appropriate cost function, like what do we mean like when using the alignment, like how do we actually add it to one natural so that after we do this kind of editing can match the target as well as possible. So that more or less what this neural developmental transformation will be based on the underlying motivation behind. The other thing that somehow relates to what you talked in the presentation. In many cases, we can convert the graph data into multiple sequences, like using the random. And then once you have the multiple random walk, essentially you can convert the graph into sequence for the multiple sequence. So lots of concepts there maybe will be helpful.
I just want to go back because I think, what I've been talking about, how to test, right? But, I'm going to, again, put my biologist hat on, if I may, and, because we think a lot about, I think that's a very important question. We think a lot about how do we do experimental validations, and we strive to do experimental validations of every of our machine learning prediction papers. And I think number one is you just have to find like the match. I say, I have to find if I'm just interested in the specific, question that the knowledge, I guess, for that pathway or that disease. If they're interested in that, I think that would be the start. Because if they're not interested in that, it's just very hard to say, hey, since you do biology, can you test this for me? I say, well, I'm not really interested in that pathway, that problem. And the other thing also is it's hard, right, with this multi-inference, right? So let's say you find this gene. I can totally appreciate there's some fuzz, allow some fuzziness in there, but if you say this gene somehow impact associated with this disease, right? Now directly from gene to phenotype, that is very hard to directly test. But if there is some sort of, so you have to go into a little bit of a molecular mechanism. If you somehow say, oh, that gene is regulating this pathway, and that pathway somehow modulates the transcriptional environment. I'm trying to multi-model, right? And then this, this gene expression pattern is associated with the disease. I think for the example that you want to experimentally test, again, for someone who has actually really think of this a lot and try to do this kind of test, because it's really hard to do this test, right? Often you test a number of them once in a work, you'll be very, very happy and put that in your paper, right? So I think you want to do the hard work, even if you want to pick the best examples, for those ones that would really have a much stronger. I think that would allow you to easier to test, right? Then you can say, hey, let's test whether this protein really affects the signaling pathway. And by affect the signaling pathway, doesn't change the transcription. And if you don't directly check the disease association, that's already several validations that you have to have.
What kind of predictions the model is making, what is testable in
Going back to the context, like in what context are making their predictions, right? So that's, we all have, maybe it's not protectable by mass spec, right? Maybe it's, maybe you can't measure the transcripts since they get degraded typically. Like there's all these sort of other confounding factors. So focusing specifically on the context.
It also takes discussion, right? You don't have to go down there. You can talk to expert in that disease, right? And then you can say, hey, and then I think it's really, you have to talk to about it, and, whatever work out a plan, validate from the basics and a little bit, steps up.
The man's knowledge graph could answer such questions. So if you link so well with information, Perhaps you want to add one other edge to connect this gene to disease, and that edge is the experiment, and then you, in your core, find the person to do that, perhaps the experiment.
That's what I'm saying, yeah, I think it's, yeah, it's very.
Probably can try the link direction to knowledge graph, try something new, and I'll add something very different. I don't directly work on unexplained data, so I don't push biologists to do some testing, but I do NLP, so I have been observing the biologists have been testing. So you look at literature, you find, you can extract the facts, the tuples from the literature, then you can populate, just comes the tuples, right? You can then, based on their frequency, to know what facts, what kind of relationships that the biologists have been paying a lot of attention to. And sometimes I even find conflicting relationships. Once they increase, they also reduce. And you look at their statements, you find, oh, actually, they have different conditions. So that could be a way to know what the biologists may be interesting. But on the other side, you also have the metadata of the literature, so you know the year. You can figure out like the trends, the dynamics of the research in biology. But I don't directly use all of it.
The definition of context is very difficult, and I don't think we'll ever be able to find it. There will be always a new context. So I think that what's important is to somehow make sure that our tools will be allowing to define how we think the context may change the situation, or what aspect of the data may be changed by because the context. So for example, you have, say, stress, they will be DNA will be stressed, will have changed some binding sites, which are perhaps weaker, maybe more. But also, it would be nice to somehow make those knowledge-based dynamics where we can maybe think about methods that will be helping us to answer how the context may affect. We cannot have almost the context.
We define like a general purpose kernel function on context, whatever we're talking about. You might have context is a gene expression state, that's probably simple, but what if it's like context is just some term I extracted from a sentence, and then the gene expression state, or times, and all these things that we've talked about. I'm curious like how we can know if whatever it is we're doing is similar or very different from this fact or this other experiment or this conclusion, and then actually probabilistically or systematically reason over those things.
So whether to move to mind the maker, that's what you try to do. That's what biologists try to do. Have a lot of rich knowledge bases.
I just want to mention, even talking about complex biological experiments now, it's actually quite advanced. I just want to, yeah, it's very sophisticated. And yeah, for example, even if you're talking about complex, right? So you could infer phosphorylation, for example, right? One of the events. It's very important for the signal. And it's very hard to do this directly, but then, you can generate, very easily can generate a phosphomimetic mutant. So then you can just test that if I use that, and that's a surrogate, it's a variable surrogate for the phosphorylation complex in the cell. So there's many different, such things, available.
I had a related but slightly different question / direction, which is network inference. So if I understand a lot of the things that were presented, like probabilistic graphical models, most of these models don't deal well with feedback or directed cycles, which are actually present in a lot of biological systems. So I wonder, I think you have thoughts, because it kind of goes with the theme of temporal data or perturbation data. Like what sort of data would we need? Where are we in terms of confirming feedback?
You mentioned two things, right? So there's perturbation and there's temporal data, and we could go down the route, I think, for either one of those single data sets. There's been specialized probabilistic graphical models to deal with both of those. Nested effects models are a couple of decades old, and I threw up a term, dynamic Bayesian networks, that that's kind of a root of these models that do structured inference specifically for time series data, and there's been a thread of methods there.
But how much do you use? What's your sort of take on the current state of getting feedback?
So DPMs have been used a lot. The problem is that, the temporal to non-DPM DPMs, the dynamic network, can model temporal feedback, but you need temporal information. If you have nice hierarchy temporal data, you can use the dynamic network. But the problem is sample size, right? I mean, you don't have enough samples to do that. The other side is to use, relax that. What is nice about Bayesian networks is that you get a valid joint probability distribution. But what you could do is you could say, I don't think there's a good joint probability distribution. Then you can just learn something like dependency network, which allows you to basically kind of predict things, predict one variable as a function of the other, and there you can have cycles. But to be honest, not a lot. Like from a formal point, I don't think I've seen a lot. There's some work from Karen Sachs, which I saw in Journal of Machine Learning Research. They were talking about like causal cycles, causal models, cycles and stuff. I don't know where it went, and I don't know what Karen Sachs did. So yeah, I think those kind of stuff.
So the in-person workshop participants can continue discussion during lunch. I do want to give opportunity to online participants if they have any questions. Okay, so let's thank Sushmita and the team. So we now have lunch, and logistics are behind this wall. So we will exit through the door. The wall is not going to move, but behind the wall is the seating for lunch. And behind another wall with a big door there is the actual buffet that we should get the food from. There are boxes in case you want to take food elsewhere with you. There's some outdoor seating on first come, first serve basis. If you go back to the lobby and kind of go straight out, but I can't guarantee that. So there's designated seating in just behind the wall. So if anybody has any questions, let me know. We will reconvene back at 1:30 p.m. in an hour exactly. Thank you all. I don't guarantee anything, so I am hungry and need to eat. Hello, everybody. We are Hello, everybody. We're about to start our third session, which was originally scheduled as the first, and now that all members of that team are here as well, we can definitely do it. Before we start, does anybody have any questions? Logistical or not scientific ones, logistical. Going once, going twice. In terms of tomorrow's rides to the airport, would it help that I send you flight start times of different people so you can coordinate with others who are leaving around the same time? Maybe, okay. I'll definitely do. I'll send you a bunch of stuff after I'm home. We are going to go with the topic of inference and comparison of biological networks. We kind of touched on this in the previous session, but I think you're going to get complementary discussion this time. And Kimberly is the one who's coordinated that along with her team.
I will do my best to keep track of the time, so we have plenty of time for discussion. There's the laser pointer. All right, thank you. This was supposed to be session one, topic one, but we're doing now, it's inference and comparison of biological networks. And when we were meeting as a group, we kind of settled on four main subtopics within this. And so we're going to break this time up into these four main subtopics. And they're roughly ordered and something that we thought was logical. Of course, they all kind of feed into each other. But we're going to start with the inference of networks, because the idea is before you can do anything, you've got to have a network from somewhere. So how do people actually infer networks? Next, we're going to go into link predictions and their applications, which is highly related, right? What if you have a partial network and you need to try to build in some missingness? Finally, we're going to talk, or next we're going to talk about condition-specific subnetwork inference with the idea that if you already have a network, then you have some data. How do you figure out some sort of important subnetwork within that? And then at the very end, we're going to talk about differential network analysis. Let's say you already have some network and you want to compare them. How is that done? How should it be done? Where is the field going? So subtopic one, inference of networks. These were the slides that I had put together. And I will say this is kind of just a high level overview of the field, kind of what people often talk about in this area in my mind. All right, so how do we infer networks from data? And in particular, I'm mostly talking about omics data in this context. Of course, lots of other ways to infer networks. We heard some about biomedical knowledge. But in this case, you can think about having a biological network. You can't really directly measure it. But what you can measure is you can measure omics data, right? You interrogate the cell. And what we want to do is we want to use that output of the network, the omics data we can measure, to kind of reverse engineer or infer the underlying network. This is an overused cartoon, but I actually do think it's very apt for this. What essentially happens is different people have their favorite omic, or different populations have different omics measured on them. And basically, we're looking at different parts of the elephant. We want to reconstruct the elephant, which is the underlying network, with incomplete data that's only an outside observable. We're kind of like these blind men. All right, so building and interpreting biological networks. I wish I could see these words better because I want to actually talk through these. I think these are some important points. On the right are different types of biological relationships, the types of things we might want to reconstruct, or the types of things we would want to infer. And what I said earlier, biological networks often aren't known a priori, but you have to infer them using genomic data. How a network is inferred or reconstructed, you'll hear me use those interchangeably, often depends both on what data is available. You know, if you only have, you know, transcriptomics data or only epigenetics, that's going to dictate how you build the network and what you want the edges in that network to actually represent. Do you want them to represent causal links or correlative links? Okay. Are they actually going to be physical interactions or some sort of other type of interactions? And finally, the meaning of edges in a reconstructed network depends on the method that is used to generate those edges. That's important for downstream analysis and interpretation. If you're doing something like a correlation network, you don't know causation. It determines how you can actually interpret those. Right, interpreting the edges in biological networks. This is kind of a very high level crude thing, but I kind of like to break down networks into three main types. The first is relational networks, which tend to be undirected. A good example of this are something like protein-protein interactions. Now, of course, those can theoretically be directed, but in a high level sense, you think about two proteins that come together, they dimerize. It's an undirected physical relationship. The second is what I call a correlation network that is also undirected. Two things go up and down together, but you can have signs in this, right? They could be negatively or positively correlated. And then finally, the third type that I like to kind of group things into is what I call a regulatory network. We have things that are directed. A causes B to do something. This can have signs, although these are typically hard to infer in practice, but something could say upregulate or downregulate the target gene. So I'm going to start with correlation networks. There's lots of caveats and limitations to correlation networks, but I think it's an important topic to kind of discuss. So how do correlation networks normally work in the very high level sense? Well, the basic approach is that you have something like gene expression data. It doesn't have to be gene expression. It can be metabolite expression, CPG methylation, any omics data. And you basically use a similarity score to reconstruct a matrix. Similarity score could be Pearson, Spearman, mutual information, you name it, your similarity score, your choice. You threshold it and you analyze it. I put over here three old algorithms from this field, and I don't want to go into them all in depth. They're pretty old, but I put them up here because they're still constantly referenced in the literature. WGCNA is weighted gene co-expression network analysis. People use this a lot. Basically take Pearson correlation networks, scale it, and find modules of genes. Arachne used mutual information, and the idea was correlation is not causation, so let's kind of snip edges to try to get rid of indirect interactions. And the CLR did an extra normalization procedure. And there have been a lot more since then, but again, these are the straw men that everyone likes to cite and then knock down for their new method of choice. So I wanted to go over them briefly. All right, correlation networks are very, very widely used in biology. We can all nitpick correlations, not causation, all we want, but they're ubiquitous, right? And these are just four examples I grabbed from the literature to show how widely they're used. Up here, these are nodes are genes. Down here, there's an example of correlation network where the nodes are metabolites. This is a paper I was actually on where the nodes are foods, where people were trying to study nutrition and diet. So they said, what do we do with this? Let's build a correlation network. And down here, these are nodes that are biomarkers. We have a bunch of patients and nodes are measured and people look at correlation patterns. So they're very, very widely used, including in recent publications and decent high-end journals. All right, but as I keep on iterating, and we all know correlation is not causation. So let's take this example here. You have two genes that are correlated. We know that co-expression can occur when, say, one gene regulates another, right? Could go in either direction. We don't really know. There's some sort of connection. The other reason it can happen is you have something like a regulatory protein that targets two genes, and voila, those two genes are now co-expressed, right? So both represent the same way in a co-expression network. You can't really disentangle these very easily, which leads me to my second type of network that I'm going to touch on in this section, which is regulatory networks. And in particular, I'm going to talk about gene regulatory networks because that's the one I know the most about, but this could also be things like metabolic regulatory networks and things like that. All right, regulatory networks. All right, so gene regulation is incredibly complex. And if we weren't trying to cover every single topic in this field, I could probably spend 10 minutes on the slide talking about all this different data and all the different caveats and what it means. But in the end of the day, what actually happens is we have different types of regulatory mechanisms, protein interactions, chromatin structure, chromatin state, methylation, microRNAs, protein expression. And we want to reduce that down to something that we can model as a network. And when I talk about gene regulatory networks, I normally talk about transcription factors, regulating target genes. And some of the target genes are also themselves transcription factors. All right, so how have these been modeled? I like to think about there being two main ways to actually produce this type of network in a reasonable way. The first is experimentally. If you had infinite amount of money and infinite amount of resources, you can imagine taking transcription factor antibodies, interrogating the entire genome in your particular cell of choice, and building this network. And that would work, but it has a lot of limitations. First, it's very expensive. None of us have infinite resources. And also there's actually limited number of experimental antibodies you can use to actually build these transcription factor gene connections. But the strength is it's high quality and it's context specific. The second is computationally. You can take something like a transcription factor, you know something about where it might bind in the genome based on its sequence. And say, okay, we know that there's a DNA sequence map to the transcription factor here, the promoter region of a gene. Let's just say that transcription factor regulates that gene. There are still limitations. We still only know these recognition sequences for about 50% of transcription factors, and they're of various quality. And also it's very prone to false positives and not context specific, but it's really cheap. You have a high-end computer, you can run this, in a matter of hours these days. So what's actually happened for gene regulatory networks is we've had a combination. We want to take something like experimental data and combine it with computational predictions. So I'm going to touch on two of those, and then we'll go on to our second subtopic. First, methods that use gene expression data. And this is a figure from an old paper now from 2012, where this was actually one of the dream challenges. So someone from Sage mentioned this earlier. This was an example of that, where they actually had data they provided to the user base, where they said, put your algorithm of choice, try to predict this gene regulatory network, and then they benchmarked it systematically. So on the x-axis are the different methods, the y-axis is the AUPR. And silico data, they did pretty well. E. coli, they did pretty well, but when they got down to yeast, in my opinion, that looks like nothing really worked. And I showed this slide to people again and again and again, because even though it's been 10 years, I think that for the most part, people who try to apply algorithms to high-level organisms will see this trend, that they're really, really, really hard to predict. But that being said, it's worthwhile to mention that there are a number of methods in this area. I'm mentioning 3 here. I'm mostly mentioning these 3 because they're highly cited, and I see them benchmarked against again and again and again in the literature. But this is an explosive field. There are a lot more out there. So inferolator is one that is still maintained, that uses linear regression, OM shrinkage, and lasso to basically combine transcription factor to gene predictions with gene expression data. Genie3 that uses tree-based and ensemble methods. And then Panda is one that my group developed that actually tried to integrate multiple types of omics data to find consistent structures that way. The second type of omics data that I've seen a lot in gene regulatory networks is epigenetic data. So in this case, we have an example where I talk about here, there's a DNA, and you have predicted transcription factor binding sites along it and open chromatin. You can imagine you can use that to basically prune the sites and thus prune your network. And this is an approach that is taken when you have high quality epigenetic data. And then going forward, here are just a couple papers and resources that use that approach. This was one of the earliest papers that came out that did this. They had DNA hypersensitivity and a number of cell lines, and they tried to build transcription factor to transcription factor networks to each of the cell lines. This is a more recent approach, TEPIC, that basically combines transcription factor binding affinities with annotation data, including distance from the TSS and chromatin data. And then SPIDER basically identifies consistent structures using the Panda method, but taking into account the epigenetic context. And so those are the gene regulatory networks and the co-expression networks. I'll end with some discussion points, and then we'll move on to the protein interaction networking and prediction. So first off, correlation and co-expression networks are easy to use, and they're widely applied, right? We can't avoid that fact. They're very easy and used a lot. However, they have multiple limitations. Correlation is not causation, and it's difficult to really know what the edges mean. On the other hand, gene regulatory networks are really difficult to infer. And also, there are a ton of methods, but the accuracy remains low. And it's very challenging to know how to take all that omics data I talked about and actually use it to infer a regulatory network. All right. I believe the next subtopic in the same theme is Mona and Lenore, and they will be,
Lenore will be able to present, just tell us when.
yeah.
Okay, so Lenore and I decided to tag team this, which would work perfectly if you were here, but make it a little more complicated.
Use the mic so that.
Okay, and how do I, which one is that?
This is the, oops, oh, I see.
How do you go backwards?
Got it.
Okay, I'm gonna go to the next slide and Lenore is gonna start off. Lenore, are you there?
I'm there. Can people hear me?
Perfectly, yeah.
Give us just a second, Lenore.
The only problem is I can't see the screen anymore. Can somebody share the screen to the Zoom so I can actually see my slides while I talk, or is that not going to work?
Give us just a second to set everything up, please.
Oh, perfect. Thanks so much. Can everybody hear me and see my slide? Great. Okay. So Kimberly has given a beautiful introduction into regulatory networks. I am going to switch gears and go back to sort of the classical physical protein-protein interaction networks. And these are the networks that are built from trying to, from experimental evidence that protein chains physically bind in the cell. Here we have an example of a protein complex, and we're going to abstract out all that wonderful 3D structure information and just basically say this little purple thing binds to the orange thing, which is in a clique with the blue thing and the green thing. And if you'll notice, this is what you would get from like a yeast to hybrid type experiment. If I was doing a co-IP experiment, then actually it witnesses co-complex membership. So I would actually also get edges between the purple and the blue and the purple and the green. If I look at all the different experimental interactions in one source, BioGrid, you can see about how many of them have been collected in these public databases. And you also see that humans are where we have the most, though I guess if you mod out by the size of the genome, we also have significant in yeast, and everything else we have much fewer. And once we get beyond just a few model organisms, we don't have any data. Next slide. Okay, so as many people in different sessions today have already alluded to, in these types of networks, there are many different ways that people use the existing graph theoretic interconnection structure of the network to predict possible additional edges. So when we get this BioGrid picture, we already assume some of the edges are wrong because they're experimental error, but even more, we assume that many, many edges are missing because not all pairs of proteins have been tested for interaction. So therefore, inferring additional protein-protein interactions computationally can be extremely useful. Very simple methods include just counting the number of common neighbors. Other simple local neighborhood methods like Adame Kadar, other things have been used also in social networks. Going to say something a little bit later about this L3 measure that was introduced by Kovac et al. And a lot of people have already been talking about random walk diffusion methods, network propagation methods, different ways of taking short random walks in order to see which nodes are kind of hit most often and in the neighborhood and nearby. And there's subtleties about the different ones of these methods that people use in different settings. Embedding methods often also have underlying a kernel that is random walk based for these methods. Glide is a method of ours. There's now Glider as well. And this little schema is from the review paper that I did with other people who are at this workshop, either here or at Zoom, where we looked at all the different ways that sort of random walk-based methods have been used to date in biological networks. But since then, they've gotten with, so Ben Raphael was involved in that work, as was Roded Sharon, who actually Roded convened us, and they're also participating in this workshop. So next slide. So this is just sort of highlighting the fact that which metric you use to talk about which nodes are most likely to have links can actually affect very strongly what you do. And it can be different than what you might want if you're asking a different question, such as functional relatedness. So the common neighbors measure. So here's a gene PARC2, and we're looking at its closest neighbors, 20 neighbors, using sort of the common neighbors measure. It's a weighted measure that I am completely describing, but basically you're closer by this measure if you have many common neighbors in the network. And by this measure, these are the proteins that are closest to this gene PARC2. Actually, this is the ones, I'm sorry, they're closest to the gene PINK1. PARC2 is also a very well-known Parkinson gene. And we're looking at the closest genes by this weighted common neighbors measure. And you'll see that in terms of the known links, these genes are quite disconnected. There are even some disconnected genes. And the reason is, of course, there's some other nodes in there that they're mutually connected to, but there's a lot here that isn't directly connected to PINK1. On the other hand, this recent measure L3 of Kovac et al. Uses sort of in a graphlet sense, a very different interconnection pattern, which pays attention to the fact that protein-protein physical interactions more often have kind of a lock and key mechanism, where you would assume the locks bind to keys, but the keys would not bind to keys, or the locks would not bind to locks. So by this L3 measure, when we look at the closest 20 genes to this PINK1, we get all these genes that in fact are known to be direct interactors with PINK1. And this gene over here, which maybe now I predict is a direct interactor with PINK1. So for actual direct link prediction, it might actually matter a little bit more which version of random walk, which embedding, or which measure of similarity I use. Okay, I'm going to turn this over to Mona.
All right, my turn. So I'm going to, follow on Lenore's nice introduction. So I wanted to start by saying that, the methods that Lenore has been talking about for link prediction are largely based on exploiting or leveraging topology in these large-scale protein interaction networks. And it's really amazing that just the topology of a network actually reflects so many important things about biology. So for example, it was shown, from the very early days that high degree nodes tend to be essential. That's been something that's been seen over and over again. Or, and we've already heard about this, I think earlier in the morning, we know that if you look at these protein interaction networks, there's a structure to them. They have modularity, and then these modules within these networks, which are just, again, defined topologically, correspond to coherent biological processes or complexes. These topological features can also actually reflect dynamics of biological networks as well, or of proteins. So for example, if you look at the clustering coefficient of a protein, if that number is high, then in general, the protein is going to be co-expressed with its neighbors in taking part of a complex. So it can actually represent, topology can also represent dynamics of proteins. And then people have shown that you can identify various aspects of disease genes, or you can use as a feature to identify disease genes. And we had an example this morning as well, by just looking at differences in topology of disease genes. But I think if you, so then it's natural to think about link prediction with topology in mind. How can you use topology to close out and find these links that we're missing, or maybe even to get rid of erroneous edges that are there. But I think it's important to look under the hood a little bit and see that these nodes have various types of well-known features. And this is before getting too deep into sequences, which we'll talk a little bit more about in a second, I think. People have determined, for example, particular annotations over the years that are associated with these proteins. They've also know things about structures. They've also, labeled these proteins with respect to what domains or peptides of functional importance that they contain. So one thing we thought a lot about in the past is when you think about these networks, can we extend the topology-based measures just a little bit to include some of this additional information? So there are also these recurring patterns. We heard about network motifs in the morning that are purely topological based units, but you can also have an extension of that where you have a topology and you have an annotation on a node. And this is something that we've called network schemas in the past, but they're a natural extension of network topologies. And so on the left here is a schema. It's just a straight line corresponding to something that's related to RAS signaling, where this RAS protein is regulated by this GAP protein, and it in turn regulates this kinase. If you look at a protein-protein interaction network, and we're talking about physical interactions here, you would see many such instances of this schema. And so this is a little piece of a yeast interaction network from a while back. But you can look at higher order combinations as well. And so you can look at pairs to start, triplets, as we just talked about. You could look at triangles, which maybe help capture complexes, and you can look for kind of branching decisions starting as these Y stars is what we call them. And so you can ask, if you look at a large scale biological network, do any of these network schemas tend to be enriched within them? So at the pairwise level, it's clear there are many of these common patterns that you'll see when you look at any biological network. In this particular example, it's looking at the yeast protein interaction network, and it's just looking to see pairs of PFAM domains that are enriched in these networks. And of course, many people have looked at domain-domain interactions in the past. And so this is another type of network. This is the network of annotations that tend to recur more often than you expect in the protein-protein interaction network. But you can do this. Oh, and if you look at them, these things make sense. So here are some pairwise schemas that are involved in signaling. And here's a sub part of this network that's involved in transport. But you can look at higher order topologies as well. And here what you would see is that there are higher order schemas that also repeat, and they repeat and they're statistically enriched even when you correct for the pairwise schemas that you observe. So, we have multiple of these schemas that recur in interaction networks, and multiple of these schemas that recur more than you would expect based on their pairwise compositions. And this is just looking at PFAM domains, and of course you could do that on any annotation type. And so I think a very interesting thing to think about for link prediction, at least for topological based link prediction, is to think about link prediction with respect to topology, but also to these very simple annotations that exist. And I think that those would be ways in which link prediction methods would certainly get kind of more power. And so I want to make a little bit of a distinction here between link prediction when you're looking at protein-protein physical interactions versus link prediction when you're looking at the types of co-expression networks that we just heard about, or functional networks. So you would tend to see these types of patterns that correspond to, say, a signaling cascade in a protein-protein physical interaction network, but you wouldn't expect to see the same types of patterns in a co-expression or functional network. So depending on the type of link prediction you're trying to do, depending on the type of domain that you have, these types of annotations would be more or less important. But to me, they're a really natural way in which to start thinking about extending link prediction when you're using topological measures. And I think we're back to Lenore. Yeah.
Okay, well, and now for something completely different. Rather than looking overall at the network, if we're predicting protein-protein physical interactions, we have a lot of individual data in the protein chains. In fact, we have the protein sequence, and from the protein sequence, we have the protein structure. And in fact, it is possible to take a pair of protein sequences and devise a method that can predict just based on the pairwise protein sequence data if there is a predicted interaction. And a lot of the methods that people have been using to do this are based on protein language models and deep learning using some kind of neural net architecture. And there are several methods out there, including Piper and others, which we will discuss in a bit. Next slide. So our offering in this space, and this is joint with Bonnie Berger's lab, is we've worked on a method that we call Descript, which seems to be better at predicting PPIs in a new species when trained on a different species than some of the other deep learning methods, which do very well when trained on human data and then are asked to predict PPIs in human data, but other methods do less well when trained on human data and say asked to predict PPI in fly or mouse. So this is just a little schema of the architecture of the model. We try to be very interpretable. So it's doing two things. One is that it's leveraging already some kind of structural information that comes, as Mona said, from PFAM, and some knowledge of structural domains that are embedded and learned by the language model for individual sequences. But then it tries to somehow learn a contact map between these sequences. So, and that makes what it's doing a little bit more interpretable and a little more generalizable, because it probably has to learn something from structure if it's trying to learn sort of how they interact. So we never read that out. The readout of the model when it's end-to-end trained is just the binary interaction prediction. And but this is some sort of evidence that in the middle, when it's saying, yes, I think these two proteins interact, that it really does somehow have some sense of what that interaction surface looks like. Okay, next slide. So at ISMB in July, we're presenting new work that tries to combine these two views. So one question is, if I could do a network prediction and I could do a sequence prediction, how could I somehow leverage them both? So the first way we ask this is in a setting where we don't have PPI networks at all, So maybe we're trying to predict PPIs in a non-model organism where we have no existing experimental evidence. But we're still somehow guiding the training by this global knowledge of network topology by one of the embedding methods. So somehow, when we're learning what makes two proteins interact, somehow we're learning something from the structure at the bottom level, and somehow we're learning something about maybe which nodes are most promiscuous or various other things from the network level. But again, this is topsy-turvy is a method that doesn't need any network information to predict. It just takes as input two sequences and predicts. It's just in training that it's somehow learning something about the network. Next slide. And then if you're in a species where you have both, where you have like humans or fly or any one of those species I showed in the beginning, you might want to ask, well, is it better to predict interactions, missing links from the network, or is it better to predict them from sequence? And of course, as you would expect, somehow we want to combine, right? We want to predict from sequence when we're not confident in the network, and we want to predict from the network. So some people say the sequence really tells you in a social network analogy which two people are compatible to be friends, but the network tells you which two people have actually met. So you do get different kinds of information from this kind of data. And in fact, we're able to do even better at correctly predicting links and cross-validation using this TT hybrid new method that uses both the topsy-turvy on just the sequences and then our glider method on the networks. So I do want to make, so this is early days. I mean, this is a method that we don't know a lot of methods that do both, but I think this is a great open problem to refine and improve these kinds of methods. I would caution that the issue of ascertainment bias becomes a little bit dangerous when trying to evaluate results. And it's really hard to measure or compete a method that's working with sequence data against a method that's working with network data on the current network data sets we have. And the reason is that, in fact, the experiments that the biologists have done are biased by pairs of protein they already think maybe interact, or proteins in the neighborhoods of some very important disease proteins or other proteins like this. So as a result, we've probably overrepresented the popular edges that network methods would infer, and we've probably underestimated true PPI sequence-based pairs that we haven't tested. Now, there's Curie, which is the human interaction network that of Luck et al. That recently came out that does a completely unbiased yeast to hybrid screen of a portion of human proteins. So if you start with purely unbiased data, then you might not have this problem. Or you could look at other ways to sort of ameliorate the ascertainment bias if you want to compare these two types of data. Okay, I will stop there and back to Mona.
All right, so I wanted to switch just for a second from networks to sequence. So, you know, we know that there's a lot of variation across the human population. We look around the room, we can see a diversity of phenotypes here. And these, of course, depend on genomic sequence. And so if you look at our genomic sequences, there are places in the genome where we vary. We're mostly the same, but we also vary as well. And then, of course, this is also super interesting in the context of disease, whether it's germline variants in inherited diseases or it's acquired mutations in diseases such as cancer. And one important thing I think we want to think about as a field, we've talked a lot about like context-specific networks and you know, temporally changing networks, but there's also networks across populations, whether healthy populations or disease populations. And so I think it's really kind of important to think about the effects of variation and mutations on networks themselves. And so just taking a protein-centric view here, you can think, to start anyway, you can think about a protein sequence and you can think about a mutation, and maybe that mutation actually adds an edge to a network. Maybe you have another mutation, maybe it changes the interaction between those two proteins, I think P3 and P4. You get another mutation, and maybe that's neutral, it has no effect on the network at all. Another mutation, and maybe it's so detrimental it kills the entire protein, affecting a whole bunch of interactions that you have. You can also have mutations outside of protein sequences, of course, and so you get a mutation in a regulatory region, and maybe that removes this regulatory interaction between the regulator and its target gene. So just thinking about this variation you can't, you have to look a little bit as to see where the variation is and what the specific alterations are. So certain positions or mutations are important for interaction networks. And this comes partly to trying to understand, the 3D structure of proteins and how they interact with ligands. So of course, you can imagine that the positions that are in contact with the ligand are going to be more important for the interactions that they make. And so I think one of the really exciting things to think about in the future is to try to predict these personalized networks. And there are kind of two ways to think about it. On the one hand, you can do predictions sort of de novo. So the types of things that Lenore just talked about, why can't we go and look at all the variants and see, how they would alter various kinds of interactions, whether it's protein-protein physical interactions or protein DNA interactions, protein small molecule interactions, et cetera. The second type of approach is, well, we already have a lot of information about networks already. How can we predict the impact of variation or mutations on existing networks? And so that's an approach that we've taken both in the disease context and shown that these disease proteins tend to accumulate mutations in positions that participate in interactions, but we've also done it in another setting. And so I just wanted to tell you a little bit about that before stopping. And so here we thought about, and this is work we just published in Recom, about predicting the impact of mutations on protein DNA interactions. And here we started off, if you look at the left, how does the pointer work? Which one is it? Okay, there we go. So, from studies like ENCODE or other large scale studies, we have lots of examples where we know a protein and we know kind of the binding preferences of this transcription factor protein. But what we don't actually know is what this interface is like. So we have the edge, we don't have the interface. In many cases, we can have models of what this interface is like. So here's the protein side, these are different positions. And it's not that all of these types of protein DNA interactions happen this way. This is for this homeodomain class of proteins, but a lot of them do. So you can come up with contact maps for certain restricted cases. And so then we showed that you can, if you have a contact model like this and you have kind of data that looks like this, you can actually learn energy terms and this kind of registration on a contact map simultaneously. So you're learning a structural interface. And then in the end, what that means is that once you've figured out the structural interface, and this is structural interface loosely defined, meaning I know how this protein, what its rough contact map is with this DNA motif, then you can think about whether you have a mutation that occurs in a particular protein position and what its effect would be, or which positions of DNA it's likely to affect in terms of binding. So this is just one type of way that you can think about mutations and the effect that they have on known binding interfaces. So overall, I guess I'd like to say that I think that the field as a whole should start thinking about the impact of variation on networks, and particularly thinking about like the diversity of populations that we have. So we have many different alleles, and there are whole families of proteins that are hyperpolymorphic, like MHC, and trying to think about methods for predicting these types of interactions when we know maybe roughly what the interactions are like or which things interact with each other, but what the impact of this variation is. And so I think that's it for me, and I'm passing it on to Ben.
Okay. The lasers on the back. Perfect. Okay, great. So we've, at least in our group thus far, talked about constructing networks, talked about predicting new links in networks. And so I was sort of tasked to talk about what we called condition-specific network inference, which is really how we use networks to really interpret and analyze other types of data. And we heard a little bit about this morning, and this sort of problem arises in many applications in analyzing biological data. I just put a few here. Sort of the classic function prediction problem, which is all of these are premised on starting with a biological network, say a protein-protein interaction network. And so if you have such a PPI network and you know something about some genes here being implicated in I can't read what that is, metabolism, then you might be able to say that other genes that are unannotated in the same subnetwork will probably also have that same function. Or if you've perhaps done a large-scale measurement, like maybe you've done a genome-wide association study, and you identify that a few genes here in red are sort of have a significant association with height in this case, but then by also using an interaction network, you might be able to pull out some coherent subnetwork that involves many other genes, which were not significant, didn't rise to the level of statistical significance. And this might be sort of some indication that it's this group of genes, this subnetwork that is associated with height. And a similar story in cancer, where you might look at somatic mutations, and many of these mutations are rare, and so you might not be able to identify all of the cancer-causing mutations just by their frequency or statistical significance individually, but if you combine them on top of a network, you might be able to then pull out sort of coherent subnetworks of cancer genes. So, this is a problem that's been well worked on by members of the community in this room, many others, and I think we've maybe already seen both these review articles listed, and there's actually even dozens of review articles with many methods on this topic. And just to sort of describe them at a high level, I would sort of classify them in really just sort of two major approaches, which I'll refer to here as network ranking and altered subnetworks. And so in both cases, you start with some interaction network and some labels on the genes. On the top here, these labels are binary. They might know the function of some genes and not know the function of others. And then in this network ranking approach, I then use some measure of network similarity. We've heard about a few of these, and I'll talk about these in a second. And then some sort of algorithm that runs through the similarity measure, and then we'll rank the genes according to their similarity. And I can sort of threshold this and maybe get some prediction about which other genes have that same function, for example. Similarly, but slightly different, I might think of having a score on each node of the subnetwork. It could again be binary, or in this case, it's shown as some sort of continuous score. Again, through sort of network similarity, I pull out, in this case, sort of these high scoring subnetworks. Okay, now I sort of showed this in a slightly different way because, of course, all of these nodes are actually on the network. So I could really think of having these rankings on the subnetworks as well, except there's sort of a subtle difference between these two approaches that I'll discuss in a slide or two. Now, these similarity measures we've heard already a lot about, sort of historically, maybe the simplest was to just look at direct neighbors and then sort of shortest paths. We heard about this morning, and then sort of more sophisticated measures that sort of propagate information on networks using random walks or diffusion or sort of network flow approaches. And so, again, we've already seen this and heard about this. So network propagation has really become one of the most popular approaches, I would say. And that really in and of itself is a term that actually encompasses numerous such approaches that involve sort of random walks on graphs, as shown in this approach. Equivalently, many of these random walks can be described as fluid or sort of heat diffusion on a graph. And we showed, Lenore already mentioned this review article, sort of how really a lot of these approaches, what they really boil down to is this process, which is often described in this iterative way of, you take iterative steps of a random walk or flows, or really just can be written down in terms of a network similarity. So the random walk, restart, diffusion kernel, nearly everything people write down really just is a similarity matrix, and network propagation, this becomes as simple as matrix vector multiplication. So it also makes it highly efficient. Just want to highlight kind of one sort of twist on this, which is sort of using kind of this propagation approach twice to really merge both those ideas. So one where you might have these binary scores of prior knowledge. This is actually work of Mona's, where you can sort of propagate this prior knowledge and then use that in and of itself to then define a new random walk, which then you can take your new information, say your somatic mutations in cancer, sort of random walk again and get sort of another prioritization. So there are many variations of these ideas. However, and this is the point I wanted to mention about the difference between ranking and subnetwork identification, is that often, especially with the more sophisticated measures, what you are really getting is just the ranking. Okay, so network propagation actually just gives you a score on each vertex. In and of itself, it doesn't pull out a subnetwork. So you actually need some other sort of scheme to decide that maybe this is your high scoring subnetwork following propagation. And if you go dig into these papers, and I pulled out sort of two, one from Rhoda Sharon's group that sort of describes how then after propagation, they go pull in densely connected subgraphs. Or in our approach, we said, go back in and pull out these strongly connected components. So there's some other heuristic going on to get the subnetworks. It's not really just that flow measure. And this is an important distinction because it really says that the topological properties are being used in a way twice and sometimes differently. So it's the random walk that's being used to change the scores, but then maybe it's actually cliques that are being pulled out of the network after these scores, okay? So there's a lot of sort of subtleties under the hood. And so, because of some of these subtleties, we sort of wanted to see if there was a simple generative model we could write down to sort of encompass what a lot of these approaches were trying to do. And so what we came up with is what we call the altered subnetwork distribution. So a very simple statistical model that's very flexible. So you start with your interaction network, here a graph. You have this subnetwork family, which I'm going to talk about in a second. This is sort of your prior on what you think the interesting possible subnetworks could be. And what you want to find is an altered subnetwork from that family, okay? You're also then given these vertex scores. And the vertex scores are distributed in two ways. They have a background distribution here, or for nodes that are in this altered subnetwork that you're going to implant, they have this altered distribution, okay? So now you've got sort of these parameters you can use to describe different approaches. What is the subnetwork family? And what are these background and altered distributions? So for simplicity, you could think of these scores as coming from two normal distributions. And this becomes like a network mixture model, if you will, where the background model is a normal distribution with mean 0. And then in your altered subnetwork, your scores are higher. So you have some higher mean mu, okay? The subnetwork families, you can then think of all the different approaches people have tried. You could think of connected subgraphs, pulling out connected subgraphs with high scores, or maybe you want edge dense subnetworks, or you want cliques, or maybe you want graphs with minimum cut size. Maybe you want somehow propagation subnetworks, whatever that means. But now, by writing things down this way, we have a version of the problem that's really a statistical estimation problem, because then we could say, all right, given a graph and given the subnetwork family, now from vertex scores, find the altered subnetwork. Okay. So now all that is a problem of estimating the parameters of this altered subnetwork distribution. And then by changing the subnetwork family, or by changing these distributions, I can then model various different problems that have been presented in the literature. And then what we can do is start to describe which versions of this problem are easy and which versions are hard. So thinking first about the vertex scores, if the background distribution and the altered distribution for my subnetwork are pretty far apart, then that's a really easy version of this problem. In fact, so easy that I don't even need the network anymore. I don't need the network information. Be very clear if I want to separate two normals. If their means are very far apart, I don't need this prior knowledge of the network. So that's easy. Of course, if those distributions are completely overlapping, the problem is hard, or maybe even impossible. My subnetwork scores are not much higher than my background scores. And then on the other axis, we can think about the subnetwork family itself also. So if the subnetwork family is really large, in the limit, we could think of the subnetwork family including all sets of nodes, 2 to the n, right? And then there's no structure at all. So of course, that's a really hard version of the problem. Or we can think about the subnetwork family involving one subnetwork. And that's a very easy version of the problem. In fact, that's just equivalent to doing like a test between two sets of nodes, like a t-test if it's the normal distribution. So now you have these two axes to think about easy and hard versions of the problem. And so the sort of interesting versions of the problem are somehow right here in the sweet spot in the middle. Okay, so you can think about where your problem is it in the sweet spot compared to very easy versions of this problem, which are what we call the scores only version, where the distributions are so far apart that you don't even need the network, or the version where the network alone is enough and you don't need the scores. There's only one subnetwork, you can just test them directly. Okay, so these are create interesting baselines of comparison. Right, there's a lot to say. I'm just going to actually give 3 sort of summary discussion points here in the interest of time. The first one is that actually many studies, we claim nearly every study, doesn't compare to the correct scores only and the correct network-only baseline. And by correct network-only baseline, what I mean is that you have to use your network information in the same way that your algorithm is using it. Okay, so for example, if I do network propagation with random walk of restart, then I should compare to page rank centrality, which is random walk with restart without any scores, to show that my network method is doing better than baseline. If I compare to like direct nearest neighbor interactions, then I haven't sort of said whether my algorithm is doing better because it used a different network similarity, or if it's doing better because it actually did something interesting for this problem. Okay, so just to show you, we sort of reanalyzed some data from a study that had done network propagation with GWAS with network, and we showed that actually when you looked at it in this way, for what did they do? 8 GWAS studies, 3 of them you could just get the same or better performance only using the network, meaning you didn't even need to do the GWAS to pick out the genes for that study. They were already encoded in the structure of the network, or the GWAS alone was enough. And you'd actually do better by just ignoring the network and just looking at the GWAS p-values and ranking genes. And it was only 3 of the 8 where actually both pieces of information were useful. And this is all sort of with a specific network propagation, random walk with restart type of centrality. But now you can do this with, choose your favorite algorithm as this last part and see if you can do better than these 2 baselines. That's one point. The second point is that many of the algorithms that we've developed, and I say we meaning even my group, so not us collectively, don't really have good statistical guarantees of when they work and when they don't. And so we investigated this in using this sort of altered subnetwork distribution type of framework for one of the seminal methods from Trey Addiker's group called J-active modules, which was a method that was developed for gene expression. And what it did was it found maximum scoring connected subgraphs. Okay, so now we know in our terminology, that means the subnetwork family are connected subgraphs. In fact, the scores were normal scores because they use z-scores. What we actually could show is that the score that they used was in fact the maximum likelihood estimator of the statistical problem, which sounds good. They're doing maximum likelihood estimation, which they didn't even know in the original papers. It's kind of cool that that's what they did. However, that maximum likelihood estimator is biased. You can actually show this. What that means is it explains a phenomenon that had been known for 15 years or 20 years since this algorithm existed, which is that it tended to produce very large subnetworks. It's actually just a feature of the statistical problem and the fact that they're using this connected subnetwork distribution. And then the third summary point that I'll sort of say is that the choice of this family, now that you expose all the parameters of the algorithm, then you can sort of ask, well, what topological assumption should we use that corresponds to, in this statistical estimation problem, which subnetwork family should we use? Do you want connected subgraphs? Do you want dense subgraphs, minimum cut? Do you want sort of somehow propagation? What does it even mean to have a subnetwork family defined by a random walk? And thus far, I would say a lot of our algorithms are really make this choice just empirically, or just almost even randomly, what seems like reasonable. And I think we should maybe think about which of these choices make sense in different particular applications. So I'll stand there and we can have some discussion maybe at some of these points.
I think you did. All right, I'm going to wrap up with subtopic 4. I'm going to try to keep it to our agreed upon 10 minutes if at all possible. And Holly and I are tag teaming this. I have the first set and I'm going to turn it over to her to wrap it up. So our final subtopic is differential network analysis. And I was going to start with a quick story. I'll make it brief. I like to introduce it this way. Let's pretend you have 2 patients, Susan and Jane. They grow up, they're healthy. They have various habits throughout their life, such as eating burgers or wine. As they get old, they have a similar disease, similar phenotype. They both go to the clinic. The doctor gives them the same drug. And lo and behold, you can see if you look closely, Susan gets better. She's smiling again, but Jane doesn't. So the underlying hypothesis of differential network analysis and why we compare biological networks is this. We can say that early on, both Susan and Jane, even though they have different networks, they're both healthy, they're both functioning. But as they get older, their network develops mutations, perturbations in such a way that they become diseased, but they become diseased in different ways that even though they have a similar phenotype, they might respond differently to something like a therapy. So differential network analysis, kind of at a high level, is generally done in two ways, at least in kind of the omics inferred network framework. One is in identifying differences in edges. So this is a paper from my group in 2015, where we compared networks inferred in two different subtypes of ovarian cancer, grabbed the edges that were most different and said, aha, here's what we think is driving these differences in these subtypes. Another is looking at differences in the nodes, such as differences in targeting. We look at something like a centrality score, a betweenness, a degree centrality, et cetera. And you basically say, okay, so the genes are targeted differently in these different networks. Maybe they're corresponding to different pathways. Maybe those pathways are important for disease. And I wanted to make the point that these two examples on the left are pairwise analyses, but they don't have to be. This is a paper from 2021 from my group, where we actually did 38 different networks, and we did 38 pairwise comparisons to say what's different in each particular tissue, and said what were the pathways in each. So the other way we often think about network comparisons is not just two groups. I talked about how we have 30 different tissues, and Mona said very nicely, what about different individuals, right? We like to think about, oh, you have subtype A or subtype B, but sometimes different people have different ages, different BMIs, different sex, different covariates, maybe they're smoking or not smoking, et cetera. So unfortunately, most of the algorithms we have to generate networks, at least with omics data, things like transcriptomics data especially, generate one network. And that actually represents information from all the different input samples. And if you have two really well-defined phenotypes, maybe you can take omics data from one phenotype, omics data from another, and compare them and get something. But in reality, everyone's just a little bit different, right? Even going back to that example of Susan and Jane, they're just a little bit different. And so more recent approaches have tried to basically take some of the outputs of these algorithms and try to generate networks for individual input samples. And I wanted to highlight two here. There are some more recent ones, but they're mostly on the bioRxiv these days, so I didn't want to throw them out there yet. These are two methods that have been published for estimating sample-specific networks. They have the same basic underlying approach. Let's say you have an algorithm to infer networks using all samples in a data set. You can apply that algorithm to all samples and all samples minus one, and then use mathematical trickery. It's not really trickery. They both have very well-established derivations to try to reverse engineer whatever that sample-specific network is. And why is this important? Well, this is important because it lets us get into some of the nitty-gritties of heterogeneity. So I was going to throw out one example. I'll go through it quickly for time. This is an example driven by someone I work with, Camila Lopez Ramos, on sexual dimorphism, which she was comparing males and females. Of course, this is important to understand the difference between men and women for health and disease, but often the women recruited in a study might be younger, they might weigh less, right? We know those differences are really men versus women or having to do with fat content and BMI, right? So what she did is she basically did a differential expression between all men and women, where she could actually model all sorts of covariates, things that were important. And this is standard bioinformatics analysis, has nothing to do with networks. But in the context of networks, we only had one network for men and women most of the time. But she was able to actually apply the blindness approach and got an individual sample network. And then what she could do is she could run the same basic statistical equation to find edges that are different between men and women. And she could compare that to say, okay, here's the differential targeting, the differential edge weight as a t statistic against the differential expression, t statistic, and actually find correlations that way. And this is one example where there's still two groups, right? It doesn't have to be men and women. It could be disease healthy. It could be a continuous variable, like something like past years of smoking or something like that. And so that's kind of, I think, where networks are going. And I'm going to actually turn it over to Holly next. And as she walks up, I'll say that I think one reason why this is a nice transition is Holly's going to talk about dynamical networks. And if you have sample-specific networks, you can start thinking about how those individual networks actually change over time in a dynamic sense as well. So I'll turn it over to you. Did I do it the wrong way? Sorry. Yeah. No.
The other slide. Look the other remote, not that remote.
This one? Oh, okay, thank you. Okay, cool. So as Kimberly has introduced, and we see lots of examples in today's talk workshop, is like we're talking about networks, right? And when we try to analyze the difference between two networks, the object of our analyze is the moleculars representing different conditions, right? And when we see these different conditions have lots of connections, because we see, even we're focused on difference, there are lots of nodes and links shared between this differential network. So when the different condition can be assembled along some orders, for example, time course, then we're interested, the new question is how this transition from one stable condition translated to another stable conditions, or what's driving this kind of transitions. One particular example often cited in biological science is this Wallington landscape in the development biologists, right? I want to emphasize here is when we say stable condition, it's kind of a group of a system. For example, the system means cell, single cell, or individual, could be both of the case. And when stable state, that means the system is stable, is resistant to small stimulus, right? You can see it still keeps what it is. Okay, that means stable. And but we all know more and more evidence shows in the biological field there also exist lots of transition states. This transition state currently popular talk in the biological field are two types. I'm saying it's reviewed last year. It's talking about pitchfork transition. That means the early state can be translated to multiple options, and this subsequent multiple options has a little competition. And another mostly often cited term, we call it set node transition. They have a beautiful example in clinical biology. It's like this, is this like in the middle? This pre-disease stage, right? It's in translation of bioinformatics, we call it early warning signature. We want to make a diagnosis when the disease can be obviously diagnosed. We know from the genome omics data, we can find some early warning signature. Another example very well cited fitting this scenario is in development biology, when the progenitor cell committed to some fully differentiated cell. By commitment, that means this transition cannot reverse, okay? So, and computational work to study this transition, currently, in my knowledge, there are two types of method. One is we call the energy-based approach. That's based on the observation. When the transition happens, the system are unstable, or at least a group of molecular unstable, you can observe the increased local energy, or we call it in different words, you name it, perturbation, something like that. Another method, which my group currently is interested in, is based on this tipping point theory. Like I exampled before, the tipping point theory means this transition is not only irreversible, but also very quickly, abruptly. And we know when this transition happens, they have two features. First, a group of molecular gain new links among them. Second, the overall system to system similarity disappeared. That's two features. Based on the two features, we have these tools developed. And okay, I want to emphasize a little bit here. You can see from the, what can I do this? Okay, fine. Let me see here. You can see this node, we have the shared nodes. But earlier stable conditions, right? The links between this, what changes the links between these nodes. That's very interesting to study. And I know in my biological colleagues interested in among this one, who, for example, my example, in the vector methodology like me, is ETV2 only is a transcript factor, only abruptly depressed in this earlier progenitor states, then it shut down. It plays a kind of role to more module the transition states. It's performant and it's linked to what is potential targets inside the feature. What's the potential next stable states? Compare these two types of method, right? And we can think about a different angle. For when review thinks about is we think, but I believe that coexistence in biological system, the transition could happen in a very, very slow, continuous model. Or sometimes, like my example, this ETV2 module, the regulation subnetwork happened suddenly irreversible. So this energy-based model can be applied to both cases. But the tipping point-based model only study this sudden irreversible translation. However, in our recently study 6 single cell level the time course data development biology, we see this. We apply these two methods to the same data sets. This result converged on the same prediction of when the transition happened. What makes them different is what driving this transition. The predicted potential driver role of transcription factor is a little different because our theory or our focus are different. You know what I will describe, the feature is different. Finally, yeah, it's too hard for me to ending this, but for the future direction, right? I believe everyone here have your own sense of what will go. Here's only my own little opinion. Is kind of we all trying to solve problem when the new technology helps and new data come to us, and spatial and time cost, temporal data is increasing, and we're trying to fit on the model. And yeah, you can read all these things, and interpolar network, we believe, and repeat today's morning's talk, is another very important question to go. And I'm happy to, if everybody joins discussion, go to a real future direction conclusion here, rather than my simply list. Yeah, thank you. Why don't you stay up there? All right, so those are our slides in our presentation. I'll leave these last questions up in case people want to spur, and I think I should invite my fellow group members up here for the discussion.
So we started this session 5 minutes late,
Yep.
so we're going to go over discussion until 3:05.
Sounds good, thank you for comping us that extra 5 minutes. Are there any questions from the audience or comments? Yeah.
I have a clarifying question to Lenore, if she's there, mainly for the sequence analysis.
Yeah, Lenore, are you there? Can you hear us?
Okay, Lenore, I have a quick question for about using sequences for the predictions, especially going between the species. I just wanted to clarify, whether any sort of multiple sequence alignment was used, or just using the individual sequences, and what do you think interplay between using the single sequences versus multiple alignment would be for the problem?
Oh, that's a great question. So the current versions of both Descript and Topsy-Turvy only use single sequences. They do not need a multiple sequence alignment. It's clear to me that if we did incorporate a multiple sequence alignment, we would do better, but we were really, really laser focused on things that were fast, and so we can run them genome-wide. But we're definitely going to look at multiple sequence alignments in the future as well. I will say, though, that saying that it doesn't use multiple sequence alignment at all is a little bit disingenuous, because it is starting with a pre-trained protein language model. And those individual sequence models are certainly leveraging everything we know about protein sequence space, including multiple sequence alignments in some way, but it's a very second order effect, right? Your input is just the single sequences.
Why don't you first and I'll do? Yeah. Yeah.
So, now that we've had this hypergraph, we started off with hypergraphs and then hyperpathways. I know that for hypergraphs there are centrality and completeness and so forth. How much of this can be applied? We thought about going there. I feel beyond that, my brain is a little confused because I have trouble interpreting hypergraphs. But are these algorithms kind of the combination of network structures, of node weights, kind of usable in a hypergraph context?
I can take one crack at this, and then we should ask Anna and Morale from the hypergraphs perspective. So we have one paper on hypergraphs where we actually started with this question, where we said, oh, let's try to do something like network propagation on a hypergraph. And we started with the simplest form of the random walk on a hypergraph, which was your random walk's really on the edges. It's an undirected hypergraph. Sorry, your random walk's on the vertices. So you start at vertex, and then you choose a random hyper edge. And then among the hyper edge, you choose a random vertex in that hyper edge. And that's your random walk. Very natural thing. And there's a weighted version of this as well. You can choose your weights according to the weights of the edge. And what we discovered is that even though that random walk has been used in the literature for hypergraph prioritization and machine learning, was that random walk actually was equivalent to a random walk on a simple graph, a non-hypergraph. And so it wasn't actually encoding higher order information. It was actually equivalent to just different weightings of this pairwise graph. So we ended up writing a machine learning paper. It was at ICML in 2019 on this issue, saying, hey, some of these hypergraph representation learning papers that use this particular version of the random walk are not actually modeling higher order interactions at all. So one has to be careful. And I think if you do directed hypergraphs and you do the random walk, what we showed is that if you do the random walk in a different way, where you actually sort of have weights on the vertices as well, then that couldn't be modeled by a random walk on a graph. But it's a subtle thing when you get into these propagation approaches and how you're supposed to sort of propagate information on the hypergraph, if that's what one wants to do sort of the equivalent prioritization.
Yeah, I would add one point. So if we only do the propagation on the regular graph and hypergraph of mathematics, yeah, I think no.
Yeah, in certain contexts, you have to.
Only for propagation, not for others.
Yeah.
It's that technical point.
Yeah.
But isn't that confusing in the sense that there are theoretical results that say that you cannot get the same cut between hypergraphs and graphs?
Yeah.
And these propagation techniques sort of depend on the cut. So how do you reconcile on this thing?
Yeah, so it all comes down to the specific version of the random walk you do and how that's modeled by cuts. And there's a whole literature on this. There's a literature in, what do you call non-hypergraphs these days? Just regular old graphs? What do we call them? When they're simple graphs? I mean, there's,
not simple.
But not simple, because that means something else. So non-hypergraphs, just pairwise graphs, the old, yeah. So there are those parallels, and there's a literature on that in binary graphs, pairwise graphs, and where things were. In hypergraphs, the literature is more complicated, and the correspondence between the random walk and the cut is a little more subtle, and depending on, there's all these different ways to do the random walk on the hypergraph, and that was sort of some of the challenge. So the theory is much more complicated.
Why don't you, Gianna, and Anna?
I have a question for Ben and possibly Wayne as well. So once one gets scores on nodes by combining the data and then identifies a subnetwork, typically how, to my understanding, methods are evaluated is enrichment of nodes in the subnetwork in something. But say that the underlying network between the given set of nodes is a tree. You can identify all the edges or any subset of them that connect all the nodes. How do we evaluate subnetwork rather than just set of nodes in the subnet?
I think most people evaluate the nodes in the subnetwork. And hardly, I mean, yeah, in a sense, they're sort of equivalent because the nodes induce the subnetwork they induce.
If you think you do subnetwork is logically important, but
Yeah.
say that the actual new subnetwork is a tree. You can take any star or cycle or anything and it's different subnetwork, which shouldn't be all equivalent to each other. So
Yeah, well, so what I think what maybe you're pointing out is another version of like how identifying the subnetwork, altered subnetwork, is different from identifying the nodes in that subnetwork. Because sometimes what you really want to think about is identifying the nodes and the edges, right? Because it's not induced. To my knowledge, at least, and like, and you guys should correct me, I don't know of anyone that in the kind of propagation sort of these type of communities actually makes that distinction, because the algorithms themselves mostly are not making that distinction.
So one possibility might be to take edge annotators. For example, you know that an edge is a part of the pathway and use those for rather than not.
Yeah, I just also, just to follow up on Ben's thing, I think it depends if you think about it as subgraph identification, or if you're thinking about identifying signaling pathways or things like that. And so in those fields, people do think about whether you have the right edge or not, right? Because it's representing some kind of causal relationship.
And just quickly to add to this, often you will have weights on the edge, and then those weights heavily influence propagation.
Yeah, sure they do. I guess what I'm sort of was the point I was trying to make was that most of these methods, when they propagate, it's really just vertex. They're just changing the vertex scores, right?
So they can output non-induced subgraph, but they evaluate the nodes.
Most people evaluate the nodes. The question is, could you pull out a subset of the edges? Like once you've sort of decided that you want this set of nodes based on your ranking or based on whatever you do, like some sort of post-processing ad hoc thing, I suppose you could, in a way, pick a subset of the edges based on these vertex scores, based on the original weights of the edges. And I should clarify, probably someone does do this because the literature is
I
vast,
think
but
it's
it doesn't come.
right? So if you're trying to determine causality in these networks, like how does a signal propagate through the networks in parallel at the edges? So if you're trying to identify, sets of proteins associated with some process, then you don't care so much about the edges.
Yes.
And so I think depending on the application, people look at different things.
Sorry.
So my question is how about PageRank info? Like you do PageRank when you move to the setting from the feature, it gives you a subgraph zone. So it's like a random move with feedback every time. It actually not you with the ranking list, but it actually gives you a subgraph around.
That's the random walk.
Based on what? Because it just.
So it's like in the pathway PageRank.
Yeah.
And it gives you the stuff. Only start from the feed. So what is that eventually gives you around that?
Well, how does it give you a subgraph? It'll just, like any sort of version of network propagation, it'll just give you some new scores on the vertices.
Yeah.
And then you have to interpret over
Now
that.
we have the information to give them a cut. So based on the propagation code, to give you cut. The thing we start from the node, so that cut itself will be some stuff. I think it is.
I mean, yeah, sure, there was, I think there was.
I'm going to tie a couple threads and then lead to a question I have for Ben. So I want to mention context is important. So when we think about signaling pathways, we're very interested in Tiana's question of, did we select a reasonable set of edges? Max flow things and sinometries, that whole family of methods, it really does want to select edges sometimes. And we've struggled with trying to evaluate that in new systems that we don't have a tag pathway or some gold standard to evaluate against. And we started to poke around looking at like what are the types of edge topologies that are completely unreasonable, because we saw this morning how we can characterize topologies maybe with like these graphlet count vectors, a subgraph count things we saw. And sometimes I can say like if I have a star graph or I have a clique, some of those things are very unlikely to be what I expect my signaling pathway topology to be. I think there's probably ways to rule out really bad things. I'm interested in trying to figure out better and better, like leading to what Ben was talking about, what is the family of subgraphs that's plausible in biology or in signaling? And I guess the thing I struggle with is it's probably something that's really difficult to characterize. And you were listing a few options like connected graphs. You know, those are fine, we know how to work with them, but I would bet there's some awkwardly shaped family that's probably realistic. And I just have no idea how to work toward defining that.
Yeah, and to be fair, I mean, the ones I listed were just ones that people have used and could be written down. And I completely agree that probably the correct one is some weird thing, but then, you know, then there's some strange circularity, right? Because if you can never design an algorithm to, I mean, maybe you can, it's the algorithm has some topological bias that is representative of biology, but you just don't know what that bias is. You can't sort of write it down. Yeah.
I can only half hear the questions, but I would add that in many domains, we have so many missing edges that it's a little bit dangerous to be making topological conclusions, or if you're going to make strict topological conclusions, you should always remember you're seeing a sample of the network that is not complete in almost all cases.
Comparing network analysis and also maybe a list of network alignment. So when we use network alignment across species, the goal was to find resolve and divergent network. I was curious, like if you have networks across individuals or conditions or whatever, how do we actually go and compare easily? You know, so we could like compare the networks and say, okay, that's a but how do we get to like higher order or like
Yeah, I can try to take a crack at it, touch a number of things. One, comparing species, networks in different species is going to be different than comparing networks, say, within the same species, because different species will have different sets of genes or different sets of molecules. When you're in the same species, you know, the issue of alignment is a little easier in that you have at least the same set of nodes, and you can find the difference in the edges. But like you said, once you get those differences in the edges, kind of what do you do with them, especially in inferred networks where they're very noisy and you don't have a lot of confidence in a lot of the edges. So what I often see people do is they take those different edges and they often reduce them back to the nodes somehow, right? So you find a set of edges and you say, okay, here's differential, you know, the set of edges are all tied around this hub protein in this one network and not in this network. And so maybe it's important in the disease context versus not. There are some algorithms I know of to look at differential community structure, including non-inferred networks, to find, say, differential modules in a very robust way. And then again, how do you evaluate that? You say, here's a community that's in one network but not the other, or shared between the two. You tend to still reduce those to pathways and do enrichment. Does that kind of answer your question?
I'm looking for like ways
Yeah. Yeah.
Dynamics of network, and within the same species, but try to find like higher order differences.
Yeah, so it's a great point. So what we see in ML literature in the last 2 years, there are papers published that focus on embedding subgraphs, but they are more or less focused on which is a subtle problem, which is you start with a graph and a set of subgraphs that you think are interesting, that you found somewhere, and then you can embed those subgraphs. So that is useful to the extent that once you embed them, you get this condensed data summary, so they become easily comparable to each other, because then you can compare those vectors.
but it's not solving the problem of can you take a graph and can you find those interesting subgraphs yourself? That would still require you to like, either exhaustively enumerate subgraphs or use random walks or some other clever techniques to find interesting subgraphs before you can embed them. So I can think of a handful of papers that are trying to find or essentially come with a differentiable counterpart of what an interesting subgraph would look like. Essentially come up with an approximation of an objective function that approximates a subgraph that is overrepresented in the graph, and then declare those subgraphs to be interested. But in that regard, it's always a problem to define what is an interesting subgraph. And we go back to lots of questions that were mentioned today. So it's really not, it's not a magic bullet to solve those things, right? It's primarily about, well, if you understand the problem well, perhaps it's really hard to come up with differentiable approximations to this problem. But fundamentally, it's still about like deciding what interesting subgraphs are. It's a bit more challenging than embedding subgraphs themselves. That's quite advanced because now we can essentially what a neural net would allow you to do is to find good aggregation functions. Ultimately, it's still about like often learning these embeddings at the node level, but then integrating them in a clever way, not by simply taking an average of node embeddings or edge embeddings, but perhaps upweight, downweight some of them based on other types of data or based on topological measures.
So in terms of subgraphs, there's a lot of stuff we're trying to find interesting subgraphs in terms of enriched subgraphs, mostly based on enumeration. And so whether that's like just topological based patterns or, you know, as I mentioned, combinations of topology plus some. And I think actually the hard part there is setting up your background null distribution about what's interesting. So like I claim that if you're looking for higher order structures, that you should control for the frequencies of the lower order structures. So that's actually really hard to do. So how do you know, look at a subgraph and say, you know, I want to make sure that all the triplet combinations that I saw, that distribution is maintained in the graph. You know, bigger subgraphs, both like an enumeration issue, plus also an issue of how do you set up your, you know, your background model.
Can I make one quick comment, like a terminology thing that would be great to sort of resolve in this meeting? There's higher order structure. And so I'm always confused whether higher order structure is supposed to mean a hyperedge, meaning it's an interaction that's more than two things, or a clique of three nodes could be higher order structure.
Both.
Causing all the members' attention is we often talking about overrepresentation when we infer some knowledge from the networks, right? And that's a little biased based on the data available to us. Currently, measurements is measured mostly the stable things, right? And also the overrepresentation means the critical signature pathway is stable from earlier state to later state, they're shared. However, biologically, they are more interested in who is a small number of them play the role to decide the future, or the small one you can translate the people's response to some drug, something like that. They're not interested in the overall, the genome background, who is shared. They're interested in this typical, very specific one, which might not have the feature of overrepresentation in the data we currently observe. But how to solve it? I mean, I hope you know. I hope everybody here expects in computational field can help me. Yeah.
So let's do one more question, and then we're gonna start the coffee break.
Just really quickly, you know, Mona, Tommy, and the speakers talk about virtual networks and talk about different from people, person to person, and different conditions. I just want to add one thing is it's touched upon, but make it even more, I guess, obvious. It's just, but when we look at two people, they don't have one. You know, each person don't have one network because different cell types will have, but all the different networks. So I just think that something should be. I just want to mention that because yeah, I think that's very interesting. We just have made a recent way looking at classifications. By looking at specific cancer types and construct a network in that cancer type that have a better performance than just taking overall and analyze all of that. Yeah.
Yeah, we just need so many samples that are individual specific and single cell specific, and yeah. Well, thanks, Kimberly and the team. We're gonna have 24-minute coffee break, and we're gonna start at 3:30 with the final session for the day.
Anais, did you have a question?
No, it was clapping.
Ah, nice, good. Yeah, on Zoom, I think they forget about us sometimes.
Yeah,
I had a lot of
somebody-
trouble hearing the discussion.
Yeah, I cannot hear anything, which is like frustrating because you spend all the answer trying to get the question. So
Yeah, no, the person at the podium should repeat the question for us.
yes,
They're not hearing
Someone in the room, other than just AV tech, should be on this call to monitor it. Tiana, maybe.
Maybe send her a message and ask if people could repeat questions more in the mic. I think that would be very useful, and I think it would be easy enough.
maybe they have a mic to circulate. Could also be.
That would be even better. Yeah. All right, I'll be back in a couple minutes.
Yeah. See you.
Interesting. Yeah, I think that's going to change. So it looks like we're going to get back to the meeting now. So nice talking to you, everyone.
Well, it's time for the last session of the day, because we deserve the break that comes after that. So the next topic is machine learning on graphs, and Arjun's gonna coordinate that.
Thank you, Taylor, and it's been great so far, and to listen to all these topics. And many of these topics I've had not worked on for many years, and it's good to listen to all these things and refresh my memory. In our group, we're going to be talking about machine learning on networks, and then hopefully I'm obviously going to say many things that are going to be related to many other things that we've already talked about today. And in addition to the people here, Sardar, Yang, Byunjun, Haiwan, I'm also probably, I should have added Marinka's name here because I'm going to cite much of our papers also. So with that, I want to, here is what we're going to do. This is a lot, so we're going to see how far we can go, and then based on that, hopefully, cut some of the last parts. I'm going to introduce a very general framework to think about machine learning on networks. Not going to go into pros and cons, what is better and worse, and all that stuff. I'm not going to talk about that stuff. And then we're going to talk about unsupervised and self-supervised training, condition-specific embeddings, and doing embeddings on heterogeneous graphs. And then Byunjun and I are going to talk a little bit about transfer learning, and then Haiwan, and then Yang, we're going to talk about geometric networks. And then we're going to stop at that point, take some questions, and see how we are doing this time. Okay. All right. So the idea of, so just to introduce this idea of machine learning on networks, I want to begin with this very simple overview that all of us, I think at some point or the other, are interested in the fact that there is some graph of nodes and edges. And then we want to do some task, classification task, right? Maybe classify nodes into discrete labels, or give a continuous number to it, or classify subgraphs or whole graphs, and then or do community detection, link prediction, or do graph alignment. These are problems that all of us heard during this day. And the traditional idea is to take this graph and then do feature engineering by hand. We basically figure out what is interesting about this network, or for example, each node, maybe its degree or its graphlet spectrum and all that stuff, and use that as input to machine learning algorithms and potentially make these do these tasks. What instead might be a good idea is to actually take this graph and then learn something like a numerical representation for every single node in the graph. What numerical representation essentially means is a vector of numbers. That single vector represents every single node, and that vector uniquely captures the structural or topological location of this node with respect to all the other nodes in the graph. And if we are able to learn this structure, then we can use traditional machine learning algorithms to do any of these problems potentially. The way to think about this in a very general way, what we can do is to design something like an encoder-decoder system where we have an encoding function. This can be whatever it is. In a way, I'm just talking about this in a very general way, that learns this numerical representation. It's one vector for every single node in this graph. Okay. And then we train a model that takes this graph as input, learns this intermediary embedding vector, and so that you can do this decoding to recapitulate its a node's position in the underlying graph, or maybe some other label that we have on top of every single node. If we do that, once we are done with this part, we can take this embedding vector, and that tells us some kind of a numerical encoding for every single node in the graph that we can then use as input to ML algorithms. Okay, so generally, what all these algorithms are doing is to minimize some loss like this. What they're essentially doing is that if you take the embedding vectors of two nodes I and j, that's zi and zj, and then run it through this decoder, and then we have some sense of how close those embedding vectors are. And if how close two nodes are in the embedding space and how close they are based on the underlying graph, if that difference is as low as possible, then we have learned a good representation of all the nodes. How this distance of the original graph is defined, how this decoder is defined, and what this loss function is the way in which all these different methods vary. For instance, there is this class of methods called matrix factorization methods. And what they essentially do is to say, once you have an embedding vector for a pair of nodes, you can combine them in two ways, maybe do a dot product or whatever it is, or calculate a Euclidean norm. And then that will be the way in which you're going to decode the embeddings in terms of a distance between two nodes. And then we're going to define the distance between the two nodes based on the original graph in many ways. For example, you can just say two nodes are connected to each other based on the adjacency matrix. Those two are close and otherwise not. Then you can define a loss function that says how far the distance based on the embeddings is from the based on the original graph. And then minimizing that will essentially give you a way to optimize the embeddings. So that's the matrix factorization ones. The ones based on random walks essentially say that we're going to define the distance between any two embedding vectors in the following way. We're going to say that we're going to do random walks on the original graph. Either they are straight up random walks or they are like biased random walks. And then once you do that stuff, what we are essentially asking is the distance between any two nodes is that if I start a random walk on node I, how frequently will I reach node j? Okay. And if that number is high, we want the corresponding embeddings to also be very close to each other. And that's what methods like deep walk and node2vec optimize for. Okay, so some problems is that methods like this do not actually share parameters across all the nodes at the encoder phase. And second is that this does not take into account any additional information we might have about nodes. For example, let's say the expression or activity level of all the nodes in the graph and stuff like that. And also they are intrinsically constructive. It means that once we have learned node embeddings for a given graph, we can't predict the embeddings for a node that has never been seen during training. So there are approaches to sort of address all these problems. And that is by way of using graph neural network models. And the idea of graph neural networks comes from the idea of convolutional neural networks on images. The fact that, the fact that you don't want to treat, for example, all the pixels in the image as totally independent of each other. There is some spatial relationship between all of them. So you can think of this as a mesh graph of all the pixels. The same way we have nodes, and then they are all arranged with each other based on the underlying graph. And there's been a lot of development where people have sort of adapted this idea and then turned this into message passing. And then some of the latest ideas is based on graph attention. Okay, so the idea here is that one simple way of thinking about deep neural network models on underlying graphs and to use learn node embeddings is this idea that you can basically train an autoencoder model, for example, that takes the adjacency vector of any given node and learns a low dimensional representation so that it can be reconstructed back. Okay, and then that low dimensional representation is a node embedding. That's not a bad idea. However, this doesn't, again, take into account, it's still transductive, and you cannot do this for any new nodes and all stuff, and it doesn't take node attributes into account. So the more modern idea is to say that you have an underlying graph, and then you do this combination aggregation based on the network structure in a recursive and sequential way, so that you can keep updating the embedding vector of A over time, and what you learn over time is the best representation. Okay. Let me just say one small thing about this in the interest of time. Again, all the graph neural network based algorithms all fall in roughly into this very general framework. And what they essentially differ on is how this aggregation is actually done. Those aggregation functions are different for different neural network models. And then how these weights are actually shared across all these nodes. These weights are actually learned during a time. And this aggregation combination. So these are the ones that are actually different across these different networks, different algorithms. And okay. Maybe one other thing that I want to say is that in the simple case of graph neural networks, what we have is that every single, let's say we take a single node at the aggregation step, what happens is that you sort of treat the value of, or rather the embedding vector of all its neighbors sort of equally. But that itself is an assumption. So you can sort of say, okay, maybe you want to build another neural network model that says how to weight the contribution of all the nodes of all the neighbors of a given node, and then how that's actually aggregated to update the embedding for a given node. That is called attention, and that's been proposed for graphs. And then finally, this is my last slide of this introduction, which is that in addition to up to this point, what I've essentially said that you have a graph, and then you can ultimately learn an embedding. But what if you not only want to generally learn an embedding, but you want to learn this embedding in the context of a supervised classification task you're interested in? Let's say you want to do, let's say, disease gene prediction or function prediction. In that case, what you also want to do is to add another loss function that basically says how well you're doing with this specific classification task. Add that to your embedding loss as well. And then you can learn this in a supervised way. Okay. And then Yang, you are the next.
Thank you, Arjun. Okay. Okay, thank you. So, so as Arjun have introduced the concepts, and the first topic we want to talk about, which covers about 20 minutes, is the embedding. This topic has already appeared in this morning. I feel like it has a lot of interest in our group. So let's spend a little bit of time on this part. So I will talk about the first part of embedding, which is the unsupervised or self-supervised pre-training. I'll explain what I mean here. So far, Arjun has shown that you can use matrix factorization or random walk, right, to learn the network embedding. Then recently, we can use the deep learning graph neural networks with the help of the labels. We might be tempted to use supervised learning for graph embedding because we feel like it's learning not only context dependent, but also task specific embeddings. But the problem of such a supervised approach is apparently the curse of the labels, right? Especially in our fields, in biomedical or biological discoveries, the labels are very expensive, right? And rare. So if we recall what happened in computer vision in the early days, they can use crowdsourcing to label images, right? Here, it's really hard for anyone, right, to mark the tumors on the brain image. But you see, very often from reCAPTCHA that you are asked to label a traffic lights, right? So it needs a lot of domain knowledge in our field to generate such label data. Not only the amount of the label data is rare, But also in our field, label data can be very context and procedure dependent. So speaking of how two proteins bind to each other, you could have different kind of assays to measure them. And then each assay have different conditions and so on. So it's very subtle. So, but the challenge is more than just that, the label. The additional challenge is from how we want to use embedding, what kind of embedding we want and how we want to use them. So if we do even manage to use a small amount of labeled data to learn the embedding, there are two issues that are central to our use of the embedding. First of all, it's really hard to generalize. So if you want to explain what we have learned from the embedding through visualization or other methods, or you want to transfer the embedding for a different task, it's very hard to use those embeddings to generalize. And the second thing is the robustness. So we have a lot of noises in our data, not just the labels, but also in the graphs themselves. So under such uncertainty, it's a very poor embedding that we can use from the supervised approach. So if the supervised learning is not the answer, just like people have already called that the next revolution in AI is not supervised, then where's the answer? Well, we can all guess if it's not supervised, then it's unsupervised, right? So I'm going to talk about self-supervised learning, which is, just like unsupervised learning. It's overcoming the bottleneck of labeled data by not using labeled data. Essentially, we're using unlabeled data, and what's even better is we have massive amount of unlabeled data. We don't have a lot of protein sequences with structures determined, but we have so many protein sequences, right? So in this case, what is self-supervised learning? It's a branch of unsupervised learning. It's trying to use part of the input itself to predict the rest. And in this example, you can see that this is an image example that we can design some so-called pretext tasks, which only involves the input itself. And then the encoder can learn some parameters, and that can be the specific form of the knowledge transfer to the downstream task. So this is the concept of the pretext versus the downstream task. So specifically, let's look at what is the pretext tasks. I want to show you from the left to right are three domains circled. One is a computer vision, CV, one is a natural language processing, NLP, and one is graph. So actually this pretext designs or self-supervised learning started with computer vision and NLP. So you have top and bottom rows representing two styles of self-supervised tasks. On the top, you have the so-called predictive kind. And as you can see in one example, right, we start with the image, we make it black and white, and then try to predict its color, right? And in the bottom, we have the contrastive kind of methods. So we start with images of different fruits, but then for each image, we generate a new view, or we augment the image by rotating or do some other kind of perturbations. And then we construct positive pairs and negative pairs. We assume that the different banana images are positive pairs, but across banana and apple is a negative pair. And we try to learn embedding to make positive pairs close in the latent space, but the negative pairs far away in the latent space. So the same idea has been propelling the natural language processing through the so-called masked language prediction, right? Masked language modeling. So these trends have recently rippled to the graph field, and I'm gonna show you two types of methods in more details, but the fashion is very similar. So for the first kind of predictive models, right? So these are two kind of examples, right? One is graph reconstruction, just like the name suggests. You try to hide part of the graph and predict the rest of it. Here you can hide part of the nodes or edges, and use the same encoder decoder architecture that Arjun has shown. And then this is one type of methods. You can also collect something that's in the input data, but not exactly part of the input data. For instance, you could summarize the topological clusters or communities in the image and then try to ask the encoder-decoder system to predict where these nodes belong to different clusters and so on. So you could use this input data itself to design different kind of pretext tasks for the self-supervised learning. So this is the first type, predictive. In the second type, self-contrastive methods, I'm just pointing out two important factors of contrastive methods. First is how do you generate the augmentation or views of the original graph? So apparently in the middle panel, you see three kinds of perturbations, right? Involving the node attributes or topology or sample perturbation and so on. And the second factor is how do you contrast the positive versus negative pairs? And you could use different objective functions to pull apart the two distributions. And in the end, I want to say a little bit about the challenges and involving this self-supervised learning. So first of all, extending from images and languages to graphs is a very non-trivial task for self-supervised learning because the data structure is different. Unlike images, which are grid data, here we are facing non-Euclidean data, and we have all kinds of things, we all call them graphs, but they are abstractions from diverse natures. So it's very non-Euclidean and heterogeneous. And in the open challenge is that, first of all, for the predictive methods, how do you design the predictive tasks? There's not a lot of universal framework about it, and people are using different kind of methods. And then what about the contrastive methods? What kind of view do you generate, right? So we try to optimize view among a pool, or we try to even optimally choosing a view in a continuous space by graph generation, but it still is an open challenge and a lot of things ongoing. And lastly, how do we better incorporate our domain knowledge into the design of the self-supervised tasks? And that's very important. So essentially, these self-supervised tasks are ways we incorporate or impose our human information into the prior of the graph. Of course, there are a lot of other topics as well, including explainability, which will be briefly touched upon as a separate topics. And then you might have other more complex graphs that my two colleagues will talk more about, Arjun and Serda. Thank you.
All right, I'm going to briefly touch upon our next topic, which is on building condition-specific network embeddings. And the idea is that we not only want to say that all of us, we discuss a lot about dynamic graphs and the fact that graphs are not static. There are potentially, the graphs are varying a lot in terms of various conditions, both in terms of node membership and edge membership. So the idea is to say, now, why don't we assume that we not only have a single graph, but we have some notion of a context, and then can we incorporate that into network embedding? And I'm just going to at a very high level talk about some ideas. So one is that, for example, the best way, one good way to think about context-specific network embeddings is to just begin with context-specific networks. So for example, if you're able to generate, for example, we generated a long stream back gene interaction networks that are specific to different tissues in the cell, in the human body. And then Marinka basically took this, did a really nice work taking these networks and then incorporating that into a single model where every single layer here, for example, corresponds to a specific tissue network, and the nodes are individual genes. And then there are connections between the layers based on how similar these tissues are, based on anatomy and physiology. And the goal is to optimize learn embeddings so that you optimize two things. One is that you want nodes within a given layer that are structurally similar to have very similar embeddings. And you want the nodes across the same nodes across layers to have very similar embeddings if those layers are also pretty similar to each other at a high level. And you want to optimize both at the same time. And then they did this nice work, and then you were able to get one embedding per node per network. And that's been shown in that paper to be useful for function prediction and stuff like that. What if you don't have one network per context to begin with. So what we are exploring now, for example, we are sort of wrapping up this work where we assume that we have a single network. But then for us, context information comes from the fact that we have some score for every single node that is being produced experimentally. For example, you can think of this as a bulk or a single cell RNA-seq profile. And then the way we handle this is to say, we're going to take a single profile and use that to modify the edge weights, and then use that to influence the random walks that essentially are, build these node embeddings. So therefore, we end up with context-specific node embeddings, and we initially see that these context-specific embeddings are much better than the original embeddings that are devoid of this context in making meaningful predictions. I also wanted to highlight another figure from Marinka's review. And this is not exactly context-specific network embeddings, but I'll talk about how it is presented here. So which is that, Marinka, correct me if I'm wrong, okay? So the idea here is that there is an underlying graph, which is static. And then let's say you have gene expression data. Let's say for every single cell type, it's not like gene expression, but let's say a set of genes that are enriched in that particular cell type. So what you can do is to do the supervised node embedding, which is to say you take this entire graph and then learn a network embedding that maximizes the ability of the embeddings to predict whether a gene is expressed or enriched in a particular cell type or not. Okay. So that is the use case that is presented in this review, but I thought this could also be used to actually generate context-specific network embeddings, where you can even think of this vector of which cell a particular gene is present in as node attributes, and then use that to potentially even generate context-specific embeddings. So that's something to think about. Okay. Sarah.
All right. So in the morning, when we discuss about multimodal data integration and knowledge bases, we talk about different data modalities, and multiplex heterogeneous networks are good ways to represent different data modalities. So you build one graph where you have multiple types of nodes, multiple types of edges, and now the problem becomes, okay, can you do graph representation learning on this complex graph? And there are different application domains. This could be a recommendation system where you link prediction between customers and items, or drugs and the genes, and different things. So there are different names. Multiplex heterogeneous network is one name. Multi-view networks, heterogeneous information networks are some of the names that you can see in the literature. But essentially, it's total number of node types and edge types is greater than 2, then we're in this territory. Again, when we do machine learning, we can categorize into 2 main groups: node classification and link prediction, and drug reproposing, drug side effect prediction, gene prioritization could be some of the examples of node classification. Drug target prediction, recommendation systems, fraud detections are some of the applications that are in the literature for link prediction using multiplex heterogeneous networks. So as in the regular graph representation learning, there are 2 main streams, shallow learning and deep learning. And the disadvantages are really similar to what Arjun explained. And so I'm not going to go too much into detail. For the shallow learning, I'll give some examples. So there are 2 main steps. One is how we're going to define the paths for the nodes that we are trying to embed. And as we discussed several times, we do random walk based methods. And basically, when we do random walk, we could do, starting from a seed gene, we can visit other nodes in the same layer, or we can jump to the other layer. So it's a random walk with restart on multiplex heterogeneous networks. We can also define method paths. Where we're doing, again, random walk, but we are particularly careful about what kind of nodes we visit. So on the graph on the left, so we can define a similarity between 2 author nodes based on their co-authorship. So we can visit paths starting from author, go to the paper node, and then author node again. Or we can look at the author similarity in the context of presenting at the same venue. So then our meta path would be starting from an author, going to a paper, going to the conference or journal that paper was published, going back to another paper, and then another author. So this kind of paths can be defined. And then the next step, we can apply like the skip-gram algorithm that was first introduced in Word2Vec, and then Node2Vec was applied. There is heterogeneous skip-gram algorithm where the idea is at the output layer, you categorize them based on the node type, or you can define a similarity probability distribution on the graph and also the embedding space, and you can compute KL divergence between these two probability distributions to optimize your embedding. But the limitations are, again, as we discussed earlier, there are node attributes that we could utilize, and shallow methods cannot do that. Meta path definition requires some semantic understanding of the graph. And if it's a more abstract graph, then it's hard to do. Number of nodes and edges could be limited depending on what method you use, or the edge weights could not be utilized. But more importantly, as Arjun mentioned, these are transductive methods. So every time you have to start from scratch to learn an embedding. And there are some hyperparameters, like what will be your jump probability or restart probability. It's hard to tune those hyperparameters. So the deep learning methods can overcome some of those issues. I'll just talk about some kind of historical perspective on some of the methods. So heterogeneous graph neural network is one of the first approaches, applying GNN on heterogeneous networks. So the node attributes, there's an aggregation for the node attributes, and node attributes in this method are coming from different data modalities. Like it could be image, text, so on and so forth, so they can be combined. And also you do aggregation at the node type level. So you learn embedding at each node type, and then you combine them together. So again, Arjun mentioned about graph attention networks, and the same of that are applied to heterogeneous networks. So heterogeneous attention network is another method. Again, we do aggregation. Aggregation is done at the meta path level and at the node type level, but then you compute attention between the nodes when you aggregate them, and then you also do attention between the meta paths in this method. Another popular method is GATNE, which has two versions. So there's transductive version, but there's also inductive version where you learn a transformation function so that you can apply that function on a new graph that's never been seen. Again, there is attention applied, and the optimization of the embedding is done by using skip-gram algorithm, like in MetaPath2Vec method. And the final example that I'm going to give is just published recently. This is a self-supervised method. So there's contrastive learning, as Yang mentioned. Contrastive learning applied on heterogeneous multiplex graph. Again, we have the multipaths introduced here. We do graph sampling based on those metapaths. So we could call them metagraphs. And we do aggregation again for each of the metapath. Separate aggregation is done. So they call this intra learning, intra-contrastive learning. And also between the different semantics, you do aggregation. So it is inter-semantic contrastive learning. So they're all combined, and you have a final loss function to learn this embeddings. Okay. So some of the challenges, some of the open problems are, again, here we also mostly rely on method paths or method graphs, but understanding which paths are relevant really depends on the semantics. So we might want to focus on network motifs as well to be able to apply this on more abstract or graphs where the semantic information is not readily available. There are also dynamic networks, as we discussed in the previous session. So like networks that change over time. So how we could apply these methods for dynamic networks. And like GNN, we know that GNN suffer from over smoothing problem. Is this same thing happening for the heterogeneous networks? That needs to be studied a little further. And something that we also, we're gonna talk about explainability a little bit, but there's also how to build those models in a fair fashion, meaning that you utilize some node attributes, but those node attributes, like there will be some sensitive node attributes as well, like demographic values. And if you don't have enough sample data, you might build a biased network, maybe biased to different race or different sexual orientation. So that learning them in a fair manner is also an important future direction. These are the papers that I cited in the slides. So I think Arjun or Arjun? Oh, okay, you're here. Oh, great.
Yep, finally. That was the last person. Very nice meeting you. I will talk about transfer learning just a little bit. I think transfer learning techniques could be a very useful class of machine learning techniques that could, make big contributions to, network biology in the future. I'll spend the next 2 slides on, oops. It's not this one. Oh, this one. Okay. Oh, great. Yeah, so many of you may be already familiar with transfer learning, but let me just briefly summarize. Transfer learning aims to take advantage of the learning outcomes for a specific predictive task in a specific system, and then try to apply the learning outcomes to a different domain or different system or different predictive task for the same system. Okay. Why is it important? Because, well, everyone that works in computational network biology knows that, well, we already, don't have enough data, right? And considering the complexity of the biological networks that we have to deal with and the predictive tasks that we want to do, well, how do we solve this problem of having insufficient data, especially for non-model organisms, for example, right? One thing that we could aim to do is, well, make the best use of existing data and see how much it can be transferred for different predictive tasks or different systems. And that's what transfer learning is trying to do. And in a sense, at a high level, we've been already trying to do that as computational biologists, right? Well, make the best use of what we already know, and then try to apply it to different systems of tasks. And machine learning could be, well, used to do this in a more rigorous and formal manner. Oops. So you may have heard about domain adaptation. It can be viewed as a specific type of transfer learning, where we make some assumptions about the feature space. For example, there may be a space of features, and then you may use those features to predict something or do some kind of analysis. And when you think about the same feature space, however, with a different distribution of the features, you typically say that you want to adapt the domain, right? So it's called domain adaptation technique. Transfer learning is conceptually a bit more general, so you don't necessarily have to assume that the feature space is identical, okay? So how can transfer learning be applied? For example, it may be used to answer the following question. For example, suppose you have found biomarkers, okay, for predicting something that is of interest to you. And then can you use the same set of biomarkers in a slightly different context or to a different system, right? For example, if you have identified biomarkers based on a mouse model, can it be applied to predicting or making prognosis or diagnosis of a human disease, right? How does the gene regulations or pathways studied in one model organism be used to study, well, another organism? can we transfer knowledge in a more rigorous and robust manner? Or how does a predictive model developed for one specific type of disease be applied to another type of disease or another class or another type of the same disease, right? So those are many of the things that motivate the study of transfer learning. Graphs and biological networks can be very useful for studying transfer learning in a more rigorous way. And I think we just started to scratch the surface of what we can do in the context of transfer learning using biological networks. And Arjun is going to give a couple of examples. Let me actually skip this. So the two things that I want to highlight for transfer learning is this idea. Let me just introduce the first idea, which is pretty straightforward that all of us can think of intuitively, I think. If we know how to do network embedding, the way in which we can think about transferring, being able to learn from one network and let's say and predicting on the other would be something like this, which is that you would run a traditional network embedding algorithm on let's say two different networks. Let's say we can take the example that John said. Let's say one network is genes interactions in humans and the other is gene interactions in some other species, let's say mouse. And the way we want to train models in one species and make predictions in the other would be to do network embedding independently on human network, independently on the mouse network, and then do something called alignment of the embedding space. So the way people do this is to say, can we, in this network, can we identify something like a set of landmark nodes, a bunch of nodes that I can somehow assume that are exactly the same for all practical purposes. Maybe they perform the same function. This could be based on, let's say, one-to-one orthologs, right? And then what you can then do is to say, once I have learned a network embedding space for human genes and then ones for mouse genes, you can then take this space and sort of do a linear transformation. And then so that you overlap these two spaces so that the landmarks align perfectly with each other. And that way you've essentially sort of put all the embeddings in the same shared space. And then you can learn in let's say a node classification task, you can learn new machine learning algorithms to classify human genes. Then once that model is trained, you can apply the same model to classify, let's say, mouse genes. The other idea, which is where this is an area that is super nascent, and there are very few methods that have actually been broadly applied. So this is really open for lots of applications. So here, this is at least theoretically the idea in which we want to do everything simultaneously in some integrated way, where we want to embed jointly while maintaining reasonable alignment, right? But this is a pretty complicated task. And then where we want to be minimizing 4 different loss functions. Right? These 2 loss functions, LSA and LSB, are essentially our traditional network embedding loss functions. We want to make sure that the distance between the embedding vectors of very structurally similar nodes in the individual networks are pretty close to each other in the embedding space. This one basically says that I want to make sure that the corresponding, for the nodes that I know are corresponding across these networks, those embedding vectors are as close to each other as possible. And finally, if I want to do this entire thing in a supervised manner, I might also have some labels on, let's say, nodes only in graph 1 or only in graph 2. And I want to optimize the fact that these node embedding vectors become inputs to a machine learning algorithm. You train a new set of weights to make predictions on these node labels, and you want to minimize all these loss functions together. So this is a very general framework, but it's yet to be applied on a large scale to real networks. So this is pretty open. Okay. All right, this is going to be our final topic before our discussion.
Thank you. And before I start, I just want to point it out that actually all the slides are collaborative work between Yang and I, and actually the whole group. And actually most of the slides are made by Yang. So I'm here giving the presentation, but all the credit goes to Yang. So I just want to, we've been talking about the whole day, networks and graphs. I want to mention that a lot of the biological molecules actually have a defined 3D spatial characteristics and requirements. For example, and it's important to know the 3D structure. For example, for small molecules, drugs, right? You have a lot of these stereoisomers, right? So if you look at their composition, they have the same exact composition. Pedro gave another example this morning, right? But in this case, there's really, if you just look at the composition of the different, they really have nothing, there's no difference. But because of this, how this 3D, spatial relationships between them, they are different, stereoisomers. One can be water soluble, and the other one can be poorly soluble, but can be an anti-cancer drug. Right? Another example of this is ibuprofen. We all take ibuprofen, but ibuprofen actually you take have two stereo isomers in the drug you take. Only the S form is active. The other one is just there for the ride. It's not active, right? So knowing this, 3D spatial relationship between all of the different, atoms and things like that is actually quite important. Same thing with protein structures, which is really what we do quite a bit. And Mona also mentioned about this, in her, she does a lot of this work as well. Well, everybody knows about this. Is 3D structures for proteins are very, very important, right? They form this, and we have now, AlphaFold, right? Can make a lot, the goal is actually to predict the 3D spatial structures of this. And of course, often the active sites, right, the highlighted this pocket, the active site has a very defined shape, right? And I'm using the shape in a very general way. Not only this actually 3D shape, but also there is hydrophobicity, all the biophysical and chemical properties you can see, right, as the color indicates, right? So these 3D shapes and surfaces are quite important for protein function and how they, you can see, right, they complement the binding of this molecule, right? So all of these, and so clearly when you talk about, did I miss a slide or something? Anyway, so when we talk about the a very straightforward way is just use the regular Euclidean CNN, right? So you have this filter or this kernel go from 2D, right? So, and you can go to 3D, but the problem is, and that's one of the topics I want to talk about, is the geometric. It's invariant to deformations of the 3D shape you're trying to actually perform the convolution on, versus if you do, so this is using sort of Euclidean distance, Euclidean convolution, but if you want to do geometric, there is a time and place for that. But for geometric convolution, you can see, right, in this case, is the future shapes around the deformation or the structure of this particular subject. Okay, so this is sort of geometric CNN, geometric convolutions versus the traditional Euclidean-based convolutions. And so I think, again, we talk about graphs. Here, I just want to very quickly talk about how do you generate from a, so if we have starting from a network is very sort of natural to go to a graph because a network is a graph. But if we just have a 3D structure, how do we go to a graph, right? And then there are two methods. One is so-called an input invariant, basically using the local coordinate, the relative local coordinates, which is, this is sort of what we do. And this is actually, I was introduced to this by Yang. This is so-called the model invariant or equivariant, basically using global coordinates, using a Cartesian XYZ coordinate. I will explain this using a recent paper. So you can create a graph from a 3D spatial relationships, and then you can do sort of a convolution, right, on this graph, create GNN or GCN, and then generate outputs. So for I would say traditional, so-called input invariant graph, so from 3D structures, you can get all the atoms right in this structure, and you can figure out the distance, for example, between one atom to all the other atoms. You can put we will talk about you can put in even angles and all of that. And you basically create a graph and you can put on for each node, is that each item, you can also put on many other things like convolution, like conservation, biochemical, physical chemical properties of the atom, and many other things, and you can do graph convolution. And as I said, you can consider distance. There are many different ways to construct, to go from a 3D structure to a graph, right? You can consider distance, you can consider distance plus angles and plus dihedral and all of this. And the things to consider is whether the representation, the conversion from the structure to the graph is complete, and how effective, right? All of these things to consider, and how effective is the representation, and whether when you do this convolution on this graph, the message passing efficiency and accuracy, all of these canonical problems. And the last thing I will talk about is this so-called model equivariant or invariant sort of way of generating the graphs. What that is that in the input invariant, however you rotate that structure, it doesn't change the graph and it gives the same output. But in this way, basically, you can think about, for example, you're trying to predict a protein ligand binding, and you want to basically, if the input rotates, your output also rotates correspondingly with it. So you basically can think about, you generate there's an XYZ Cartesian coordinates with this input, and you construct the graph that way, so that if you changes, the prediction also changes with it. There's a paper on this, just published this year. And yeah, so that's, anyway, so I will stop there. And I think Arjun for other emergent problems.
Maybe we just pause and then take questions. That's fine.
Okay, sure.
So we've been strongly encouraged to use the microphone when people are not in front of that one. So just an FYI. People online couldn't hear as well.
I will ask a question about the selection of neural network-based models, actually. So there is graph convolutional networks we use for simple models, but when the problem becomes more complex, we are using attention mechanism like graph attention networks, and then we can even go to message passing algorithms. So I just wonder, what should be the criteria, in your opinion, to select the proper GNN model? For example, there's a paper which suggests the second version of graph attention networks. In that paper, it is the how attentive are graph attention networks, something like that, the paper. And they are showing that with a simple modification, they are improving graph attention networks. Okay, for proving that, they are trying many data sets. But interestingly, for many data sets, they see that the models without attention mechanisms are better than those attention networks. So in your opinion, what should be the criteria to select the proper GNN model? How to decide the complexity, where to stop? I just wonder your thoughts.
Arjun, you want to use the mic?
No, I said that there's probably no free lunch. There is, I think without the context of what the problem is, it's pretty hard to figure out. I think, let me say a couple of things. I think one is the complexity of the model, at least the amount of training data or the way in which you are able to do unsupervised learning, or the number of unlabeled data that you're able to use should also scale with model complexity in some way. Otherwise, it's probably not going to be possible to use more complex models. The other thing that I can say is from our experience, there are many cases where we have been we are trying to use things like graph convolution neural networks and all stuff, and many times even these models are actually, they are baseline models that are based not on the original graph or not based on convolution neural networks, are actually sometimes very hard to beat if the problem is pretty straightforward to actually solve. So I don't have a better answer than this. I'm sorry.
Or there is not enough training.
Yeah,
Data
right.
set is very limited, it's severely limited, right?
Yeah, that's a good question. I don't have an answer, but just want to make the question even more confusing. Because I think for, if you're thinking about GNNs, there are two levels of understanding. One is at the theory level, one is at the practical level. So at the theory level, you wonder how much your networks are representative, can represent